Good morning, everyone. My name is Mulugeta. I'm the Associate Director for Policy and Research in the Division of Pediatrics and Maternal Health at the FDA. On behalf of the FDA, I would like to welcome you to this collaborative workshop hosted by the FDA and the University of Maryland Center of Excellence in Regulatory Science and Innovation, or CERSI, entitled Accelerating Product Development for Pediatric Systemic Lupus Erythematosus. The goal for today's workshop is to discuss the barriers to timely drug development for pediatric SLE and to explore approaches to overcome them. Throughout the meeting, we will focus on several key topics, including extrapolation, clinical trial design consideration, dose selection, and safety assessment. Before we begin, I would like to briefly highlight CDER's role in the review of new therapies. Within CDER, drug products are reviewed by multidisciplinary teams, and the Division of Rheumatology and Transplant Medicine is responsible for reviewing therapies for rheumatologic diseases, including disease-modifying anti-rheumatic drugs and biologic DRDMDs. CDER does not review biologic or cellular therapy products such as CAR T-cell therapies, which are regulated by a different center within the FDA. So accordingly, today's discussions will focus primarily on products that fall within the purview of our center, which is CDER. As you can see, we have a full agenda, and we kindly ask all speakers to adhere to their allotted presentation time so that we have ample opportunity for discussion and audience engagement. And to help keep us on schedule, we encourage participants to hold clarifying questions for speakers until the panel discussions, when there will be dedicated time for questions and discussions following each sessions. We have a lot of participants who are joining us virtually, and to the virtual participants, we encourage you to submit your questions through the online chat throughout the workshop, and we will monitor the chat and address as many questions as time allows during the panel discussions. We are also very pleased to welcome the patients and families joining us today, both in person and virtually. Your lived experiences and perspectives are invaluable, and we deeply appreciate your willingness to share them with us today. We have included a patient or family representative in every session of today's workshop, so that these important perspectives remain central to our discussions throughout the workshop. Although this is a public workshop, we want to emphasize that we really take the input that we receive from this workshop very seriously to advance regulatory approaches that will facilitate the development of timely and safe and effective therapies for children with SLE. Once again, thank you for joining us in person and virtually. We look forward for 2 days of thoughtful discussion, collaboration, and shared learning. So without further ado, it is my pleasure to introduce our next speaker. Mary Tran is the Director of the Office of New Drugs at the FDA, and she'll be providing introductory remarks.
Good morning. Thank you, Lily. It's truly an honor to be invited to give introductory remarks for this 2-day workshop covering many important topics to help advance drug development to treat pediatric patients with systemic lupus erythematosus, or SLE. For nearly 3 decades that I've been with OND, I've seen many advances in regulatory science that have enabled efficiencies in trial design, endpoint development, streamlining drug development programs that have supported approval of many novel therapies to address common diseases, rare diseases, serious and life-threatening conditions. These therapies included small molecules, biologics, and combination products. However, these advances are generally observed first in the adult population in diseases that often also affect children. It wasn't so long ago that we had little to no information in product labeling to inform prescribers how to treat children with these same conditions as adults. FDA, through the pediatric rule in 1994, encouraged sponsors to use existing data, employ PK modeling, or conduct new studies to add pediatric dosing and safety information in labeling. But there was little incentive to include in a drug development program studies to evaluate safety and efficacy of the drug in children. That changed in 1997 under FDAMA, where Congress enacted a law providing 6 months exclusivity in return for conducting pediatric studies. In 2002, the Best Pharmaceuticals for Children Act, or BPCA, provided an incentive of additional marketing exclusivity to sponsors who voluntarily complete pediatric studies outlined in a written request issued by FDA. These two legislations, often referred to as the carrot to sponsors to conduct pediatric studies, was followed by the stick with passage of the Pediatric Research Equity Act, or PREA, in 2003, which gave FDA the legal authority to require companies to conduct pediatric assessments in certain circumstances for new drugs. And in 2012, both BPCA and PREA were made permanent in legislation under FDASIA. While legislations were a critical drive for pediatric drug development, it is the collaboration of academicians, regulatory agencies, patients and advocacy groups, industry, many of you here today, who have shaped the landscape of pediatric drug development. For example, through the M-CERSI program, FDA has held several Advancing the Development of Pediatric Therapeutics workshops, or ADEPT, covering topics such as dose selection, drug dosing in pediatric patients with renal impairment, complex innovative trial design, pediatric formulation development, and so much more. For today and tomorrow, this collaborative spirit and scientific information exchange continues to help advance therapies to treat SLE in pediatric patients, a chronic autoimmune disease with heterogeneous clinical manifestations which can be debilitating to patients and can affect multiple organs and tissues. This is a rare condition, and in the United States, it is estimated to affect 5,000 to 10,000 children. Given the small number of affected individuals and the unmet need, innovative programs to support extrapolation of efficacy from adults with SLE, safety assessments in the pediatric population, and alternative trial designs are critical topics to cover. This workshop brings together expertise across many different disciplines and organizations. The planning committee includes members from the agency, including the Division of Rheumatology and Transplant Medicine, Division of Pediatric and Maternal Health, Office of Biometrics, Office of Clinical Pharmacology. The committee also includes nominees from the NIH, Pediatric Rheumatology Collaborative Study Group, and the Childhood Arthritis and Rheumatology Research Alliance, representing the pediatric academic community and investigators. And finally, nominees from pharma and bio representing industry were also part of the planning committee. We sincerely thank all of you for your time and dedication over the past months to make this workshop possible. We also want to thank partners and their families. We also want to thank patients and their families joining us today to provide us with their personal lived experience with SLE. All the efforts to advance drug development must always keep the patient's needs, what matters to them, at the core of all efforts, from product formulation, endpoint selection, trial operations and execution, and more. And with that, I would like to invite our keynote speaker, Mckenna, to provide us with the patient-family perspective on SLE. Thank you, everyone, for being a part of this important program.
Good morning, everyone. My name is Mckenna and I am a lupus patient. I was diagnosed when I was 16 years old. Since 2022, I've also been working as a patient advocate and research partner for the Childhood Arthritis and Rheumatology Research Alliance, also known as CARA, and I'm currently a second-year medical student at the West Virginia University School of Medicine down in Morgantown, West Virginia. I want to begin by thanking you all for your time and for the opportunity to speak to you all today. It's an incredible honor for me, and I'm very excited to spend today and tomorrow working alongside you to help advance medications for patients with lupus. When I first received my diagnosis, I never could have imagined what the 7 years between then and now would encompass. Initially, I, like most others, entered an unfamiliar territory, uncertain of how this diagnosis would impact my life. Fortunately, with an excellent support system of family, friends, and providers, the period following my diagnosis was not defined by uncertainty and fear, but rather by resilience and inspiration. Those around me showed me that instead of using my diagnosis as to define my future, I could become something with it. Since then, I've had the privilege of experiencing lupus from various perspectives, as a patient, as an advocate, as a student, as a researcher, and now as a future physician. And each one of these roles has taught me something incredibly valuable. But my experiences as a patient advocate in particular have been incredibly fulfilling and have given me valuable perspective. I appreciate the role of the patient voice in research, and I understand the profound impact that those living with lupus and their caregivers are capable of making. My patient advocacy work began shortly after receiving my diagnosis. I sought support and began involvement with a local chapter of the Lupus Foundation of America. This was my first opportunity to connect with other patients living with lupus, listen to their stories, and begin building a community. And as this community grew over time and I progressed through undergrad, I was introduced to CARA, which provided me with the opportunity to collaborate with hardworking and dedicated physicians and scientists by contributing my expertise of lived experience to the research team. I felt I was able to truly make a difference, that I was influencing research, reviewing grants, providing feedback on study design and outcomes. But perhaps one of the most meaningful and impactful components of my work with CARA has been the opportunity to engage within the patient and caregiver community. Listening to the various perspectives and diverse perspectives within has allowed me to understand that despite a shared diagnosis, the needs, values, and experiences of those living with lupus often vastly differ from one another. One space that this often becomes apparent is talking about access to care. Early into my advocacy work, I was shocked to learn that the challenges that many others faced were often ones that I had never thought of. I was incredibly fortunate to have access to outstanding pediatric rheumatologists, parents who had flexibility with their work obligations and could take me to and from appointments, an insurance plan that covered the cost of my treatments, and access to medications that successfully managed my disease. I recognize that for many others, this is not the case, and these barriers can often serve as a significant burden for patients, from finding providers to managing transportation and different obligations, managing financial and insurance concerns, and finding treatments that work. I also came to appreciate how significantly these experiences could influence the patient perspective, particularly regarding their care and regarding awareness of research and treatment options. The decisions made by those living with lupus and their caregivers are directly informed by their lived experiences. Their stories provide the context for why they make the decisions that they do, which is why it is essential to collaborate with those living with lupus and their caregivers. It is through patient engagement that we can identify studies that patients would realistically participate in, find what barriers feel reasonable, what risks feel acceptable, and identify studies that patients would feel safe enrolling in. If we truly want to achieve patient-centered research and develop products that address the needs of patients, then those living with lupus and their caregivers must become partners from the very beginning. Listening to others, opening myself up to new perspectives has undoubtedly made me more empathetic and understanding and has continued to empower me. Likewise, it has encouraged me to reflect on my own care and treatments, on not only what has gone well, but also some unexpected and challenging consequences that I have sometimes overlooked because I've had many great successes along the way. For example, one of the first treatment-related obstacles I faced resulted from high-dose prednisone that I was started on around the time of my diagnosis. After a couple months of treatment, in addition to more of the expected side effects like irritability and weight gain, I developed a significant swelling and pain in my knee. I had to undergo major knee surgery to repair an osteochondritis dissecans lesion attributed to steroid use less than 6 months following my diagnosis. Recovery for that surgery took an additional year, and since then, my providers and I must be sensitive to how much and how often I use prednisone out of concern for future damage. Recently, I've been navigating some new surprises regarding my treatments. For the past 2 years, I've been managing complications related to rituximab use. While rituximab has been remarkably effective in controlling my lupus, it's left me with an immunoglobulin deficiency and consequent recurrent sinus infections. Unfortunately, the immunoglobulin replacement therapy to manage this deficiency has been anything but straightforward and has presented a new set of obstacles. Early IVIG infusions left me with debilitating migraines, sometimes lasting days following the infusion. And when I transitioned to subcutaneous immunoglobulin replacement therapy, I developed even more severe headaches, neck pain and stiffness, and brain fog. Discontinuing these treatments meant returning to recurrent infections and repeated courses of antibiotics, but continuing these treatments meant preparing for days and sometimes weeks of debilitating side effects. It was a very challenging cycle to be caught in, and I felt isolated and stuck at times. I developed a deep sense of empathy for those experiencing similar cycles as myself. While side effects are expected and could simply be a cost of treatment, I understand and sorry. I recognize that for myself and many others, a significant challenge of the disease is navigating the treatments that we depend on to control it. As I reflect on these experiences now, I'm led to a space of wondering, what else is possible? While I'm incredibly grateful that my treatments have been effective to some extent, in a way they also did not work with these unexpected and sometimes harsh side effects. I'm wondering if there's a way to be even more effective with the treatments we create, of creating options that solve one problem while minimizing additional ones. Lupus patients comprehend that a cure does not exist today, so in turn, there is a universal desire for lifestyle improvement. Any options or alternatives to test or try would be greatly welcomed by a patient base that is desperate for quality of life improvements. Unfortunately, I know my story is not uncommon, which is why this workshop matters. With access to more treatment options that are effective and safe, the lives of pediatric lupus patients can be transformed. Through meaningful and insightful discussions, learning the perspective of patients, innovative clinical trial and study designs, and learning through data we already have through methods such as extrapolation, we've already improved the lives of patients and we will continue to do so. Change is good. Children living with lupus and their caregivers have a common desire to improve the lives of those affected. Is it possible to make some or more medications already approved in adults available to children? Is it possible to provide treatments under testing to pediatric patients earlier in the process? Is it possible to involve patients and caregivers in this process in any capacity? I encourage you all to ask yourself, would adolescents be willing to participate in this study? What would they be willing to risk? Could you benefit from this? Could I benefit from this? Could we collectively benefit from this? My experiences are my why. They are the reason I decided to go to medical school. I want to be able to provide the same level of care I receive that has transformed and continues to transform my life. I want to be part of the team that is giving pediatric lupus patients more options, raising the standard of care. But I recognize that developing new and better options is a daunting task. This is something that will take time, and it will need to be a collective achievement. But I'm excited because I see firsthand the work being done. I'm a recipient of it. I'm encouraged, and I know that others are encouraged too. And I'm hopeful because I see how much you all care. I see the time and energy invested into this. I know as more treatment options become available, I will be first in line, and I know I won't be alone. Everyone in this room has chosen to be here, working towards better answers. We have an opportunity to do something meaningful. We have all the right people in this room: patients, caregivers, physicians, scientists, industry partners, right here, right now. This is how change happens. This is how we start to transform the lives of those living with lupus. On behalf of all pediatric lupus patients, thank you.
Thank you so much, Mckenna. What an amazing keynote talk. I think I'm going to be able to skip some of my slides because Mckenna has addressed some of these points. Before I do that, I do want to recognize Vincent Delgazo, who's the Director of Strategic Partnership and Patient Engagement. He's the one who identified Mckenna and other patient representatives, worked with them, prepared them, and really just took the lead in helping us engage patients and families. So thank you, Vincent, if we can just recognize his contribution. Okay. So I do want us to keep what Mckenna shared with us in mind as we go through the workshop. I think that should be front and center for us. Again, my name is Lily Mulugeta, and in the next few slides, I'm going to provide the regulatory framework for pediatric drug development. And as Mckenna mentioned, really our goal is to provide timely access to safe and effective therapies. And one framework that we have accessible to us is pediatric extrapolation. So I'll touch on that as well. I have no financial relationships to disclose, and these are really my views. So as Mckenna mentioned, there are two aspects to timely access to therapies for pediatric patients. So the two dimensions are, one is timely access to clinical trials, and the other one is timely access to approved therapies. So for clinical trials, the goal is to begin once prospect of direct benefit is determined and overall risk benefit has been considered to allow enrollment of children into clinical trials. The second one, in terms of access to approved therapies, pediatric trials should begin ideally or should complete before off-label use becomes so widespread that completing trials is challenging. And from experience, this has been really around 2 to 3 years after approval in adults. So what does the regulatory framework look like for pediatric drug development? In the US, for drugs and biologics, FDA has two primary mechanisms. And I think Mary touched based on this earlier. The first one is the Pediatric Research Equity Act, or PREA, which requires pediatric studies for indications being pursued in adults. So it's the indication that's being pursued in adults. I want to note that products with orphan designation are generally exempt from PREA, and this may have relevance for a pediatric SLE. The second mechanism is the Best Pharmaceutical for Children's Act, or BPCA. This pathway is voluntary, and sponsors who conduct studies requested by the FDA under BPCA are eligible for an additional 6 months of market exclusivity. This pathway is slightly different from PREA in the sense that it can include indications that are different from the adult indication that's being pursued. And it can also apply to orphan designated products. But for the purposes of our discussion today, the context is really a drug or biological product developed for adult SLE, studied in adults SLE, and then investigated for its potential use in children. So pediatric drug development is increasingly global, and this workshop hopefully reflects this global approach. As you saw, we are delighted that we have Dr. Roberto de Lissa from the European Medicines Agency joining us later this morning to provide the regulatory perspective from across the Atlantic. But I just wanted to provide a brief comparison. In the US, pediatric study plans, or IPSPs or PSPs, are typically submitted under PREA early in development, and this is typically after the end of phase 2 in adults, and are required for submissions involving new active ingredient, new indication, new dosage form, new dosing regimen, or new route of administration. So it's not for all submissions. The indication for the pediatric study plan is the same as adult because this is under PREA. If the adult development program is for general SLE, the pediatric study plan would be for general SLE and not lupus nephritis. In the EU, the framework is slightly different. And again, Roberto will expand on this later this morning. The Pediatric Investigational Plan, or PIP, follows a slightly different framework. The PIP is typically submitted after the end of phase 1 in adults, which means there's limited efficacy and safety data for that indication in adults. And it applies to all medicine, so it's not restricted to those triggers that I mentioned earlier. And it can include indications broader than what's being sought in the adult population. Both include a plan for development of age-appropriate formulations, which will come up in our discussions today. And there's an ability to request for a waiver if the disease prevalence is too low or if the trials would be infeasible. There are other criteria as well, but I'm mentioning this specifically because it does apply to SLE. And for most agreed IPSPs for SLE, we typically agree for a waiver in pediatric patients less than 5 years. I just wanted to briefly go over the content of a pediatric study plan, since this is not something generally outside of industry and regulators people would be aware of. So the PSP outlines the available data, provides an assessment of the extrapolation approach, proposed studies, and timelines for the pediatric studies, as well as any waivers for conducting studies in one or more pediatric age groups. The plan should also address any need for juvenile toxicology data to support safety and enrollment of younger patients, and this is typically in patients less than 12 years of age, formulation requirements, and what clinical studies would be needed. I also want to note that the PSP includes any agreement with other regulatory agencies. And so when FDA notices that there is a difference in approach, FDA uses existing mechanisms to engage with other regulators to understand the rationale for the divergent approaches and see if alignment can be reached. And an alignment cannot always be reached, but we attempt to reach an alignment. Sponsors also have the option of requesting product-specific advice from both FDA and EMA concurrently under the Parallel Scientific Advice Program. Just so that we are all on the same page, pediatric drug development is really held to the same rigorous evidentiary standard as adult drug development. A product approved for children must demonstrate substantial evidence of effectiveness and clinical benefit, and the submission must include sufficient information to allow for appropriate risk-benefit analysis. So this brings us to pediatric extrapolation. So in the US, and as Mary mentioned earlier, in addition to the statutory requirement for substantial evidence of effectiveness, there's also statutory acknowledgement that the development of a drug or biological products in adults provides a rich source of data to inform drug development in children. And given reasonable similarity between the adult and pediatric population, extrapolation of efficacy data from adults may reduce evidence required from pediatric trials to evaluate the effectiveness of products. We also know that it's ethically imperative to consider how data in adults can be leveraged, because children should only be enrolled in clinical trials if the scientific objectives cannot be met by enrolling subjects who can provide informed consent, such as adults. So the concept of extrapolation has evolved considerably over time. Although it was first introduced in 1994 as part of the pediatric labeling rule, extrapolation has really evolved from a binary decision to a rigorous evidence-based continuum, with each guidance really building on the last. So the last guidance that you see at the bottom of the slide here, the 2024 E11A Pediatric Extrapolation Guidance, provides a more comprehensive framework for extrapolation. And I'm going to discuss this over the next few slides. Okay. So this slide illustrates the key domains we evaluate. The three that are listed on the slide really are an assessment of the disease similarity. So the disease itself, the drug's pharmacology, and the response to treatment. These are the three main key domains. For each domain, we ask whether the available evidence supports similarity between the reference population, which is typically adults, and the target pediatric population, or whether meaningful differences exist that require additional data. And for this workshop, this will be the core of the presentations and discussions in session 1 and 2 today. So before designing any pediatric program, we must rigorously identify knowledge gaps. What are the identified gaps in knowledge? Do these gaps require additional data collection in adults? What specific data should be collected in pediatric patients to close those gaps? And this will be the basis for the presentations and discussions in session 3. ICH E11A, and that's the pediatric extrapolation guideline that I mentioned to you, also reinforces that the aim of the pediatric dose selection is often to target exposures in pediatric patients that are known to be safe and efficacious in adults. And of course, that pediatric dose has to account for maturational and developmental changes that may impact PK. And that's typically done by conducting PK studies in the target pediatric population. The guideline also recognizes that a separate standalone PK study is not always necessary. But in addition, it does recognize that there may be considerations or circumstances that could lead to the need to evaluate different dose or exposure levels. So these would be exposure levels that are either higher or lower than the exposures approved for use in adults. And these could include uncertainty in the disease similarity or response to treatment, potential age-related differences in the target expression. So this could potentially impact safety or efficacy, evidence from early phase trials or other programs indicating need for higher exposures, or really insufficient dose exploration in the adult program. So it's really critical that we think ahead and understand what additional data we may need in the adult program to minimize additional evidence requirement from the pediatric trial. And this will be relevant for our discussion today. This is really a simplistic presentation, but I thought it might be easier to start this way before we go through the additional slides. Really, the appropriate extrapolation approach depends directly on the degree of disease and response similarity. So when the disease and response are the same in adults, we really just need PK and safety data because we can rely on the efficacy data collected from the adult trial. When the disease is clearly different, a controlled trial is often needed to establish efficacy in the target pediatric population. But where most pediatric programs lie is really in between those two. And when the disease is similar, but there's some uncertainty, then the study design must be calibrated to address the specific degree of uncertainty. So this slide really captures the continuum concept that was introduced in the last ICH 11A guideline that was published in 2024. really demonstrates that as similarity increases, the evidence requirements from additional pediatric data decreases. So the key principle is that we make optimal use of all available data, and this is not limited to the adult data. This will allow us to reduce the burden on pediatric patients while still meeting the rigorous evidentiary standard that I mentioned to you. And this will be the core of the discussion for session 4, which will be tomorrow. I know this slide may be a little bit difficult, but I thought it would be important to present examples of potential extrapolation approaches, ranging from the most streamlined, which would be a PK safety study that's presented at the bottom of the slide, to the most data intensive, which is one or more adequate or well-controlled trials, which is presented at the top of that slide. I just want to be clear that this is really not a comprehensive list of potential approaches, but it's really to demonstrate that the appropriate approach for any given program is determined by where the program falls on the continuum of similarities that I just discussed: similarity in disease, similarity in drug pharmacology, and similarity in response to treatment. I also want to briefly address safety extrapolation. I think for the longest time, the general policy was that safety cannot be extrapolated. ICH 11A introduced formally the extrapolation of safety, which is often considered separately from extrapolation of efficacy. So even when we do agree to extrapolate efficacy, we must still carefully evaluate the safety profile in adults and whether it can be assumed to apply to children. We know that developmental and maturational changes can affect safety in a way that may not be predictable from data in adults alone. We also have to consider how much of that safety data should be collected before approval of the therapy in pediatric patients versus post-approval in the target pediatric population. And this will be discussed tomorrow. So I just wanted to walk you through, this is a lot of concepts that we're throwing at you, and I know most people outside of industry and FDA may not be familiar with this guideline. So I just wanted to share a concrete example, one example that many of you are familiar with, and one example really that demonstrates the effectiveness of engaging the public the way we're doing with this workshop. So this is a framework that was published by ICH 11A as a training material, and it uses a hypothetical TNF-alpha inhibitor noted as drug X being developed for PJIA. So we start assessing disease similarity, and we know PJIA and adult RA share the same underlying pathophysiology, including TNF-alpha mediated joint inflammation. They use the same outcome measures. So the evidence of disease similarity in this case was very strong, so no knowledge gap was identified. Next is drug pharmacology and response to treatment. TNF-alpha inhibitors as a class have a well-established mechanism of action, and available data from RA and PJIA clinical trials show that pediatric patients respond similarly to adults. Again, strong evidence, so no gaps identified. So from there, we moved to just general gap identifications. We didn't have pediatric specific information for this particular TNF-alpha inhibitor, so additional data to inform dosing was needed. There was also a known class-level long-term safety concerns, specifically risk of malignancy and new autoimmune disease that needed to be characterized in the pediatric populations. So those gaps then directly determined the study type. In this case, efficacy could be extrapolated. So an efficacy trial was not needed in pediatric patients. Instead, the gap supported a PK and safety study in pediatric patients 2 years and older to establish the right dose and to begin characterizing the safety profile of this particular TNF-alpha inhibitor in pediatric patients. I know Mckenna already mentioned this, so I feel like I can almost skip this slide, but another valuable and increasingly used approach or strategy to expedite access to therapies is enrolling pediatric patients directly in adult trials. We typically think of adolescents, but this approach is not limited to adolescents. This is, of course, considered on a case-by-case basis and requires strong evidence of disease similarity, sufficient data to support dose selection if we go below 12 years of age, formulation availability, and preliminary evidence of clinical efficacy and safety in adults, which we typically have at the end of phase 2 when PSPs are submitted to the FDA. At the same time, we have heard very clearly from previous workshops that operational considerations have to be included. Site selection, frequency of assessments, endpoint suitability for pediatric patients must be considered. So we hope to have some discussion around this aspect as well during the workshop. Finally, just additional considerations as we think again of expediting pediatric programs so that pediatric patients have timely access to clinical trials and approved therapies. So additional innovative approaches that we typically leverage is a PK lead-in study design instead of a separate PK study, use of modeling and simulation to estimate dosing in pediatric patients, and we're going to hear about that today, and leveraging data from all other data sources, and it can be from other indications as well. So this slide really just tries to frame the workshop discussion. The question for us is, where does pediatric SLE fall on the continuum that I shared with you, the continuum that was published in the ICH 11A guideline? What is the degree of similarity between adult and pediatric SLE in terms of the disease, drug pharmacology, and response to treatment? And given that assessment, what existing data from studies in adults could be leveraged? What additional data do we need in the target pediatric population? And then the final question is going to be, then what is the optimal trial design to obtain the necessary data? Before I close, and I think Mary did this very nicely for us as well, I just really want to thank everyone who made this workshop possible. And I'll turn it over to our next speaker, who's Emily Gotschlich, and she's the clinical reviewer and DRTM at the FDA. Thank you.
Okay. Good morning. Welcome, everyone. Thank you so much for being here. Lily, that was wonderful. Thank you. I'm going to try to expand upon that a little bit and talk to you today about the FDA perspective of drug development on pediatric or childhood onset SLE. So in my talk today, I will briefly discuss the background leading us to organize this workshop, including our experience with drug development in SLE, where we have historically required efficacy studies, which have been randomized placebo-controlled trials, and specifically our experience with belimumab as the only approved product for pediatric SLE and pediatric lupus nephritis. As part of this, I'll discuss the relevance of extrapolation of efficacy in this context, and then the goals of this workshop over the next day and a half. So first, I want to mention the origins of this workshop and the successful use of the extrapolation of efficacy over the past 6 or more years in the Division of Rheumatology and Transplant Medicine. In 2019, we held a similar workshop entitled Accelerating Drug Development for Polyarticular Juvenile Idiopathic Arthritis, or PJIA. This was a collaborative workshop hosted by the University of Maryland, M-CERSI, and the FDA. The goal was to address barriers to drug development in JAA and to develop steps to overcome them. There was a lot discussed at this workshop, including trial design considerations, dose selection, as well as the topics of extrapolation of efficacy and the level of evidence required to establish safety and effectiveness in JIA. Ultimately, these discussions were very impactful and led to a pathway for using extrapolation of efficacy to expedite approvals in JIA. So extrapolation has now been the basis for a number of recent pediatric approvals in rheumatology. Since the JIA workshop in 2019, the agency has approved 4 products in PJAA using an extrapolation approach and 11 products in juvenile psoriatic arthritis using an extrapolation approach. And you can see these products listed here on the slide. And so you can really see the impact the use of extrapolation has had in JIA. So now I'll move on to discuss our experience with drug development in SLE. Though you just heard this from Lily Mulugeta, as a brief reminder, the Pediatric Research Equity Act, or PREA, is a mandate that requires companies to assess the safety and efficacy of certain new products or biologics in children. And the intent of PREA is to encourage sponsors to identify needed pediatric studies as early as possible in product development. But importantly, to initiate pediatric studies, evidence is needed to support a prospect of direct benefit to justify the risks of exposing children. In general, this evidence is first derived from adequate and well-controlled studies in adults where efficacy and benefit risk of new products is first assessed. And this generates the prior knowledge to support the consideration of extrapolation to pediatric patients. So the only FDA approval in pediatric SLE and pediatric lupus nephritis has been for belimumab. In April of 2019, the FDA approved the first treatment of IV belimumab for children 5 to 17 years of age with active seropositive SLE receiving standard of care. So IV belimumab was first approved in adults in 2011 and then 8 years later for the same indication in pediatrics. It was approved in 2020 for adult lupus nephritis, and only 2 years after that was approved for pediatric lupus nephritis in the IV formulation, and then 5 years later in the subcutaneous formulation, both due to our ability to use extrapolation. This product remains the only approved treatment for pediatric SLE and lupus nephritis. And the recommended dosing for pediatric patients is the same as that for adults listed here on the slide. So the details of this program will be more thoroughly discussed in later sessions, particularly during session 2. But I do want to just highlight it briefly here for context. The approval of IV belimumab in pediatric SLE was supported by a single post-marketing required randomized controlled trial that evaluated the pharmacokinetics, safety, immunogenicity, and efficacy of 10 mg per kg of IV belimumab versus placebo in 93 pediatric patients. They looked at an SRI-4 at week 52, and the results of this study show that SRI-4 was achieved in 53% of belimumab-treated patients versus 44% of placebo-treated patients, all on background standard of care. Due to the rarity of childhood onset SLE limiting enrollment, this study was not adequately powered to make formal statistical inferences on its own. So determination of efficacy was therefore primarily based on PK and efficacy results from this study, as well as extrapolation of the established efficacy of IV belimumab from the 2 phase 3 adult studies. Both of these studies in adults were multi-center randomized double-blind and placebo-controlled studies in over 800 patients with active SLE, examining the efficacy and safety of 2 different doses of belimumab with a primary endpoint of the SRI-4 at week 52. In order to support extrapolation of the data from these adult studies, we considered the similarity between the adult and pediatric programs and then used Bayesian borrowing. And again, as I mentioned, these studies will be discussed in more detail during session 2. Now, in the 2022 approval of IV belimumab for pediatric lupus nephritis, rather than a clinical study, a PK extrapolation approach was used to support approval. Because of disease similarity between lupus nephritis in children and adults, efficacy in adult lupus nephritis could be extrapolated to pediatric lupus nephritis. And this was further supported by the existing efficacy data of belimumab in the pediatric SLE randomized controlled trial conducted in the pediatric SLE population. We relied heavily on the existing SLE data to support this approach. A PK bridge between the 3 populations of adult and pediatric patients was then established, and safety information was leveraged from the existing pediatric SLE data. Now, these three products, anifrolumab, voclosporin, and obinutuzumab, are approved in adults and have ongoing or completed studies in pediatrics. Historically, under PREA, as I mentioned, we have required randomized controlled trials in pediatric SLE as well as pediatric lupus nephritis. Anifrolumab was approved for adult SLE in 2021, and the sponsor is now conducting a study called Blossom, which is an ongoing randomized double-blind placebo-controlled study in patients 5 to less than 18 years old using a stepwise approach based on weight and age. This study started in 2024 and is estimated to be completed in 2028, which will be 7 years after its initial approval in adults in 2021. Voclosporin was approved for adult lupus nephritis in 2021, and this sponsor has completed a study called VOCAL, which was a double-blind placebo-controlled study of voclosporin looking at renal response at week 28 in patients 12 to 17 years old with active lupus nephritis. This study was started in 2023 and then closed in 2025. They were able to only enroll 9 patients. And the open-label extension to the VOCAL study was completed in 2025 as well, with only 4 subjects. Lastly, obinutuzumab was approved for lupus nephritis in adults in 2025. This sponsor is conducting an ongoing phase 2 study called Posterity in biopsy-confirmed lupus nephritis in adolescents aged 12 to less than 18. They are also enrolling a smaller number of younger patients for open-label safety and PK data, and they're planning to use this phase 2 study in adolescents as a pivotal trial to support approval. And just as a note, all the information on this slide is available at clinicaltrials.gov. So lastly, I'll discuss some of our goals over the next day and a half. You have seen this concept of extrapolation of efficacy and disease similarity presented in the previous talk from Lily Mulugeta, where the increasing confidence and similarity of the pediatric and adult versions of the disease leads to an increasing relevance of adult data to the pediatric population. And I raise this concept again now in order to address this major question of our workshop. How significant are the knowledge gaps and the uncertainties regarding the similarities and differences between the adult and pediatric SLE? And are these gaps in knowledge, these disease differences, and other uncertainties substantial enough to prevent the use of extrapolation approaches in these populations? In the case of belimumab, we considered adult and pediatric SLE to be similar, but with some differences, requiring a randomized controlled trial in the pediatric population in order to address some of these uncertainties and then use Bayesian borrowing. So one major question for today is whether we might consider pediatric and adult SLE to be similar enough that we may not need to require an efficacy study, that is, a randomized controlled trial in pediatric patients. And instead, could we use exposure matching with or without alternative trial designs to expedite drug development? So this brings me to my last slide to address the goals for the workshop today and tomorrow. Our number one goal is how can we best advance drug development in pediatric SLE and best serve pediatric SLE patients? Can we address the uncertainties regarding the differences in the adult and pediatric SLE populations such that we could consider the use of extrapolation? And if these uncertainties cannot be addressed, such that we do need additional clinical efficacy data in pediatrics, then what is the best way to achieve that? Thank you. And with that, I'll introduce our next speaker, Roberto de Lisa, here from the EMA. Thank you so much, Roberto.
Thank you very much, Emily and Lily. And thank you so much to the FDA colleagues for organizing this workshop. I praise their dedication and the effort that they put in the careful selection of the session, of the speakers, and the material to discuss. I would like to start with what has been our perspective on development in this difficult area. And please go ahead with the slides because some of them, they have animation. So you can go ahead and clicking and then I'll follow through. We can skip the disclaimer clearly, everybody can read that. And the presentation will focus mainly on, go ahead, please. On regulatory pathway to pediatric development, what is the adult pediatric similarity, on better what we think are the key point on this. Next one. And next one, please. You please go ahead clicking because there's no problem until the slide is complete. Yeah, first of all, how do we differ from the FDA? Yeah, we in Europe agree a development plan very early. Next slide. So specifically when in general it is recommended by the law after the phase 1 results are provided. So it means that we only have PK in adults. We don't have any specific indication of efficacy, not even from phase 2. Our PDCO opinion is in a way complete, meaning that it includes several measures for development. It includes a recommendation and obligation to develop a specific formulation, for instance, then the necessary juvenile animal studies that need to be performed, then, of course, the clinical development, where we list what are the key elements of every study, so the design, the population, the number of patients to be recruited, and then the analysis to be carried out. And we have seen particularly extrapolation. What it means extrapolation? It means looking carefully at the data that have been provided in adults to see what can we leverage, and particularly how the PK is analyzed and how the exposure and the clinical response go together. Next one. As I said, of course, even if we agree them pretty early, then, of course, companies can come back and modify them according to how they go, or improving the design and having feedback from opinion leaders. So at least it's not stuck in stone. But it is what the advantage of agreeing these studies very early, what it is that we can shape, or we hope to shape, the adult collection of data in a way that is more useful to then build useful pediatric trial and then complement the information that is needed for us. In particular, we are able to distinguish different characteristics of the disease in this way and focus on key aspects that then will be used later. The grades of similarity of adult versus childhood are always been at the focus of our discussion. And then we talk about particularly what is the organ involvement, specifically now that the scientific community has moved a little bit on and they are considering like organ involvement not a characteristic that, let's say, distinguish the type of the disease, but as part of it. But it's particularly important to consider as including patients with renal or neuropsychiatric disease. And of course, there is also an evolution on how biomarkers could be similar or different in the two populations. Next one. The clinical characteristics of juvenile or childhood SLEs, that are causing us more trouble are, so we have said, like different organ involvement, so more frequent renal involvement with nephritis of CNS involvement. We are still unsure, particularly in light of the new literature data, whether organ damage accrual is faster in younger than in adults. But you will see that this will be also picked up by other presenters during the different sessions to have a more in-depth discussion. In essence, the question sometimes is, do children or adolescents need to be treated with higher doses of the approved medication? And does this mean that the response to treatment it needs to be evaluated differently, so with a different clinical design? But particularly, we are concerned that the specific pediatric needs are covered during the development in a way or in another. Next one. What has been our experience so far? I can tell you we've been pretty busy evaluating plans. Next one. And we have been also busy, not busy, I mean, of course, we encourage the dialogue with different stakeholders. We have interacted with the scientific society. Of course, as Lily mentioned, we have a discussion on single products during our pediatric cluster with the FDA to pursue global development. We also recently published an article with the help of very known academicians that are also participating to this workshop, which I thank. And we also launched our own studies because in Europe we have our network of real-world evidence sources to investigate potential aspects where we were concerned, let's say, the treatment was different, or try to understand the reason for this. Next one. So far, we have agreed 24 PIPs. So for 24 substances, there is an agreed PIP. Some company have decided, maybe because the pipeline was not going ahead, the plans were not going ahead for adults, to withdraw them. And we also have many that are currently under evaluation. So you see, it's a pretty busy pipeline. Next one. For most of these PIPs, what I wanted to emphasize is that we have agreed as a pivotal study, a randomized control study. In many cases, in several cases, this has also been randomized with a withdrawal design, randomized control trial with a withdrawal design. We will come back to this issue also to discuss in details why we are why this is a subject to debate also on our side. And in some cases, but particularly for products that were targeting lupus nephritis, we have agreed single-arm trials. Next one. What is peculiar in this area, but resembles a little bit what is happening also in juvenile arthritis or other rheumatic diseases, that all these substances are scattered around different mechanism of actions. Products that interfere with the B-line cells or with the T cells. We have, like most recently, we had CAR T cells. So this, of course, it's another challenge because some of them, they were first in class. So the conclusions that we reach on one may be not exactly the same that can be applicable to another one. And needless to say, it's important to underline that many of them impact, of course, on immunity. So knowing that sometimes in juvenile lupus, increased risk of infections is one of the factors. This is another factor that pushes us to have even more rigorous data to be collected. In this slide, please click once more, you will see that all of them, we have agreed, they have a huge overlap in timing of their running. So this means that we expect competitive recruitment in the area. And this is another challenge that we have to face with few centers and relatively few patients, even globally. This is one of the things that is causing us troubles to agree, like more effective guidelines and how to intertwine that to the adult development. Next one. Challenges, another challenges that we have faced are, of course, to the inclusion criteria. I mentioned it before. So, for instance, patients with renal and CNS involvement, particularly for products that are developed for adults where they are intended to be second or third line and not first line, the indication in adolescents or in children would not exactly match. So do we need a specific trial that includes a specific subgroup of patients with some organ involvement? Allocation to placebo, we have been challenged a few times for trials that have placebo as a control arm, provided that, of course, is not a real placebo, so to say, because it's always like as an add-on therapy generally. So patients are treated to the best care as possible. As I mentioned already, the randomized withdrawal design, sometimes this decision has been, we have had feedback from stakeholders that probably was not the most efficient design given the ethically challenging risk of relapses, of the carryover effect from an open label phase where all the patients receive the active substance. Next one. We talked about extrapolation. Of course, we detailed the extrapolation plan in our PIPs. And just to mention, on top of what colleagues have agreed, the extrapolation principles and the way to act and to approve an extrapolation plan as laid down in a document that is a document recognized internationally. So it's the International Conference of Harmonization. So it's something that is happening globally to promote effective pediatric development. Next one. And following one. So what are the gaps that mainly concern us? Okay, but essentially, the most important thing is, as I said, the 4 differences that we have underlined. So it seems that sometimes the disease it's more severe and with a worse outcome and is treated with the higher doses in adolescents or in children. But this will be, of course, subject of discussion during this meeting. So our main question is, does it impact on one of the key concepts of extrapolation, so similarity of exposure? So is the effective dose that is in adult that provides a certain exposure the one to be targeted, or we need to target another one? Next one. Just to highlight that also a few new publications, of course, they add clarity sometimes, but sometimes they also add like a few points that needs to be discussed. So the long term, the remission time, how long we need to observe patients, similar to adults, more than adults. As I said, the similarity in exposure response, the key predictors in damage accrual, and the new concept, for example, of treat to target that is developing also for more advanced in a more advanced way for children and adolescents rather than for the adults. Next one. What are we trying to do to resolve all this gap and to increase our knowledge in the disease and in the difference and in the similarity of disease with adults? Next one. We have tried to interrogate, as I said, we have a network of centers for real-world data, and we have tried to interrogate that specifically for SLE. We have had like some results that probably confirm what was already known, so that high glucocorticoid prescription in pediatric patients is seen. Of course, there are explanations for that, the fact that they are first line, the fact that they have limited therapeutic options compared to adults. Yeah, we're gonna also rerun the study with more, given that we have more databases and more center available to provide data to see and to investigate how patients are treated and to provide then in the design of clinical trial involving pediatric patients the best care that they are being given to. Next one. Of course, belimumab is also indicated in the EU, and this was based on a randomized control trial that I would remind and did not reach statistical significance, but overlapped in a way the response that we had seen in adults, and particularly also in the secondary endpoint with the PRINTO endpoints that are specifically designed for pediatric patients. There was a greater possibility to see differences between the placebo arm and the control arm. So also belimumab was approved on this on this basis without the need on our side, for instance, of a Bayesian approach. Reanalyzing the primary endpoint with a Bayesian approach, we consider that evidence was sufficient. The problem here, I wouldn't say the problem, but the fact here to underline is that this was not planned at the beginning. So we would like to see things developing as planned, like with a proper extrapolation concept and analysis agreed beforehand rather than post hoc. Although, of course, this was the first one and the first milestone that we reached in Europe also for pediatric treatment. Okay, let's go to the main conclusion and key message. As I said, we're gonna take this point for discussion later during the meeting. So for the time being, our regulatory expectation remain broadly consistent among different substances. So at least, you know, there is a clear path to plan a pediatric development that then can be reshaped as soon as adult data become available and give us more insights. There are certain features that leave some residual uncertainties that limit a little bit the reliance on single-arm trials alone. We expect to see adaptive trial design, possibly also basket and platform approaches. I know some of the speakers will touch upon this and their feasibility. And what I wanted to stress is that a universal model for extrapolating efficacy across so many different substances and so many different adult development, it's difficult to be pursued. We need a structured approach that defines the gaps, it identifies the strategies to address these gaps, and there's no single recipe to address extrapolation across all the different substances. Some programs would need to have more data with some specific designs, where some others would not. What is particularly important, and of course, adult trial, they need to have usable evidence. What does it mean this? Like, for example, if we have a clear exposure response that is defined, that has been built, if we have clear PK data that have been collected in adult, this will facilitate, of course, then implementing an extrapolation plan. And with this one, I think I just ran 2 minutes over time. I'll thank you so much, and I leave the floor for any question or for the next speakers. Thank you so much.
Thank you so much, Roberto. So this takes us to session 1 now. And session 1, we're going to try to tackle one of the key domains to assessing similarity, which is going to be assessing similarity of disease. So we have 3 academic talks bundled under the first one. So pediatric SLE, presentation and relationship to adult SLE. Our first speaker is going to be Stephen Balevic from Duke. He will be followed by Rebecca Sadun, who's also from Duke. And then finally, Stacy Ardoin, who is from Nationwide Children's Hospital. So I'll turn it over to Stephen. Thank you.
All right. Thank you very much for the introduction and for the invitation to speak. This is such a fantastic workshop. And we've already heard from the patient perspective how really important this is. So today, again, I'll be discussing clinical manifestations and classification criteria. I've listed here my conflicts of interest and disclosures for your review. And as the first of the academic talks, I'd like to just briefly introduce really what this session will cover. And so again, I'll be discussing mostly clinical laboratory manifestations. Our colleague, Dr. Rebecca Sadun, will discuss pathobiology next. And then Dr. Stacy Ardoin will discuss outcome measures and disease trajectory. Now, what's really amazing and unique is that all 3 speakers this morning are board certified in both adult and pediatric rheumatology. So we all see lupus patients across the age spectrum. So we'll be able to provide not only some data, but also our clinical experience as well. So I'm actually going to start backwards, and I'm actually going to start by showing you the claims that we hope to make during this session. And then the rest of our talks are going to be the data to actually support these claims. So we hope to convince you that SLE, or what I'll refer to as lupus, is really the same fundamental disease that can present in childhood or during adulthood, but that we have very limited data in our youngest children, with typically less than 5 years with monogenic disease. I'll show you that there's complete overlap in disease manifestations, but there are some different phenotypes. So disease activity and certain organ involvement can be higher or different in children. And then the same classification criteria and disease activity measures are used in children and adults for the purposes of drug development. So first, what do we see when we walk into the room as a clinician? Well, one of the first things that we can see is cutaneous or skin manifestations. And so here I have a picture of acute cutaneous lupus, and on the left-hand side, a pediatric patient, on the right-hand side, an adult patient. And you can see the very characteristic redness in this what we call malar or sort of butterfly distribution, sparing the nasolabial fold. So it's a very distinctive rash. But what you notice is that they're really indistinguishable, the pictures on the left and the right? They look the same in children and in adults. So how about other things that we may see with our eyes? Well, how about subacute cutaneous lupus? Well, again, looking at this photograph, you could not tell whether this is an adult or a pediatric patient, right? And that's intentional here, right? Because they're indistinguishable. How about chronic cutaneous or discoid lupus? Well, once again, you can't tell from this photograph if this is an adult or a pediatric patient, and again, that is intentional. We also see other characteristic lesions. So how about ulcers, right? Lupus characteristically causes these ulcers on the roof of the palate, and we see that in both, again, adults and children. So unfortunately, I don't have time in this 10-minute talk to go through all the different clinical manifestations of lupus. So I thought what may be helpful is to present a Venn diagram, right? So on the left-hand side, things we only see in children. On the right-hand side, things we only see in adults. And of course, the area in the middle here are areas of overlap. And so when you look across all the protein manifestations of lupus, there's complete overlap. Right. So stated another way, there's nothing that we see in our pediatric patients with lupus that we don't also see in adults. And similarly, nothing we see in our adult lupus patients that we don't also see in our pediatric patients. Now, how about as a sort of clinical manifestations? How about laboratory manifestations? Well, same exact thing. We see complete overlap. So the characteristic cytopenias, low complement, autoantibody generation. So complete overlap. Now, although there's complete overlap in the clinical and lab manifestations, there are some very important phenotypic differences that I'd like to highlight. So we do know that children are more likely to have higher disease activity at the time of diagnosis. So higher SLE-I scores, which is a disease activity measure we'll talk about later. But children are also more likely to have renal or neurologic involvement as well, and that often drives these higher scores. Conversely, children are less likely to have arthritis or pleurisy or pulmonary involvement. So how then do these phenotypic differences impact drug development or potentially impact drug development? Well, in clinical practice, we see that children often receive more aggressive treatment compared to adults. And this is largely because we know that activity equals damage. Right. So when lupus goes uncontrolled, it can cause permanent organ damage. And children have typically more years of potential life with which to accrue damage. So what this means is pediatric rheumatologists is that we tend to have very low threshold for treatment and treat very aggressively to try to obtain remission in order to optimize or reduce damage. Now, I think one thing that's very important, so throughout some of these lectures, we're going to look at different treatment patterns, right? Glucocorticoid use or things like IV cyclophosphamide. Very important to realize that treatment patterns can be difficult to interpret, right? Because they can be impacted by provider bias, patient family preferences, and disease comorbidities. So I'll give you a classic example. Let's say we have a 60-year-old adult who has chronic kidney disease, maybe stage 3 from diabetes and hypertension. If that patient develops new onset lupus nephritis, it's very possible they may not receive as aggressive of treatment because the ultimate need for dialysis may not be changed, and because there's higher risk of using glucocorticoids in somebody, let's say, with uncontrolled diabetes. I'll take that same case, but now a pediatric patient who's previously healthy and has completely normal kidneys with no chronic kidney disease. You can imagine you want to do everything possible to try to reduce lupus activity to really optimize the long-term kidney outcomes, right? So the children or child in this case may receive higher dose glucocorticoids, maybe even cyclophosphamide. So treatment patterns, again, you have to interpret with some caution. Now, we're going to talk about drug targets later, so I'll skip over that for now. The last thing I'm going to mention about phenotypic differences, though, before we jump topics here, is that we see something similar with other pediatric diseases, especially pediatric autoimmune diseases. So, for example, we know that type 1 diabetes also has a more severe phenotype in children compared to adults, yet has, compared to adults, the same genetic risk factors, autoantibody development, destruction of the cells in the pancreas. And so again, phenotypic differences may not necessarily mean that's a different disease. You have to look at the individual disease. Dr. Sadun and others are going to talk a little bit more about monogenic lupus, so I'm going to keep it very, very brief here. Again, this is a lupus that tends to present early in life, usually due to a single gene defect. I will say for my purposes that the cardinal features of monogenic lupus still seem to be similar to lupus in older children. So mucocutaneous disease, neurologic, arthritis, nephritis, so same types of clinical manifestations. All right, then lastly, I'm going to talk about classification criteria. So for those of you not familiar with this, these are criteria that we use to develop a homogeneous group of patients for research studies, including regulatory drug studies, which is why we'll talk about it today. Some clinicians use them as diagnostic criteria, but they were not designed or intended for that purpose. We can debate that later. So again, just following our same Venn diagram, we see that there's 3 common classification criteria that are used for lupus. And again, we see that there's complete overlap between adults and children. So we use the same classification criteria. Now, what's used for regulatory purposes? Again, this is not exhaustive, just some examples. We know that the ACR 97 criteria we used in both the adult and the pediatric belimumab program. And then more recently, the EULAR-ACR classification criteria, again, have been used either in a clinical trial or in a regulatory submission for both adult and pediatric products. Now, these classification criteria are generally developed first in adults, and so the question comes up, well, how do they perform in children? And the answer is pretty well. So these are studies that have looked at the sensitivity and specificity. So you can see, especially for our newer criteria, sensitivity in children is quite high. So, probably 95%, just kind of looking across these studies. Specificity is also, again, quite high, usually high 80s or low 90s. That's actually similar to what we see in other adult cohorts when these are applied outside of the validation cohort. So really what this reflects is, again, high overlap in clinical and laboratory manifestations, but also just similarity. The types of adult patients who enroll in clinical trials look similar to the types of pediatric patients enrolled in clinical trials, at least for the purposes of these classification criteria. So then in conclusion, again, hopefully I have tackled number 2 and number 3 here, showing you this complete overlap in disease manifestations. We do have to be cognizant of these important phenotypic differences, but the same classification criteria are used for the purposes of drug development. And with that in mind, I will hand this off to Dr. Sadun. So thank you very much.
Thank you so much for that wonderful presentation and setup. I have been asked to discuss the biology of lupus and the development of the immune system in 10 minutes, so buckle up, here we go. To start with, we know that there are genetics involved in the development of lupus. Specifically, there are about 50 genes that have been associated with what's known as monogenic lupus, meaning all you need is a mutation in that one gene for it almost certainly to develop into lupus. Beyond that, GWAS studies have identified over 300 susceptibility loci. So these are different genes that if you have this different code, you are more likely, but it is not inevitable, that you would go on to develop lupus. And so what are the triggers that can interact with those susceptibility loci? Those are UV light exposure, certain viral infections, certain medications, et cetera, these environmental triggers, such that even if we have identical twins with the very same genetics, one has lupus, about 25 to 50% of the time, not 100% of the time, the concordant twin would also have lupus. So what does that look like? On the lower left side of the screen, we see DNA. That's the genetic predisposition. And then over time, environmental interactions and triggers that result in first some dysregulation of the immune system, and then ultimately crossing that dotted line into clinical immunity or the development of clinical lupus. We see here in this picture that was the small piece of DNA, and that's the idea of those susceptibility loci versus a monogenic lupus, a huge genetic component that needs much less to trigger and develop into lupus. Okay, let's talk about the two arms of the immune system and how they get dysregulated in lupus. There's the innate immune system and the adaptive immune system, and we'll tackle them individually. So starting with innate immunity dysfunction, and this is true across all ages. We see the same sorts of things happening. So one of the more common places, one of those more common areas for mutations is defective clearance of apoptotic debris. And this means that nuclear contents, DNA and other nuclear contents, are exposed to the immune system. That winds up resulting in activation of plasmacytoid dendritic cells, also known as PDCs, which leads to excessive production of type 1 interferons. And this is a gene signature showing the upregulation of those interferons. That itself can promote dysregulated netosis. And so this results in more nuclear contents being exposed. And that triggers more activation of PDCs, more production of interferon, and as you can see, a vicious cycle. In addition, the dysregulated netosis can wind up stimulating toll-like receptors, particularly TLR7 heavily implicated in lupus. And that can lead to maturation of myeloid dendritic cells, or MDCs, which then wind up increasing antigen presentation to lymphocytes and increased production of BAFF. So here I wanted to highlight just a few of these pathways which are the targets of many of our medications. So high-dose steroids, pulse-dose glucocorticoids can actually tame some of the PDC activation. Interferon is the target of anifrolumab, and of course hydroxychloroquine, sort of the backbone mainstay for treating mild lupus, is targeting through one of its major mechanisms, the TLR7. Also wanted to point out, in addition to this being a vicious cycle, that lupus can start at any one of these time points. So a different mutation might start you at 0.4, and then that cycle still perpetuates. At the bottom here in this picture, you can see the parts of the innate immune system. So those activated plasmacytoid dendritic cells, the type 1 interferon, BAFF production, et cetera. And then you can also see how that interacts with the adaptive aspects of the immune system, stimulating and activating, for example, autoreactive B cells. So that transitions us to the next point. I want to talk about dysfunction in the adaptive arm of the immune system. So autoreactive B cells, highly implicated in lupus. We see increased survival of autoreactive B cells. We see loss of self-tolerance and tolerance checkpoints. We also see increases in double negative B cells, increases in the population of AIDS-associated B cells for whatever a patient's age expectations would be. And we see T cell abnormalities. We see dysfunction of T regulatory cells. There's now a newly described double negative T cell population. There are differences in the ratios of what would be expected for T helper cells, and there's increased stimulation from the T cell of the B cell, of the autoreactive B cell in particular. This leads to increased lymphocyte proliferation, increased lymphocyte activation, and ultimately the detrimental effects of the adaptive immune system being activated. So here's a picture that shows a T cell activating a B cell that is an autoreactive B cell, therefore produces an autoreactive plasma cell that produces autoreactive antibodies. And you can also see at the bottom the BAFF helping to activate that autoreactive B cell. Here again, we have targets of our medications. So in particular, there's many targets for autoreactive B cells, from belimumab, which is going to reduce the BAFF activation of these autoreactive B cells, to bispecific T cells, to CD20 targets to deplete B cells, to even CAR T, all addressing at the B cell level. And then many of our DMARDs, mycophenolate as one example, but many of them are actually working by preventing proliferation and activation of lymphocytes. So that small picture inside the lymph node is in the center of this, and you can see all around it the aspects of the innate immune system, and you can see again that perpetual activation, the innate immune system with the interferon and the BAFF stimulating the B cell, then the B cell producing antibodies that develop immune complexes that then wind up stimulating more of the PDCs. And so that self-perpetuation that is hard to turn off. I think this brings us to what are the known differences, because I've been talking about these mechanisms which are in common, and actually the known differences are extremely subtle. So what we do know is if you look at the whole population of childhood onset lupus, we see about 80% of that population will have the type 1 interferon signature, which is a little bit higher than the adult population where it's estimated about 60%. So more than half in both, but not the exact same number. We also see some differences in how genetics interplay. So genetic loading means if you have those susceptibility loci, how many of them might a person have? Certainly could have more than one. What happens is the more of them you have, the more likely you are to develop lupus sooner. And so children tend to have higher genetic loading with more loci than adults. And then monogenic lupus, which is not really the thrust of this workshop, but important for us to discuss, is a much higher rate in children. Again, if you have one of those monogenic mutations, then you're much more likely to develop lupus that much sooner. So because we're not talking about it, I wanted to give you all a picture of what is monogenic lupus. This is one example. So SAVI is known as sting-associated vasculopathy with onset in infancy. Here is a picture of a patient with it. What happens is a gain-of-function mutation for the STING1 gene. It leads to constitutive activation and production of the STING protein, and that is a powerful stimulator of interferon genes. So these patients develop neonatal onset fevers, cutaneous manifestations, vasculopathy, and interstitial lung disease. And historically, sort of the mainstay of treatment has been JAK inhibitors because they can dampen some of the interferon signature. There's new evidence and case reports coming out, not at all surprisingly, that anifrolumab is a wonderful way of targeting this disease that is based on overproduction of interferon. So as you can see, it is much easier to target when there is a specific gene and a specific initiator of lupus. Finally, I wanted to touch on the developing immune system of infants and children. And the good news is, again, more similarities than differences to the adults. So this here shows the different cell populations in a lymph node, and you can see very little difference from infant to preschooler to school-aged. If we look at peripheral blood and we look at the cell types and subsets, again, it's not that one of these colors starts small and becomes large as it goes over age. There's some small fluctuations across age groups, but the distributions are very, very similar. And then I think the other thing to mention is immunoglobulins or antibodies. So this is well known that when an infant is born, they actually have a lot of immunoglobulins, but they're not immunoglobulins made by the infant themselves. They are mom's immunoglobulins that transferred across the placenta. So if you look at their total immunoglobulin level the day of birth, it's reasonable, but then mom's antibodies, which are in purple, are going to depreciate over time, right? The antibody breaks down, we lose it. But that happens at the same time as the infant starts to make his or her own immunoglobulins, which is the green color that starts sort of at 0 at day of life 1 and increases. So you see this nadir between month 4 and 6, where immunoglobulin levels are at their all-time lowest point. And then as the infant and then young child makes more of their own, you can see that by the age of 2 or 3, it starts to get remarkably similar to adult levels, and certainly by school age has approximated them. And then the final things to touch on, really the thymus, which continues to grow after birth through about 4 to 6 months of age. And then we spend the rest of our lives with that thymus tissue involuting. And it does so at a very linear rate. So 3% per year, which continues into mid-adulthood, certainly into the 30s. And then finally, the repertoire. So our repertoire of antibodies has to do with what pathogens have we been exposed to? And that repertoire starts to approximate an adult repertoire by the age of 5. We all know toddlers get lots of infections and they've been exposed to most of what's out there by the age of 5. All right, so just to hit the highlights here, we know that the susceptibility genes are the same between pediatric and adult onset lupus, although there may be more in children. The pathogenesis, the pathophysiology is the same. All of those things I showed in the vicious cycle are seen both children and adults. The immune system is considered mature by the age of approximately 5, and the pharmacologic targets across the immune system are fundamentally the same. And now we'll pass it off to Dr. Ardoin.
Well, thank you very much, Rebecca and Stephen. And as a physician who takes care of children with lupus and also as the immediate past president of CARA, I want to extend a wholehearted thanks to the FDA for organizing and all of you here participating in this really important effort to improve access to safe and effective therapies for children. My task today is to talk about outcome measures, disease outcomes and trajectory, and comparing this in childhood and adult onset lupus. Here are my disclosures. For some reason, the text is coming in a different color than the slides, but I hope you can read it. So my objectives here are to compare adult and pediatric instruments for measuring disease activity and damage across the lifespan, and to evaluate the use of these outcome tools in adult and pediatric lupus trials that have been conducted to date, and then to understand the similarities and differences in lupus disease outcomes and concerns across the lifespan. So if we look at disease activity measures, we all know that we use composite measures that combine aspects of clinical presentation and laboratories for patients who have lupus, both in clinical trials as well as in clinical care. And I'm going to here compare, starting with disease activity, looking at the tools used for adult onset lupus and compare them to childhood onset lupus. So as you can see in bold, most of the instruments are exactly the same. We use for disease activity the SLE-Di, both in adult and child onset lupus, the BILAG, the Physician Global Assessment of Disease Activity. Validated only in adults is the SLE Disease Activity Score, or SLE-DAS. When we look specifically at the development of flare during clinical care or a trial, we use both the SLE-Di flare index, as well as the SLE-Di flare index in adults and children, and the BILAG in adults and children. There is a specific childhood onset lupus tool which has been developed by Hermina Brunner and colleagues, the ACR provisional criteria for global flares in childhood onset lupus. We also, of course, want to consider the disease state of the children, and it has been increasingly important to assess both remission and low disease activity in both children and adults in lupus, as we know that this can correlate with long-term outcomes. So if we look at adult studies and pediatric studies, again, we use the same criteria to determine lupus low disease activity. These components of the LDAS, as it is known in the adult side, and the C-LDAS in childhood, both use the clinical SLE-Di of 0. They both require a physician global assessment of disease activity on a 0 to 3 scale of less than 0.5. What is different between the two thresholds is the target for steroid. And this makes sense because the same dose in an adult may be a very much higher milligram per kilogram dose in a child. So the glucocorticoid threshold for adults is a prednisone dose of less than 5 milligrams per day, whereas in children, it's less than 0.1 milligrams per kilogram per day, but up to a maximum of 5 milligrams per day. And then, of course, there's the same requirement for maintenance therapy. Again, with the stricter definition of SLE remission, which of course can be attained both on and off medications. In the adult side, this is known as the DORIS criteria, and recently proposed by Eve Smith and colleagues is the CSLE clinical remission criteria. So both of these, again, use the SLE-Di, requiring a SLE-Di, not the clinical SLE-Di, but a total SLE-Di less than or equal to 4 with no major organ activity. Both require a physician global assessment of less than or equal to 1. And again, we see the differences as previous with glucocorticoid threshold and stable medications. So we can see we're using these same definitions just with adjustments for dosing. Again, when we look at flare, response, and damage measures, for lupus flare, again, we're using the same indices. For treatment response, we're using the SLE Responder Index 4 for both adults and children, as well as the BICLA. However, there have been some pediatric-specific tools developed called the Printo ACR 50 Childhood Lupus Improvement Index. And then for damage, we are using the same tool, the SLIC ACR Damage Index. There has been some interest in including additional damage measures in children, including growth and pubertal delay, but these have yet to be validated. Now, where we see some differences are in the use of patient-reported outcome measures. And this makes sense because the lived experience of a child is different from that of an adult. One is in school and dealing with peers, another may be having a job and dealing with family. And in general, there are common tools using the PROMIS measures. We use patient global assessments, of course, for children, and the patient-only assessments for adults. We can use the SF-36 for adults, and then there are some pediatric-specific measures that we can use as well, including the PEDS-QL, the Childhood Health Assessment Questionnaire, which focuses on function. And then again, for lupus-specific quality measures, we can look at various instruments which have been validated in the adult population, including the Lupus Quality of Life Index, Lupus Pro, SLE Quality of Life, LQUAL, and the Lupus Impact Tracker. And the only childhood onset lupus tool specific that has been validated for children is the Smiley Quality Improvement Tool. So what happens when we actually look at what these measures use in trials? So the two trials that we have to look at for children, of course, are Pluto for the belimumab trial and for Blossom for the anifrolumab trial. And we can see for both the adult and pediatric belimumab studies, the eligibility criteria included a SLE-Die greater than or equal to 6. As we can see for the anifrolumab study, the same measures were used, SLE-Die greater than or equal to 6, or BILAG greater than or equal to 1, more than 1 BILAG A or more than 2 BILAG B, in addition to an elevated physician global assessment of disease activity. Again, the same efficacy measures are used in these trials. So for looking at disease response, for the primary efficacy for the belimumab trials, and for adults was SRI-4, and for children, again, the SARI-4. For anifrolumab, the BICLA was used for both populations. And for secondary efficacy, we're saying the same measures used as well, with the pediatric measures being incorporated in the pediatric trials. And again, the patient-reported outcome measures, as expected, were different between the two trials. Now, what do we know about the experience of children with lupus compared to adults? This has already been covered in other talks, but we know that the disease activity measured by SLE-Die or BILAG tends to be higher at the time of diagnosis for children compared to adults. We know that the prevalence of lupus nephritis is higher at diagnosis, and also the lifetime risk of lupus nephritis is significantly higher in the childhood population. We also know that the prevalence of neuropsychiatric lupus is significantly higher in children compared to adults. Now, what do we know about survival? Well, the 10-year survival depends on where you live, unfortunately. It's very high for both adults and children in high-income countries. As expected, it's lower in low-income countries, with a significantly lower survival in children compared to adults in low-income countries, likely reflecting access to pediatric-specific care. As discussed previously, the standardized mortality ratio for childhood onset lupus is significantly higher than adults, keeping in mind that in this instance, we're comparing children to their peers, which have a much lower mortality rate overall. And again, end-stage kidney disease significantly more likely to happen over the course of the lifespan in someone who has childhood onset lupus compared to adult onset. So what are the concerns for a patient for lupus across the lifespan? I would say that they are very similar, although different things become, of course, very important at different times. The three things that are particularly significant for children that may have less of an impact on adults include growth. We know that growth can be impacted both by lupus disease activity as well as prednisone exposure. Education, as of course, lupus disease activity can significantly interfere with educational attainment, as well as delay in puberty, which can also have impact on bone health. So I hope what I've shown you today is that the same preliminary eligibility and efficacy measures can be used for adult and pediatric populations. We do have to make weight-based adjustments for glucocorticoid dosing, that disease activity and damage accrual are higher in childhood onset lupus, and that the lifespan concerns are similar with the addition of growth, puberty, and education for children. Thank you very much.
Thank you so much, and thank you to all of our speakers for helping us stay on time. Our next speaker is going to be Ekemini Ogbu from Cincinnati Children's, and she'll present on the treatment paradigm, landscape of product used, and unmet needs in pediatric SLE.
Hello, everyone. Thank you for the kind introduction. So I'm very pleased to be giving this talk, and I'm thankful to the organizers for the invitation to speak. These are my objectives: to review the treatment paradigm for childhood onset lupus, briefly review the landscape of products being used in the treatment of pediatric lupus, and then describe unmet therapeutic needs in childhood onset lupus. I have no relevant financial disclosures, and of course, I will be discussing off-label use of therapies for pediatric lupus. First of all, I'd like to say, because we've had a lot of background so far about what pediatric lupus is and differences, similarities to adult lupus, we are not yet at the finish line because though our 5-year survival has improved from the 1960s, we still struggle with getting optimal clinical outcomes despite advances in therapeutics and in clinical practice and the increased availability of pediatric rheumatology practices, which is important. Thinking about the treatment paradigm for lupus, this centers around treatment principles, treatment goals, and treatment effectiveness. In essence, the ability to maintain disease remission, achieve and maintain low lupus disease activity, reduce lupus-related morbidity and mortality, and minimize treatment-related toxicities. Central to this is the early introduction of immunosuppressive therapies to minimize overall corticosteroid use. More recently, we're adopting the treat-to-target principles as part of our treatment paradigm. And basically, it's centering on shared decision-making to achieve better outcomes. So thinking of the current landscape of treatment products for childhood lupus, here on the screen, you can see medications listed down there. These are the non-biologic disease-modifying anti-rheumatic therapies we use in lupus. And you can see the different guidelines highlighted there, the 2025 ACR lupus guideline, and then lupus nephritis specific guidelines, and then guidelines from our European colleagues as well, and our nephrology colleagues for lupus nephritis. Across board, as a snapshot, you can see that many of those medications are referenced in those guidelines for different reasons. So there are nuances to when we use them in the treatment of children with lupus, but they are used in both adults and pediatric onset lupus. Similarly, this table shows medications that are used. These are the biologic medications this time, and that's anifrolumab, belimumab, intravenous gamma globulin, IVIG, obinutuzumab, rituximab. And you can see at a snapshot that these medications are referenced in the guidelines. And so in practice, in practicing evidence-based medicine, clinicians do lean on those guidelines to inform how they use the available therapeutics. But looking again, because this was discussed earlier about sort of the discrepancy in what's been approved for adult lupus in compared to pediatrics, this table just really shows glaringly the number of FDA-approved medications for lupus. These are biologic medications, and then voclosporin. And the year they were approved for adults in comparison, on the other side, you see the year they're approved in pediatrics. It's just belimumab for pediatric lupus. And you can see the timeline difference very clearly. For example, belimumab, lupus nephritis in adults in 2010, 2011, whereas for pediatrics, we had it approved just in 2019 with the subcutaneous autoinjector approved just only in 2024. Currently, you can see that none of the other biologics have been approved yet for pediatric lupus. So most therapies for childhood lupus in clinical use are still very much off-label. But we know that as we learn more about the immunology of lupus, we know on the horizon there is hope. We know that there, continues to be identification of treatment targets. And this slide here shows the multiple medications that are either under study or were in previous clinical trials. Some of them didn't quite make the endpoints and so failed to be approved. But you can see across board, and Dr. Sadun had nicely talked about the different pathways that are active in lupus. You can see here we have the T cell, B cell axis, T regulatory cells, the innate and adaptive immune system. Basically, we know a lot about lupus now, enough to distinguish new targets, create opportunities for medication, but still we are failing to take these medications to the bedside, to the pediatric patient. So thinking more about unmet needs for childhood lupus, this paper was as a result of work that was supported through CARA, the Childhood Arthritis and Rheumatology Research Alliance, in conjunction with our nephrology colleagues and dermatologists and Lupus Foundation of America. They looked at research priorities in childhood lupus to determine research gaps and propose way forward and where we should target our research efforts. If you can see here, clinical trials was one of the top 5. And then when you look at, in terms of organ domains, nephritis and neuropsychiatric lupus were part of the top 5 areas of focus. Under lupus nephritis and neuropsychiatric disease in particular, there were subtopics, and principal amongst this were development of new therapies, effective therapies for lupus. Again, reiterating that we need to address this. So at the core of our unmet needs for childhood lupus, we have to think about 2 main things: identification of effective and safe therapeutics, but also how to improve access to these effective therapeutics to get them to the bedside. On behalf of the investigators shown up there, I'm going to present our work that we recently did using the CARA registry. For those that do not know the CARA registry, it is a large longitudinal registry from over 70 active sites in North America. That's the US and Canada and some other countries outside of these. We looked at data from here to look at what the unmet needs therapeutically are for childhood lupus. So the objective was to actually quantify these unmet therapeutic needs, thinking about this in the space of off-label medication use, corticosteroid dependence, and inability to achieve low disease activity. So we looked at data from January 2017 to March 2025. We looked at all patients in the registry who were diagnosed with lupus at age less than 18 years, who also had medication exposures occurring during childhood. And the visits within the registry are done every 6 months. So a really nice longitudinal data. We defined unmet needs because we had to define that to be sure what we're looking for. And we had 5 criteria we used. The first was treatment with medications not approved for childhood lupus, treatment with medications not considered in ACR lupus nephritis or general lupus guidelines. And third, treatment with medications not used in standard of care for children or adults with lupus. And just as a reminder, I had shown that other slide where we had medications that were mentioned under different guidelines. These were some of the classes we looked at specifically. Apart from steroids, we looked at the non-biologic disease-modifying anti-rheumatic drugs, as well as the biologic agents. The 4th criteria was inability to discontinue oral corticosteroids or achieve prednisone equivalent dose of less than 7.5 milligrams per day. And we assessed this at 6 months, 12 months, 24 months, and 36 months after diagnosis of lupus. The 5th criteria was inability to achieve low lupus disease activity states at similar interval. So this is what we found. We had 1,275 patients. Of these, we had mostly female at 87%. We had a good mix of racial and ethnic diversity. And the median age in years at lupus diagnosis in our cohort was 14 years. And the median age in years at registry enrollment was 15 years, so about 1 year or thereabouts after the diagnosis of lupus. And the median disease duration in years was 4 years. The remainder of analysis focused on the subset of patients that had medication data available, and so that was 1,256 patients. Of these patients, 96% of them were treated with hydroxychloroquine at some point. 86% was treated with a non-biologic DMARD, and just about a third of them were ever treated with a biologic DMARD. So this slide shows basically the distribution of the different medications used within the registry. In the top corner, A, it shows the non-biologic agents, and you can see there about 62% of patients were on mycophenolate, and the next more frequently used non-biologic medication was azathioprine at 19%, followed closely by methotrexate, and then cyclophosphamide at 15%. When we looked at the biologic therapy in B down there, rituximab was the most common biologic medication used at 21%. Belimumab was used in 15% of patients. At least 52% of patients used at least one biologic DMARD, and 26% used at least one non-biologic DMARD. When we looked at prednisone dose by visit over time, we found that a lot of our patients are still on corticosteroids even after 36 months. And after 12 months of follow-up, a significant amount of them were still on prednisone doses, even though they were low-dose prednisone. This figure shows lupus low disease activity status in the registry, also by the same interval, that 6 months, 12 months, 24 months, and 36 months. And you can see that only a fraction of the patients met LL-DAS, and that's the yellow segment. At 6 months, just 22% were at LL-DAS. And at 12 months, and it's important to note the 12-month cutoff because that's kind of a goal for our current ACR guidelines to, really come down on the steroids that we're using. Only 30% of patients met LL-DAS at that point. And so basically, almost all patients with childhood onset lupus in the registry had substantial unmet therapeutic needs. At 12 months of diagnosis, 1 in 5 had a high level of unmet needs, as defined by having up to 5 meeting 5 unmet needs criteria. And so that's 21%. 50% of them met 3 to 4 of the criteria, which is very significant. And points again to the fact that a lot of our patients are still needing a lot of steroids despite what we're doing at the bedside with the medications that we have available. And so this slide shows very clearly that at 6 months, we have at least 56% of our patients still on steroids. And at 24 months, it's about 27% of patients still on steroids. And we have to think about the long-term exposure of steroids on the children when you think about growth suppression, delayed puberty, metabolic effects. There's a whole slew of effects of steroids, which I know is going to show up in another talk. But just to point this out about the gravity of what we're having to face and why this workshop is very relevant. And so the take-home message here is pretty clear. Attainment of treatment principles and goals for childhood lupus really hinges on the ready availability of effective therapeutics at the bedside. Our study leverages real-world data from the largest multi-center prospective registry of lupus in North America, arguably in the world, to underscore the limited effectiveness of currently available therapies. There really is an urgent need to expand regulatory approval for therapies for children, adolescents, and young adults with lupus to improve outcomes. These are some references, and I definitely would like to thank my co-investigators, and they're all in the audience, Kara and all the founders, the funders of this work. And thank you all very much for listening.
Thank you so much, Ekemini. We would like to go ahead and ask the speakers from this morning to join us for the panel discussion, as well as our two patient family representatives. If you can come up to participate for the panel discussion, I will turn it over to the moderator, Suzette Peng, who's a team leader in the Division of Rheumatology and Transplant Medicine.
For the first panel discussion, if we could we everyone introduce themselves, and then we'll kind of get into the questions. And maybe we can start with Elizabeth, if you want to start.
Hi, my name is Elizabeth Santa Cruz from Kearney, New Jersey. Since my daughter's lupus diagnosis, I have become a dedicated lupus caregiver, care partner, and lupus advocate. For years, I have worked to advance lupus research, engage in congressional advocacy, and national efforts to ensure that patient and caregiver voice shapes the future of lupus care and treatment. Hi, my name is Mia from Kearney, New Jersey. I'm an NJIT student and I was diagnosed with lupus at the age of 9. Since my diagnosis, I have dedicated my voice to advancing lupus research and patient advocacy by speaking at national meetings, participating in research and awareness initiatives, and helping ensure that the experiences of children and families living with lupus are heard by researchers, healthcare providers, and the FDA. Thank you, FDA, Kara, Lada, for inviting me to participate to represent the lupus patient.
Hi, and thank you for this opportunity. My name is Rashma Patel. I'm a pediatric rheumatologist in Central California, as well as a medical director at Biogen in one of our lupus programs. And again, thank you for the opportunity to be here.
Hello, everyone. My name is Alvina Chu. I work at AbbVie in immunology clinical development. I'm also a practicing adult rheumatologist.
Good morning. I'm Stacy Ardoin, an adult and pediatric rheumatologist at Nationwide Children's and The Ohio State University. I'll have to get that in there. And also the just ended my tenure as the president of CARA.
Hello again, Rebecca Sadun. I am another adult and pediatric rheumatologist and practicing in North Carolina.
Hi, everyone. My name is Tressa Mbukin. I'm a pediatric rheumatologist and a medical officer in the Division of Rheumatology and Transplant Medicine here at the FDA.
Hi again, everyone. I'm Ekemini Ogbu. I'm a pediatric rheumatologist at Cincinnati Children's, and I do lupus research and a lot of other things. But
Well, thank you, everyone. I guess I think Lily already introduced me. I'm an acting clinical team leader in the Division of Rheumatology and Transplant Medicine. I'm an adult rheumatologist. All right, so let's start with the questions, and then also if questions come up in the audience, we'll try to field those as well. But the first question for Elizabeth and Mia, living with pediatric lupus or as a caregiver of someone with pediatric lupus, what aspects of your disease have been most difficult to manage with currently available treatments? And what would a meaningful improvement in treatment look like?
As a mother receiving the diagnosis that my child, at 9 years old, had lupus was one of the most scary moments. It was a world of specialists, injections, lab works. It was a world of uncertainty and question about her future. One of the most difficult realities was to face that and learning how limited the treatment options were, especially for children with lupus. While there are therapies that help manage the disease, many were not developed, especially for children and pediatric lupus patients in mind. Families often have to make difficult decisions, balancing the needs to control a serious autoimmune illness, while also worrying about the impact of treatments on our child's developing body and future. As a caregiver, we also see symptoms that are not always captured in a clinic visit. We see the days when our children are exhausted, when they're struggling to focus, when they're not able to be the child they were before. Lupus fog is a perfect example. It's a symptom that can deeply affect your child's education, confidence, and daily life. Yet, it remains difficult to measure. As we know, there's not a simple test, a simple scan, a simple machine that a doctor can send you and ask and check how your child's brain is doing. As we know, as parents and caregivers, we know when your child is struggling. We also experience the importance of listening to children and families. Her joint pain was dismissed as growing pains. As parents, we know our children when something feels different, when something is off. We need healthcare providers who listens and considers that autoimmune illness can also affect children. A meaningful improvement in treatment will be having more pediatric lupus therapies that are targeted, safer, and developed with the voices of patients and caregivers included from the beginning. We need treatments that focus not only on controlling the disease activity, it's about improving the quality of life for the child. A child can have a stable labs and still struggle. Success is about giving the child a future. Success is giving the children their childhood back. We hope our voices help shape the future of lupus care so that the next generation of children with lupus has better options, fewer limitations, and more hope. Thank you, FDA, Cara, Lara, for this opportunity.
When I was diagnosed with pediatric lupus at 9 years old, I never imagined that my childhood would be changed by a disease that I never heard of. I went from being a child who wanted to go to school, spend time with friends, and enjoy everyday activities to a child who had to learn about medications, appointments, blood tests, and a disease that would become part of my daily life. One of the hardest parts of living with lupus has been that many of the symptoms are invisible. People can see a broken bone, but with lupus, people cannot always see the exhaustion, the pain, or what it feels like when your brain does not work the way it used to. Lupus fog has been one of those challenges. There were times when I struggled to concentrate, remember things, or process information, and it was frustrating because I knew what I was capable of, but my brain was not always cooperating. Lupus fog is a symptom that many patients experience, but it is often overlooked because there's no simple machine or test that can show what is happening inside the brain. Just because something cannot always be measured does not mean it is not real. I experienced joint pain that was dismissed as growing pains, and I knew it wasn't normal. Looking back, I wish there had been more awareness that children can have lupus and that their symptoms deserve to be taken seriously. A meaningful improvement in treatment would be having therapies created specifically for children living with lupus. Treatments that are safer, more effective, and designed to protect not only our organs, but also improve our quality of life. We need treatments that help us feel like ourselves again. For me, success is is not just a lab result showing that my lupus is under control. Success means being able to dream about my future, pursue my goals, and live my life without worrying about lupus. I hope my voice today helps researchers, clinicians, and the FDA understand that behind every clinical trial and every treatment decision is a child who wants the chance to simply be a child. Thank you, Vincent from Cara and Kathleen from Lupus and Allied Diseases for this opportunity.
Thank you so much for sharing. And really, that's the basis of why we're here and how we're going to look at what we just discussed in session 1. So the differences and similarities between childhood onset and adult lupus. So we'll move on to the next question and kind of get the panel's opinion. So based on the earlier presentations, I think we heard a lot about similarities and overlap, but what are the most clinically meaningful differences between pediatric onset and adult onset lupus? And which of these differences are most relevant when evaluating the efficacy and safety of therapies in pediatric patients? And how should they influence pediatric drug development strategies? I'll kind of leave it open and see if anyone wants to respond.
Thank you. So I think as we presented earlier, the disease is generally considered to be very similar across the age spectrum, with the exception of very young children who may have a more severe disease phenotype, as with monogenic lupus. And I think the clinically meaningful differences are really in the severity, in the severity of illness that we see that is higher in children, which really speaks to the importance of getting these therapies to children sooner. They should not be receiving them later because their disease is more severe. In terms of evaluating efficacy and safety, I think we demonstrated that the same measures that we use to assess disease activity or in response to treatment can be used in both populations and are used in both populations. So those are very similar.
I agree with everything that was just said. Having an interesting conversation over the coffee break earlier where I was asked about some of the manifestations that are more common in pediatric onset lupus. So more lupus nephritis, more neuropsychiatric involvement. And a very insightful question about, does that mean that there's something mechanistically different about the disease? And what I actually find to be really helpful in understanding it is a graph that shows by decade how likely patients are to have each of these manifestations. So lupus nephritis, if you develop lupus between the age of 0 and 10, the rate of lupus nephritis is about 60%. If you develop it in your second decade of life, it drops down to about 50. In every decade of life, it goes down approximately 10%. So it's very linear. It, to me, suggests not a fundamental mechanistic difference, just more severe disease in the younger presentations, which means that we have a greater need for the medications that are really effective, that are really targeted, that can really interrupt the immune dysregulation.
I was going to add to that as well. I mean, if anything, it's a resounding, there's a lot of severe disease in the younger population. It's presenting at early age. And so that's almost another reason to do these studies and get these drugs to this urgently, or this group that urgently needs them. And so I think it actually highlights the fact that pediatric population needs therapies. And this last talk just showed us all the steroids and the overuse of so many different immunosuppressants. And even rituximab, right? And so there are other drugs being used, but it highlights the need that this population needs more therapies and tools available.
Roberto, did you want to comment?
Yes, please. Yes, please. I wanted to highlight a point that I'm not clear about, because we are here recognizing that the disease requires sometimes more aggressive therapy, and sometimes it's more severe representation. So how do we tackle this difference compared to adults? Because this would mean that a treatment that is used in adults at a population level would inherently be less effective. Am I wrong?
This is Alvina. I'd like to respond to that question. So typically, and again, I can speak to the drugs that have been in development or some that are currently in development right now. Typically, the drugs for adults are being developed for moderate to severe lupus. I would think that when we're thinking about the severity of childhood disease, that it would still fall within that spectrum of moderate to severe, but perhaps fall more within the severe spectrum. So it may not be that a different dose necessarily is needed or a different medication or mechanism of action is needed if the science behind the disease is the same. It would remain to be seen, obviously, with some clinical evidence, whether or not the treatment for adults would apply to children.
And I would add that I think the problems with access to medications for children make it harder to treat, right? So having access to the effective therapies earlier we may see differences in our ability to control disease earlier. So it also has access is also a component of that as well.
And I'd add one more thing, which is that children with severe lupus, when things go well enough, grow up to be adults with severe lupus. So actually, many of the patients for whom these medications are designed and targeted and treated initially had childhood onset disease, and fortunately do respond to the medicines.
I was going to add that we know from everything we've done so far in immunology that the underlying biology of the disease is the same in pediatric and adult. And even if the clinical phenotype is more severe in children, perhaps reiterating Dr. Ardoin's point, earlier intervention with treatment might help us to really get ahead of having those very severe phenotypes early. Because the underlying biology is the same, I think there is better confidence to say that if it's effective in adult onset lupus, it is effective in childhood onset lupus.
Yes, of course, this I understand that, but so far we don't have any direct data that compared at a population level the same response in adults versus children, so to say, versus placebo. We only had the belimumab example, and in fact in there the response was a little bit lower. One may argue that it was because it was measured with SRI-4 and maybe with the Printo criteria, which underlines that the outcome measure needs to be different. This is another aspect. That could be the same, but unfortunately it was only one. I don't think that there is any direct data of comparison yet, which confines us in a little bit of an area of uncertainty. Another option, one option could be, for example, to try to look at some subgroup by severity grade in adult patients and see if they have a comparable response to the rest of the other population, or if they are identifiable subgroup of patients that could be more representative for children or adolescents. Possibly this way could be a way forward. But listening the confirmation that in any case, there might be situation, there might be reasoning for that, but there is a more aggressive disease that needs to be controlled more quickly rather than in adults. Yeah, well, it's an uncertainty to be addressed in development, I would say.
Yeah. Roberto, I think in session 2, we do talk about exposure response. And Dr. Balevic, did you have a comment or?
Thank you. No, I was actually just going to give a plug for session 2. So we are going to, I think, address this, where we're going to look at whether adults and children with lupus respond similarly to treatment at the same dose or at the same exposure level. So that'll be session 2. Only other quick comment, I'll sort of echo, I think, what the panel said, which is that even though we do see disease activity higher in pediatric lupus, I think 2 points. One, the average SLEDI score is only marginally higher. I'll have to pull the meta-analysis, but I think it was only about 2 points higher in pediatric than adults. And so while it is more severe, we have to sort of keep that delta in mind. And then 2, we know that in both adults and children, right, lupus is an incredibly heterogeneous disease. I've taken care of many adults who were much sicker at presentation than children and vice versa. So it really does for the individual patient run the full spectrum from mild to very, very severe disease. The point is taken when we approve drugs, we do so at a population level. But hopefully we'll address that more in session 2. So thank you.
Thank you. Did you have a question? Oh, sorry, Dr. Yao.
Lynn Yao, FDA. So I'm looking forward to the second or the later session on exposure or response to treatment, because I think that is a question that I have that Roberto has raised too. You know, how confident can we be that a drug that is effective in adults in a particular population will be effective in a pediatric population? Given this, I think, acknowledged difference in potentially the severity and then the lifelong risk of disease burden, right? We don't want to approve drugs that have sort of marginal efficacy in adults, and then we have confidence that that will work in a pediatric population that's more severe. So agree with that. But I had a question about the scoring, and it's mostly because I'm not, I don't understand the scoring well. I am not a pediatric rheumatologist. I'm a pediatric nephrologist. So is there any potential for differences in that composite score to potentially lead to differences in the outcome measure or the effectiveness of a drug? In other words, we've already heard that renal disease and neuropsychiatric lupus tend to be higher. So I imagine that the SLEDI scores sort of include that more on the pediatric side. But if that composite score is different or the components of it are different in the adults, do you think that could lead to meaningful differences in how we might assess outcome?
I can start with that one. So I think that we're measuring the same components in children and adults in both, you know, with all the measures that we use, the SLEDI, the BILAG. The distribution may be different. I think one of the challenges can be typically we're looking at lupus nephritis, you know, separately. So that large prevalence of lupus nephritis in children is less likely to have an impact on a trial where we're obviously looking for general lupus. And usually neuropsychiatric lupus is excluded, and so that's also excluded. Did you have another question?
Well, no, so that's exactly my question, which is the distribution of, you know, I know the components are the same, but what are differences in the distribution of those specific components between adults and children? And do you think that may affect, again, the measurement of an outcome?
I mean, I think it just all depends on the population that's being studied. And if the challenge with lupus trials, of course, is that we've tried to silo lupus nephritis trials and general lupus trials when, you know, patients generally have manifestations that kind of cross those lines. But if we silo them, I think we're comparing apples to apples when we look at nephritis. Even if it's more severe in a child, we're still looking at a delta from the beginning to the end. For an adult, I don't see that it would change the outcome. It just maybe the initial starting point. Do we know?
Well, based on validation studies, there's no reason to think that the individual items are differentially endorsed in children or young adults as compared to older adults. Biomarker studies, especially in lupus nephritis, suggest the very same thing. So also when you look at the histology, they are comparable. And I think what you see in children is that you have more multi-organ disease, and because we don't have more biologic medications, we have to resort to steroids. You know, even getting rituximab can be a challenge because it's not approved for children with rheumatic diseases. So what options do we have? We need to take more steroids and we need to use more cyclophosphamide.
I was just going to add, I think your question gets at the heterogeneity of lupus, which Dr. Balevic well described. I think about it in terms of a Venn diagram. The circle is huge for adult lupus because there's so much heterogeneity. The circle is huge for childhood onset lupus because there's so much heterogeneity. They just happen to be completely overlapping, very big circles. And if you pick any two patients of any age at all, they may be very different, but the whole spectrum overlaps fully.
Yeah, I think some of your question had to do with measurement and whether we should weight some aspects, parameters within a scoring system more heavily. I'm going to use lupus nephritis as an example. The way we measure disease activity histologically is the same in adults and in children, right? And we don't really see a difference per se, even when we apply that in clinical decision making and in clinical trials, like to Hermina's, Dr. Brunner's point. And we know we have other measures that are currently in the works to outcome measures for clinical trials. And from the get-go, it's pulling in the perspective of pediatrics as well, pediatric lupus, in trying to make sure that we're thinking comprehensively. And of course, there'll be additional validation studies. But like Dr. Brunner pointed out, the validation studies so far show that it's similar in adults and pediatric.
Yes, sorry. I just wanted to add on to what Dr. Brunner said. I'm at Columbia University in New York City and at Harlem Hospital. And I think, sorry, but to my European colleagues, but we're having new problems. I've been doing this for a while. And at this point, we're not even able to get medications that we've been using for years, like rituximab for our lupus patients. And forget about the newer medications in terms of being able to get obinutuzumab or, cephalosporin. The insurance companies are kind of fighting us much more effectively now, and we're going into AI, protocols. We can't get human beings on the phone, and the delay for our patients has been much longer. It used to be within a week or two, you could get someone to approve a medication for a severely ill patient. We're going on months now where we can't even get a human being on the phone. So the lack of FDA approval is having like real, effects on our patients, ability to get any medicine. I'm even getting pushback on MMF now, which is, unbelievable for patients who have renal disease. So the fact that we haven't approved any medications, in pediatric lupus, we're we're finally coming up against a wall where we are not able to treat the patients. And it's creating a confidence problem in the patients also, because then they're saying, well, you want to give me this medication, it's not approved, we don't trust you. It's creating a kind of a real storm that now I'm getting pushback on both sides. The patients don't trust the doctors, or they don't trust, they don't have a trust in the methodology. And we're not getting the medicines from the insurance company. So, and that's different. I mean, it's different than it used to be. And I think we're delaying treatment on these very sick patients, which is unfortunate.
Yeah, 100% agree with that. And that just adds to the cumulative damage that the burden of disease causes, as well as that steroid damage on these children that are growing. And so the bone impact that's happening. And so there's a really, really urgent need. And that delay adds to that. Absolutely.
Yeah, my question is maybe a little bit different, but it's, I think, related to the how do you develop treatments for kids who are more severely ill? So one part of the question is, are kids more severely ill because they're not arriving at rheumatologists' offices when they have milder disease? So the ones that pediatric rheumatologists are seeing are the ones who clearly have blatant disease. So that's one piece of it. But the other piece of it is with disease heterogeneity. The younger the kids are, you've said it this morning, we know this, there's a lot more genetic loading. What do you think the role of assessing that genetic background is in both the development of medicines and in the choice of medicines? It's a small population to begin with, so you'd be subsetting it further, which makes clinical trials even harder and drug development even harder, but it may be much more apt in terms of choosing the right treatment for subpopulations.
I think this is a very insightful comment and question. I hope I made a little bit of an indication with the pathophysiology that we see, that a lot of times, even if it starts with one or two genes, in the end, it's a lot of the same things that are going on. So if I could inexpensively get genetic testing on every one of my patients and just magically know all the genes. There might be a few occasions where that would be helpful and could point to some things. Most of the time, of course, we're only going to order something when it's going to change our treatment. And in the vast majority of the times, ultimately, it culminates in sort of the same immune dysfunction with the same multiple pathways involved. And so the majority of the time, we're still able to effectively treat without knowing these 4 susceptibility loci versus a different 4 susceptibility loci.
I would hope at some point we could follow in the footsteps of our oncology colleagues and have molecular genetics guide our therapeutic choices. That's certainly a vision for the future that we haven't realized yet.
I mean, part of what informed the question is the knowledge that, at least in a couple of cases, people who have responded well to CAR T therapy that's been CD19 directed have relapsed when they have B-cell reconstitution. And at least in one or a few cases, those people have been shown to have interferonopathies. So yes, I take your point that if you interrupt the cycle, you can get a benefit. But if you have components to the cycle that are necessary in order to have manifestations of disease, sometimes interrupting the cycle, if you provide all the components, it may not be enough. So sometimes I think you really need to know.
I agree with you. And Dr. Ardoin, I think the dream would be personalized medicine in the future.
Yeah, I think I was going to say the same thing, and it addresses the last question here is one of the unmet needs, right? Biomarkers, the field is going in that direction in terms of personalized medication. But we need those tools, and hopefully in the next couple of years, 5 years, decade, that might be available. But until then, we don't have any additional tools.
I think we could move on to that last question and wrap up this panel discussion so that we can go to lunch since we are a little bit behind. But Dr. Ogbu, you talked about the unmet needs and like the high steroid use and not being able to achieve the LLDAS. Do you have thoughts as to why that's happening?
I mean, the first thing is just the ineffectiveness of the regimens that we have at bedside. We use different combinations. We have our guidelines, but then we don't have the easy access to the tools, even like you've heard, like getting rituximab is a challenge sometimes, though it's the most commonly used biologic in the registry. So that's part of it. But I do think just in general, we have better science that's directing treatment targets. And we're seeing, some studies are reaching their endpoints. Hopefully, as they also are approved in adults, they are quickly also made available to the pediatric population. Otherwise, we're still going to end up with the same thing. So this is our best effort in trying to use whatever tools we have available. And we're still coming up short where a lot of our patients are still very much on high-dose steroids at time points that we think are very necessary to prevent long-term damage. And I think that's the take-home message. I forgot to mention this, but when we think about childhood lupus, put this in perspective, 1 in 5 individuals with lupus started with their disease in childhood, 1 in 5. So if you look around this room and count in fives, you can imagine how much the impact of that. If we're able to get medications to those people as well, we'll really be moving things forward and meeting really unmet needs.
Thank you. Any other unmet needs?
Just wanted to acknowledge where we started, where we heard from our patients, and our patients have clearly stated that brain fog, just feeling fatigued, perhaps feeling pain as well. Those are very real symptoms. And at least from a clinical trial perspective, we're not necessarily, we don't necessarily have good tools right now to measure and capture exactly what you're feeling when you're, for example, saying you feel very foggy. You don't think you're thinking straight. So that's certainly an unmet need that in industry we recognize, and hopefully we'll be able to continue to work with academic colleagues to help develop new tools.
I think there's one more thing that's been hinted at, but maybe not stated directly, which is the tremendous impact that this can have at a sort of critical age. So if you have a 40-year-old who's gone through their entire education and has been working in their job and then has a year or two of disrupted life due to severe disease, they can get back on track at the other side of that reasonably well. If somebody loses their freshman and sophomore year of high school and spends years of not being able to learn effectively in school and to have the social connections and has severe depression at that very, very vulnerable stage, it is often not possible to get back on track. And so it's not just more severe, but it's also more severe at this very vulnerable time point. And immunologically, there may be a narrow window where if we can hit things in the right way, effectively and early, we can actually prevent the sort of perpetuation of things that happen. And so we in pediatrics, even more so than an adult, that being able to act quickly with the right medicine and not have to go to fourth line and get there eventually may be really critical.
Well, thank you all so much for being part of this panel discussion. Roberto, sorry.
Yeah, no, just for clarity, I don't think rituximab has an indication for lupus in adults by the MEA, for sure not, because I don't think they met the primary endpoints in the study, in the randomized control study that they did. And I don't think also from the FDA, but colleagues can confirm me wrong or not. So it would be very difficult that an indication is also granted in pediatric patients without randomized controlled clinical trial data. So I'm pretty sure that I saw the reviews but they are mostly based on post-marketing data and off-label data. There's no comparative data. Thank you.
I was just going to point out that, the use of rituximab clinically is because anecdotally there has been improvement. Yes, it didn't meet the clinical trial endpoint, but we're in a scenario where most of the other medications are not even approved, and we know that best as we use them, whatever combination, we're still getting poor outcomes. And so clinically, we're able to push and still get medication like rituximab available for our patients and help some patients get better. That's the best we can do as we wait for other data and other medications to get approved. And hopefully through this workshop, we'll start to move the needle forward.
Thank you. That's a great transition. So just to kind of thank you all and thank you all for joining us this morning. So as we move through the rest of the workshop, we should kind of keep these similarities and differences in mind. And then in after lunch in session 2, we'll talk about similarities and differences in exposure response. And maybe that will kind of help us further kind of know where adult and childhood onset lupus sort of fits along that disease similarity spectrum. So I think we can break for lunch and back at 1:15? Yeah, 1:15. All right. Oh, no, thank you.
And session 2 is going to focus on evaluation of dose selection and response in adult and pediatric SLE. We're going to begin by 2 talks by FDA colleagues, Tao Liu and Jeet Ghosh. Tao Liu is a team leader in the Division of Inflammation and Immune Pharmacology in the Office of Clinical Pharmacology. And Jeet Ghosh is a mathematical statistician in the Office of Biostatistics at the FDA. And they'll provide a presentation focused on the pediatric benlimumab program. So I'll go over.
Thank you, Lily. Good afternoon, everyone. It's my great pleasure to be here today. My colleague, Dr. Gotsch, from the Office of Biostatistics, and I will co-presenting to share our experience with belimumab, the only FDA-approved treatment in pediatrics with SLE. We will be also highlighting the key lessons we learned from both clinical pharmacology perspective and a biostatistical perspective, and how this insight can impact future SLE drug development in pediatrics. Sorry. The other way. So before we begin, this is our standard disclaimer slides, noting that the views presented here today are our own and do not represent FDA's official guidance. To give you some background, belimumab is an IgG-based BLyS monoclonal antibody. It was first approved for adult patients with SLE in 2011, supported by 2 pivotal clinical trials, BLyS 76 and BLyS 52. Concurrently with the adult approval, a pediatric trial was required as a post-marketing requirement. This pediatric trial was originally slated for completion in 2016. However, due to significant recruitment challenge, the FDA agreed to extend the timeline and amend the protocol, reducing the enrollment target from 100 subjects down to at least 70. The trial was eventually conducted from 2012 to 2018. In April 2019, belimumab was officially approved for pediatric subjects 5 to less than 18 years of age. This milestone arrived 8 years after the initial approval in adults. Today, we will look at the 2 pivotal efficacy trials in adult following IV administration, BLyS 76 and BLyS 52, and the pivotal clinical trial that supported the pediatric IV approval, and the BLyS sub-Q trial that supported the subcutaneous route administration in adults. I will walk you through a cross-study comparison of the trial design, patient characteristics, efficacy response, and pharmacodynamic response. Finally, we will discuss the key lessons we learned from comparing these SLE trials across adult and pediatric populations. The 4 pivotal clinical trials we were discussing today shared a remarkably similar overall study design. As you can see on the slides, they were all conducted as randomized double-blinded placebo-controlled trial. A key shared feature across these clinical trials was the inclusion of a steroid tapering phase, which generally took place between week 24 to week 50 or week 54, depending on the specific protocol. Finally, for all these 4 clinical trials, the primary efficacy endpoint evaluated was the SLE Responder Index 4, or SR4, measured at week 52. Now let's take a look at the patient characteristics. Across all 4 clinical trials, patients were diagnosed using the American College of Rheumatology criteria. They entered clinical trial with very similar levels of active disease, defined by a baseline SLE score of 6 or 8 or higher. Consequently, the baseline disease activity was highly consistent across the adult and pediatric patient population. In terms of seropositivity, patients were required to have positive anti-nuclear antibody and all anti-double-stranded DNA antibody. Across the trials, approximately 90% of patients were NA positive, and about 70% of them were anti-double-stranded DNA positive. The background standard of care was also remarkably similar between the adult and pediatric populations. Systemic corticosteroid use was covered nearly in all pediatric patients at baseline, around 95%, compared to 70 to 90% in adults. Antimalarials, primarily hydroxychloroquine, were used by approximately 80% of pediatric patients, compared to 60 to 70% in adults. The use of other immunosuppressants, such as azathioprine, mycophenolate, or methotrexate, as well as NSAIDs, was highly comparable across both populations. Finally, the exclusion criteria were also identical. Patients with severe active lupus nephritis or active central nervous system involvement were all excluded from all 4 clinical trials. Concomitant use of IV cyclophosphamide or any other B-cell depleting agents were also strictly prohibited. When we look deeper into the specific organ manifestations, we see a striking similarity in the clinical characteristics between the pediatric and adult cohorts. In both populations, mucocutaneously immunologic and musculoskeletal manifestation were the three dominant organ involvement, affecting 70 to 90% of patients in both adult and pediatrics. Conversely, other organ involvement, such as cardiovascular, respiratory, hematological, and vascular involvement, were much less frequent, each occurring in less than 10% of the patients. Finally, as you can expect it, based on the exclusion criteria, active central nervous system and renal involvement were less frequent across all of these pediatric and adult patient populations. Now let's dive into the efficacy outcomes. First, it's crucial to establish that systemic exposure was comparable across all the clinical trials, regardless of the age groups or the route of administration. This means the clinical and pharmacodynamic response we're comparing today are truly at an apple-to-apple comparison. Looking at the primary efficacy endpoint, SRI-4 at week 52, the treatment benefit was remarkably consistent. The odds ratio for the achieving an SRI-4 response ranged from 1.5 to 1.8 in both adult and pediatric trials, demonstrating that the pediatric patient benefited from belimumab to a similar magnitude as adult. Furthermore, if we discontract the SRI-4 into its three individual components, the 4-point Selena SLE disease activity index reduction, or no worsening in the physician global assessment, and no new B-like flyers, the treatment effect were highly consistent across both age groups. However, we do observe some nuances where we look at the corticosteroid tapering. Pediatric patients were slightly likely to successfully taper their steroid compared to adults. This was partially driven by the different in clinical trial design. The pediatric trial was evaluated tapering in all patients on baseline corticosteroid, whereas the adult patient clinical trial only analyzed patients who started on a clinical significant dose of greater than 7.5 milligrams per day. Additionally, pediatric protocol did not mandate tapering below 7.5 milligrams per day threshold. So more broadly, we observe a more aggressive steroid use in pediatric patient populations, which likely reflect the clinical reality of patient practice, where physicians are more highly protective against pediatric flyers. Finally, the disease flyers data revealed a very interesting pattern. Pediatric patients in the placebo arm experienced a significantly higher rate of severe disease flyers compared to adults, about 32.5% versus roughly 20%. Interestingly, because the pediatric placebo flyers rate was so high, pediatric patients also showed a substantial larger absolute benefit from belimumab in reducing severe disease flyers. There was simply more room to show improvement, highlighting the high unmet medical need in this younger age group. So lastly, let's take a look at the pharmacodynamic response. By binding to soluble bleeds and reducing B-cell activities, belimumab decreased the serum IgG and anti-double-stranded DNA antibody levels, while simultaneously increased complement 4 level. Across all these PD markers, we observe a striking consistency between pediatric and adult cohorts. This consistency applied not only to the ultimate magnitude of the change, but also to the overall time course, with both populations following the same biological trajectory. This identical PD footprint provides a strong mechanism evidence that the biological drug effect of belimumab is fully translated from adult patients to pediatric patients. So to summarize the comparative findings across these clinical trials, first, patient population are remarkably aligned. Pediatric adults, SLE patients entered this clinical trial with highly similar baseline disease activity and seropositivity. Furthermore, their background treatment, including the corticosteroid, antimalarials, and immunosuppressant, is closely matched. Second, the placebo group behavior are generally consistent with two key exceptions. Pediatric clinical practice tend to favor more steroid use, and pediatric patients experience a significantly higher rate of severe baseline disease flyers using the standard of care. Most importantly, the therapeutic response is highly consistent. when we look at the clinical efficacy as SL4 or the underlying biology as a PD markers, pediatric patients respond to belimumab in the same exactly manner and magnitude as adult. With clinical and pharmacodynamic foundation in place, I would like to hand over the presentation to my colleague, Dr. Gotsch. He will share the belimumab pediatric approval from a biostatistical perspective. Dr. Gotsch.
Okay. Yeah, thank you, Dr. Liu. Now, and also good afternoon to everyone. Building on the disease similarity and the response to treatment similarity pattern he just outlined, I will now focus on the statistical challenges that made the Bayesian borrowing from adult not just attractive, but also essential. The Pluto trial, which is the pediatric study for belimumab, was not designed to support formal statistical hypothesis testing on its own. This is because pediatric SLE is rare enough that a placebo-controlled phase 3 powered study was not simply feasible. So despite enrolling for more than 6 years, only 93 patients, pediatric patients could be recruited, and waiting for adequately powered cohort would have delayed the potential treatment for the children for a very, very long time. So with that reality reframes how we think about the statistical approach. So Bayesian borrowing wasn't simply a methodological preference here. It was one of the scientifically sound paths forward to support regulatory decisions without compromising any evidentiary standards. Okay. Now, if we look at the trial similarity results, the 52-week treatment period of Pluto, or the pediatric study, was randomized and double-blind, comparing belimumab 10 milligram dose versus placebo. The primary efficacy analysis of SRI response rates at week 52 in the adult and pediatric studies are given in the table. The adult study showed clear statistically significant efficacy for 10 milligram with odds ratio of 1.5 and 1.8, both with confidence intervals excluding 1. In the pediatric study, the observed odds ratio was also 1.5, with placebo rate of 44% and a treatment rate of 53%, nearly identical to the second adult study, BLISS 52. So the pediatric results are directionally similar to the adult study, but with only 93 patients, the confidence interval was very, very wide, 0.6 to 3.5, which is inconclusive on its own. So this raises a natural question. If the disease and the drug's mechanism of actions are similar between the adults and children, can we formally leverage the adult information into the pediatric trial to supplement some of the pediatric information? Now, the answer is yes. This is basically the pediatric extrapolation approach, which Lily and Emily already mentioned in the morning session. And this is precisely what we do here. Rather than throwing away the adult data as irrelevant, we can use it scientifically justified way. The extrapolation framework relies on a core principle. So when disease biology, pharmacokinetics, and clinical response patterns are sufficiently similar between the adults and children, adult evidence can legitimately supplement the pediatric data. This is consistent with the ICH 11 and FDA's pediatric extrapolation guidance. What Bayesian borrowing method adds here is a formal, transparent, and quantitative mechanism for doing this. Now the question is how we do the Bayesian borrowing. So it mainly has 4 steps. First, we summarize the adult data. For belimumab, we had 2 adult studies. So we use a meta-analytic way to combine these 2. studies, and the combined odds ratio was 1.67. In the second step, we set a borrowing weight W, ranging from 0 to 1, where W equal to 0 means we are using only the pediatric data. We are not borrowing anything from the adult. When W equal to 1, we are fully relying on the adult data. That is, there is no discounting on the adult data. The weight controls the degree of borrowing, and it forms the basis of our prior knowledge. FDA's Bayesian draft guidance also mentioned that the prior should be discounted to avoid the adult data overwhelming the pediatric results. The prior used here for the belimumab pediatric study exactly does this. It automatically borrows less if the pediatric data look very different from the adult data, adult studies. In step 3, we blend our prior knowledge from the step 2 with the 93 pediatric patients' results to provide a posterior estimate. And finally, in step 4, we ask the most important question, which is, what is the posterior probability of efficacy, and is it greater than 97.5%? This is equivalent to traditional one-sided p-value of less than 0.025 test. And here I have the results of the Bayesian borrowing. So we tested for every borrowing level from W equal to 0 to W equal to 1. At W equal to 0, that is the pediatric data alone without borrowing any adult information. The credible interval was 0.6 to 3.5, which is inconclusive. If we increase the W at W equal to 0.55, we reach the tipping point where for the first time, the credible interval excludes 1, which achieves the posterior probability of threshold of 97.5%. Again, this is equivalent to the one-sided p-value of 0.025. At that weight, the prior from step 2 in the previous slide is equivalent to recruiting additional 466 pediatric patients into the trial. So we can quantify the prior knowledge in terms of equivalent additional subjects needed. Now, what is the conclusion of that? Now, combining with the favorable secondary endpoint results, this was sufficient to support expanding the indication to children 5 years and older. In conclusion, belimumab was the first treatment approved for the pediatric SLE in the United States in 2019, which demonstrates that first-in-class approval does not preclude any extrapolation-based approaches. Bayesian pediatric extrapolation allows flexible data-driven borrowing when disease and response similarity can be demonstrated, reducing the evidence burden from small or rare pediatric population without compromising any evidentiary standards. Because the analysis was post hoc, pre-specification of the borrowing weight at the study design level would have strengthened our credibility, and early regulatory engagement is essential to agree upon prior information as well as the model design. Calibrating the degree of borrowing requires statistical, pharmacological, and clinical input, and the sensitivity analysis also should be pre-planned to assess the robustness of the conclusion. We see this case as a replicable template for pediatric disease where large trials are not feasible. Thank you very much. And here are the list of references that we use here, and also here are the acknowledgements. With that, now I will hand the podium to Dr. Balevic. Yeah.
All right, while we're waiting for the slides to get loaded, I will just say that I have a very enviable task, which is to characterize exposure response against 9 different drugs. So looking forward to reviewing some of this data with you. I think, let's see. Yeah, I think this, there we go. Okay, so we're ready to get going now. So again, listed here my conflicts of interest and disclosures for your review. And so the purpose of exposure response analysis is that it can provide really strong evidence for similarity of drug effects and response to treatment. So the data from this study, or that I'm going to present, actually comes from a study that we worked on with many collaborators here at FDA. So I'll point you to this manuscript to learn a little bit more. But in terms of what we did, so we did a literature search to look at drugs used to treat both adults and children with lupus. And these studies had to describe either an exposure response, so drug levels and response, or a dose response effect. And they had to have some shared outcome measure. We limited ourselves to what was just publicly available, so what we could extract from tables, charts, and figures. So we didn't use any private data, for example. So what we found was that there were a total of 6 drugs across 16 studies. So again, this is a lot of data to try to summarize in 15 minutes. So I'm going to hit the high points. But again, happy to answer any questions that arise later on in the talk. Now, what I will say is that we have a bonus drug here. So we did this study back in 2022, 2023, and a prednisone study was just published last year. So for fun, I included that at the end of the talk as well. So for each of these drugs, again, this is going to be rapid fire. This is going to be like drinking water from a fire hose. I'm going to give a quick snapshot of the drug. So mechanism of action, FDA or on-label, off-label uses, and ontogeny. So ontogeny is a super important word to talk about maturation, in this case, drug targets. So do children have the same drug targets as adults, so the same levels of these drug targets, and then selective studies. So the first drug we looked at, not surprising, is belimumab, but since we just had a fantastic presentation on this, I'm going to skip over it. I am just going to point out one thing about ontogeny here. So the bliss levels in children were similar to those in adults. So again, no maturation difference that we saw here. Other thing I'll point out is in the belimumab trial, we did see a higher placebo response in children, but the net effect was actually the same between pediatric and adult studies. So the first drug I want to spend some time talking about is mycophenolate, because we use this drug a lot. So the active metabolite mycophenolic acid is an inhibitor of this enzyme, IMPDH, and this results in inhibition of T and B lymphocytes. And we use it extensively off-label in both adults and children with and without lupus nephritis. So IMPDH activity does not differ, thankfully, between healthy children and adults. It's reassuring. In terms of studies, there were several studies. I'm going to present 2. So the first was an exposure response. So we have one prospective study in adults, one retrospective study in children, and they looked at mycophenolic acid AUC, or exposure, and the same outcome measure, SLE-I. And then we have a clinical trial, which I'll go into more detail later. So this is the exposure response data, and I do have a pointer. So what we have here on the y-axis is our disease activity measure, SLE-I. X-axis, drug exposure, mycophenolic acid AUC. The blue line here, which you can see is the adult exposure response fit. This is the lowest curve. And then the black line is the pediatric fit. And so what you can see from these curves are 2 things. One, there is an exposure response relationship. As mycophenolic acid AUC increases, SLE-I decreases. And then you can see that these curves are pretty similar. Now, related, I think, to one of the questions from session 1 is, well, are patients with severe disease excluded from some of these studies? Again, this is not a clinical trial, but in patients with active lupus, the average SLE-I was about 13.5 in children and about 11.5 in adults. So severe disease. And about probably half of about 25 patients, both adult and pediatric, had that high disease activity. So what we wanted to do was put an equation on this. So we did something called PD, or pharmacodynamic modeling, which allowed us to estimate this really important parameter, AUC 50. And so this is sort of a measure of drug potency. I'll skip all the modeling methods and just let you know that the AUC 50 was highly similar between adults and children. And then we actually looked at age as a covariate. And while it was statistically significant, it only explained 2.5% of the between subject variability and potency. So this allowed us to sort of mathematically conclude then that there wasn't a clinically meaningful difference between mycophenolate potency in adults and children with generalized lupus activity. All right, so I'm going to spend a moment talking about this clinical trial because this has a lot of data for us. So the ALMS trial was a randomized trial in lupus nephritis. Okay, so we're leaving generalized lupus, looking at lupus nephritis. And what was amazing is, we'll talk about study designs later, but they had 346 adults, but they co-enrolled 24 adolescents. And so this allows us to kind of compare the response between adolescents and adults. And so in the study, 2 phases. So there's a 24-week induction phase where mycophenolate and IV cyclophosphamide were compared. Patients were randomized. And those who responded, this was a composite measure of renal response, patients were re-randomized to receive either mycophenolate or azathioprine as maintenance. Now, again, what's great, same enrollment criteria, same visit schedule, same outcome measures, right? So it allows for a really nice apples-to-apples comparison. So we also get a look at 3 drugs, mycophenolate, IV cyclophosphamide, and azathioprine. So first looking at mycophenolate in terms of induction. So because we did not have the raw data, we couldn't do hypothesis testing. But what we can do is just kind of look at the numbers. And what you can see is that the number of responders was at least numerically higher for adolescents and adults. So 70% of adolescents responded, 55% of adults. Okay. Now, how about mycophenolate for maintenance now of lupus nephritis? Again, treatment failures were highly similar across the ages, 12.5% versus 16.7%. Okay, so again, numerically, we're not seeing a major gap here. So again, this directionality, I would say, argues pretty strongly for adolescents having a similar response compared to adults. Now, how about switching gears, new drug, right? Cyclophosphamide, this is a prodrug metabolized to an alkylating agent that has a cytotoxic effect to lymphocytes and other cells, used extensively off-label with and without lupus nephritis. So from an ontogeny perspective, as Dr. Sadun mentioned, lymphocyte count function really approximates adult values by late childhood to adolescence. So by the time most children develop lupus, not a significant difference. We're looking at that same clinical trial we talked about. And again, renal response, highly similar, 57% versus 52%. So nothing that, at a 10,000-foot view really causes any concern. All right, next drug on the list, same clinical trial, but now azathioprine. Okay, azathioprine is also a prodrug because we love prodrugs. Gets metabolized to an active metabolite, gets incorporated in DNA, reduces purine synthesis, stops lymphocyte proliferation. Once again, used off-label in both adults and children. And now here's where we saw the only potential, and I highlight potential difference, was in maintenance of remission. So in adolescents, 62% experienced a treatment failure with azathioprine compared to only 29% of adults. Now, again, we have to acknowledge that these are very small sample sizes in adolescents, right? 8 adolescents. So, we have to be cognizant of that. But here's also what was interesting, right? In clinical practice, right, we use mycophenolate over azathioprine in most cases for maintenance of remission in lupus nephritis. And we saw that preferential response in both adults and adolescents. So in this clinical trial, adults on MMF had better maintenance of remission compared to azathioprine. Same thing in adolescents. Adolescents had a higher rate of remission compared on MMF or mycophenolate compared to azathioprine. So we still see, right, a similar exposure response relationship. And actually, I guess I should comment and correct myself there. This clinical trial was a dose response, not an exposure response, right? Because they did not look at drug levels. So this was a dose response relationship. All right, I promised rapid fire. We're almost to the end there. There's a couple more to talk about. My personal favorite drug, hydroxychloroquine, has multiple mechanisms of action. For our purposes, we'll focus on toll-like receptor inhibition. It's FDA approved in adults, but used extensively off-label, really the standard of care in children with lupus. And we use the same weight-based dosing in adults and children. TLR7 function approximates adult values by 2 to 5. Again, not worried about a maturation effect here. So there was one retrospective pediatric study, one prospective adult study. And what they did is they compared whole blood hydroxychloroquine levels, which we see here on the y-axis. These are random levels, by the way, and then disease activity. And what I'm going to do is point our attention to the left of these dotted lines, okay? And what you can see, this is comparing patients who have active lupus, adult versus pediatric. And you can see there's huge overlap in blood levels here, okay? Now, if we look to the right of this curve, these are patients with inactive lupus, okay, adult and pediatric. Again, you see wide overlap in concentrations. And then what's interesting, if you look, adults with inactive lupus tend to have higher blood levels than those with active lupus. And same thing in pediatrics, higher blood levels in those with inactive disease compared to those with active disease. Now, again, this particular data is fairly limited, but again, at a 10,000-foot view, it suggests at least a similar exposure response. Rituximab, I'm going to skip over. I know this is one of everyone's favorite drugs for the sake of time. And this is largely because we don't have great data here. So we have 2 adult clinical trials and then a number of observational pediatric studies, but the pediatric studies were mixed. So different rituximab dosing, number of doses, outcome measures, baseline characteristics. And so it was really hard to compare to the adult data to do an apples to apples. Doing the best we can, we put this together in a table, again, which you're welcome to reference in our manuscript. But again, at a 10,000-foot view, when you look at DNA antibody, C3, C4, so our biomarkers of lupus disease activity, again, they overlap between adult and pediatric studies. All right. And so we are now on our last drug. This is prednisone or prednisolone. Multiple mechanisms of action, again, for our purposes. One of the main ones is inhibition of NF-kappa beta. It has a very broad immunosuppressive effect, though. And it turns out there was one prospective observational study published last year out of France where they enrolled 34 children. And this is one of our younger age cohorts, down to 6 years, and 32 adults. And so what they did is they looked at cumulative AUC, so the total amount of prednisone somebody got over a 3-month period, and they did measure drug levels. And then they evaluated SLE-I, again, our disease activity measure, at month 3. A lot of data here, but I'll again walk you through this. So on the left here, we have total prednisolone, okay, prednisolone, the active metabolite of prednisone. Here we have free or unbound drug. This is a drug that's pharmacologically active. It's actually impressive they measure this because most studies don't measure unbound drug. And they dichotomize patients into non-responders or responders based on this SLE-I index. And what's interesting is that they actually did not see an exposure-response relationship. So higher prednisolone did not seem to indicate responder versus non-responder status. But for our purposes, what's really important is they looked at age as a covariate, and it was non-significant, right? So they did not see a differential effect in adults and kids in terms of exposure response with prednisolone. So now, again, as with all these studies, there are some caveats and some limitations. They also did not see an exposure-safety relationship, which I think is a bit unexpected. So it's possibly too short of duration or underpowered for some of these outcomes. But nevertheless, adds an additional level of reassurance. So last summary slides. I know it's a whirlwind, but to try to bring it all together now, we found that there's actually very good high-quality clinical trial data to suggest the response to belimumab is highly similar. I didn't show you that. Our last speaker did. Response to mycophenolate. Also, we have pretty good high-quality evidence suggesting the response is similar between adults and children with lupus nephritis and likely SLE without lupus nephritis. And then all the other drugs we can kind of lump together, right? Because the preponderance of all the data, all the directionality, even with some limitations in the study designs, every drug except with a possible exception of azathioprine suggests adults and children respond similarly to treatment. And so this is several different drug classes, several different mechanisms of action. So The only thing I would say as a possible exception, almost all the, in fact, all the studies we looked at were in children typically greater than age 5. So we don't have exposure response data in children less than 5. Exposure response may be similar. They may not be. We just don't know, at least based on this study. Reassuringly, the ontology of the immune system suggests treatment response is unlikely to differ based on age, again, with this caveat of children 5 and older. And so therefore, we really should be leveraging adult data, which is why we're here to guide pediatric drug development. And with that, I'd like to thank my collaborators, and I'll hand it off to the next speaker. So thank you very much.
I assume I have to go all the way forward, correct? Yeah, there we go. Thank you very much. And first of all, good afternoon, everyone. Let me just begin first by thanking the organizers for the invitation to participate here today, and also to thank them for a really well thoughtful and a very timely workshop on this very important topic. My name is Vikram Sinha, and what I'm going to do over the next few minutes is talk to you a little bit about the role of model-informed drug development. Many of you in this room have probably heard this terminology. It's called MIDD, in how we can use this framework to accelerate drug development and optimize dose selection. In many ways, the preceding talks have spoken in great detail about this concept, and you've actually seen very good examples at work here that illustrate this concept. These are my disclosures. So today I'm going to cover maybe 3 sections very quickly. The first one, why pediatric SLE is an ideal case for MIDD. This has been spoken to at great length, but I'll just recap a few important points. I'll spend most of my time talking about dosing in children and predicting and assessing pharmacokinetics in children. And finally end by talking a little bit more about exposure response analyses and ending on the role of MIDD for new therapies. I want to convey a couple of key concepts for you. One is that the term MIDD is just not about model and mathematics. It's actually a framework that actually helps us increase scientific confidence and, if possible, minimize pediatric trial burden and shorten development timelines. And I think that's the framework in which we are actually approaching this entire workshop. This slide, I think, has been spoken to at great length this morning. Indeed, it's many of the cases for lupus are childhood onset. It tends to be more severe, and it can be characterized as a rare disease, making the conduct of large phase 3 trials very difficult. But what is MIDD? MIDD is an approach that allows us to integrate concepts of clinical pharmacology, pharmacometrics, systems biology, and regulatory science to inform dose selection, trial design, and extrapolation. The scientific basis for extrapolation in pediatric SLE has been spoken at great length before. I think it meets at least 2 of the key 3 criteria that are needed. One, the disease progression being sufficiently similar. The mechanism of action is expected to be comparable. And finally, if the first 2 are met, we can aspire to achieve the same concentration in children to get the same clinical benefit. In pediatric SLE, much of the disease pathophysiology is shared. And so MIDD is now using these 3 fundamental concepts to predict what would likely happen in children. So how do we actually dose children, and how does modeling help us? The key question we are looking to answer here is what dose in a child will give us an adult equivalent concentration. So for this, we primarily are using concepts of allometry and organ maturation to understand what would happen in a child across the age range from birth till about 18. And what you can actually see here in the green line is that fundamentally after the age of 2, the clearance for any given drug based on size and maturation function tends to be very similar in the child with respect to the adults. But that we mean that if we gave the same dose adjusted by factors of body weight and maturation, we would get typically the same concentration. And I think the previous speaker also gave many examples that alluded to this concept. But this is something that can be taken into account, and it can be taken into account with a great deal of confidence if you have a good adult data set. Another concept or another platform that has seen more recent use is the use of physiologically based models. These are pharmacokinetic models, which tend to integrate many of these different aspects and give us a prediction of the clearance in different age groups, which allow us to then assess what dose would be meaningful in that particular child. Pharmacokinetics is one aspect of dosing in children. The second aspect is response. And the previous speaker from the FDA actually spoke about the similarity of response they saw in the one drug that has been approved for treatment. But this comes essentially from the concept of exposure response analyses. Exposure response analyses ask the key question of what concentration in a child will result in the same therapeutic effect as in the adult. And this is a fundamental question I think we are looking to answer in any pediatric program. So an exposure response analysis is allowing us to select a dose by age group. It's allowing us to establish effective concentration thresholds based on adult data. Hopefully, it's a way to reduce trial size. We didn't actually see exposure response analysis do that for the example that was shown by Dr. Ghosh on Liu, but indirectly, it does so by reducing the number of arms one needs to test in any given pediatric trial, and hopefully a way for us to integrate and improve safety assessments. How does it actually do this? It does this by integrating concentration, biomarker response, and clinical outcomes. So what is the framework that exposure response analysis uses? The first principle is the adult exposure response data. One, we have, as was shown, a large volume of dose-ranging data from adult studies. These studies are then used for us to characterize what we would call a therapeutic window. PK modeling predicts the concentrations in a child, adjusting for weight and other covariates. We use simulation approaches and Bayesian approaches, as was shown in the particular example by Dr. Ghosh, to design a particular trial. We confirm the exposure in children, and finally look to optimize doses before entering large clinical trials. The whole concept of exposure response analysis is geared to reducing the burden that we would need in a particular pediatric population, as has been shown. I will not touch upon this example in great length. It's actually been talked upon quite extensively. But what perhaps may be worth referring to and can't be seen very well here is the dosing recommendations that came out of the Pluto trial. So the adult dose was 10 mg per kg given intravenously, but the dose in children was actually a body weight-based dosing that was stratified based on body weight below and above 40 kilograms of body weight. So this is the framework that actually was used in designing the Pluto trial, selecting the doses in the Pluto trial, and reducing the number of arms that one needed to test in children. So this then takes us to what perhaps could be a generalized pediatric SLE development workflow. And I put this up maybe in the lens of those who are developing drugs in this space and what they need or could consider as these programs are maturing. One is foundationally the role of the adult exposure response data. This is foundational. This allows us to understand the therapeutic window. And as this is happening in the phase 2 and phase 3 domains of the adult program, the pediatric strategy and design begins for any pediatric program. Most drug developers will then enter into some discussions with regulators and get agreement on pediatric plans, try to establish whether a full extrapolation approach is feasible, and start working on age-appropriate formulations. A third piece of this is then scaling the PK from the adult data to predict what doses would be needed in adults, and then design some kind of a PK-guided dose escalation study in children, and finally move to an efficacy and safety study looking with specific endpoints. The point below is trying to actually get a sense of the size of these different programs. The adult foundation is typically built on hundreds of subjects. The pediatric study design then doesn't require actually any subjects. It can be done in the world of simulations. And finally, the scaling and dose selection in pediatrics requires relatively small cohorts to confirm PK in a given population. In addition to the framework of predicting doses, MIDD has some additional benefits. One is that it is actually limiting the number of procedures that a child may have to undergo in the context of clinical trials. Because of the methodology being used with MIDD, one can use sparse sampling approaches. The sparse sampling approaches now have become greatly advanced that allow remote sampling approaches. The child often doesn't need to come into the clinic to get these measurements. But the value of this is ensuring that we get some PK data in that efficacy trial that allows the confirmation of the exposure response analysis. I think many speakers have already spoken about this. We have many new treatments emerging in this particular space. Many of them are antibodies. We have new CAR T-cell therapies emerging, dual B-cell targeted approaches, and immune reset strategies. These are new frontiers that are being explored in SLE. Many of these drugs are antibodies. The questions remain the same. What concentrations we target? How do developmental changes influence pharmacokinetics? Can biomarkers bridge efficacy? And how much pediatric data is required? The one point I'd like to just make here is that for antibodies outside the very young child, that is less than 2, we usually have a very good handle in predicting concentrations. A standard milligram per kilogram approach of dose adjustments works very well to match concentrations in the adults. We do not need extensive confirmation of this PK data in the child. So this actually is an avenue to actually think about how we could get into that pivotal section of confirming efficacy in children rather than PK studies, which takes its own time and can be very time consuming for any drug development program. So beyond exposure matching, and I put this up to actually just leave where the future may be headed towards. There has actually been significant progress, and I think we've seen this. We've actually seen an example from an observational study today where the authors shared data from long-term exposure to many treatments. This is allowing the use and development of disease progression models. We're getting more and more insights with quantitative systems pharmacology models, which allow us to bring in these mechanisms and interrogate what would be an effect of a target being modulated. And finally, a lot of real-world data is coming to the forefront. What this is all allowing us to do is perhaps understand what is the likely treatment effect in a child before we even embark on a particular program. I personally believe this is going to be the future. It's coming very, very soon. And I think anyone who's interested in this particular aspect will be thinking about bringing many of these aspects as part of their drug development programs. Thank you for your attention, and I look forward to answering any questions as part of the panel.
Thank you so much, Vikram, and other speakers. I'm going to go ahead and invite our panelists to go ahead and come and join us for the panel discussion. And for those who have not had a chance to introduce yourself, I will give you about a minute to briefly introduce yourself so we can get started. Maybe we can start with Laura, if you can introduce yourself.
Yes, good afternoon, everyone. My name is Flora Musamba. I'm not sure my camera is working, actually. I'm sorry I couldn't make it in person for the meeting, but it's a great pleasure for me to join the panel discussion. Thanks to the organizer for the invitation. I am an assessor at the Belgian Medicine Agency, and I am involved as a member of the Methodology Working Party and the Scientific Advice Working Party at the EMA, and also as a member of the Network Data Steering Group. And I have been also involved in the drafting of the ICHM 15 guideline, which is the one related to model-informed drug development. Thank you.
Thank you, Flora. Laura?
Hi, I'm Laura Schanberg. I'm a pediatric rheumatologist at Duke and have been involved with CARA since its inception, including as original PI of the CARA registry. I'm involved in clinical trials in lupus and in JIA.
Amit?
Hi, my name is Amit Golding. I'm an adult rheumatologist, and I'm a clinical team leader in the Division of Rheumatology and Transplant Medicine.
Hi, my name is James Travis. I'm a statistician here at FDA, and I lead I'm technical lead for the Pediatric and Maternal Health Statistical Scientists.
Hi, good afternoon. I'm Jianmeng Chen. I'm a clinical pharmacologist. I'm currently Associate Director for Therapeutic Review in Division of Inflammation and Immune Pharmacology in Office of Clinical Pharmacology.
Thank you. Mckenna, we're going to start with you. And so the first question is to you. Has there been a time when a treatment seemed to work differently than expected? And what would you want the scientific community to better understand how treatment feels different at different stages of childhood?
Yeah, first of all, I just want to thank you again for the opportunity to be on this panel. But I would say, so when I was diagnosed, I was started on prednisone, Cellcept, Plaquenil, rituximab, and recently IVIG. But the rituximab, Cellcept, and Plaquenil, all of those treatments were started with the intention of managing my lupus. And from that perspective, I would say they've worked exactly how we expected them to. So from that perspective, it has been great. A little bit repetitive with some earlier points from my earlier remarks. There have been some unexpected side effects. For me, that's been mostly the side effects of the treatment. So with the steroids, dealing with some of the bone and cartilage damage I had, and the rituximab, dealing with some of the immune deficiencies. That's been a really challenging aspect. Going into those treatments, I was told of the side effects. I didn't think I would appreciate how bothersome they could be at times and how severe they could be. So I would say, for me, that's been the main aspect of how they've worked differently than expected. I will also add, and this was an excellent point brought up earlier, dealing with insurance and scheduling and a lot of those like external factors, not necessarily the treatment themselves, has also been a very unexpected side of those treatments. And honestly, a little bit of a frustrating one to manage. So I think that that's an important point to consider as well. For the second point, this is a little bit more of a challenging question to answer. For me, the treatments have felt very consistent for the 7 years that I've been on them. When I think back to what's varied, it's mostly been points in my life. For example, a point in undergrad got very stressful and my lupus symptoms started to get more severe, so we had to change doses, specifically for my Plaquenil. But I would say in terms of the treatment themselves, those have been pretty consistent during the different stages of my childhood and now into early adulthood. So.
Thank you so much for that perspective. I think we've heard so far that there is significant overlap between adult and pediatric SLE, although we've consistently heard that pediatric SLE is a more severe disease compared to the adult disease. And we've heard that in terms of management, that they're managed similarly. It would be nice for the panel to opine if therapy has been shown to have an effect in adult SLE, is it reasonable to assume that it would have a similar effect in pediatric patients? And if not, why not?
I guess I can get us started. Yeah, so I think that's the fundamental question, right? And I think the question that sort of came up earlier was if children have higher disease activity and more organ involvement, how can we assume that the same exposure level or the same dose level given to adults is going to be effective in children? And that's what we tried to answer in our publication, realizing that a lot of the data we have, is not clinical trial data. But I'd say very reassuringly, when we look at dose response, like in the lupus nephritis trial, patients who have lupus nephritis, almost by definition have high disease activity. And again, we saw a very, very similar sort of dose response relationship. And then, in the other exposure response studies, again, overall, there was a high degree of similarity. And so, again, in the mycophenolate study that I showed, most patients did have high disease activity, about 25 patients with SLEDAIs, in the 11 to 13 range. And so even though children may have, again, a little bit of higher disease severity, let's say 11 versus 13, more frequent organ involvement, I would say the preponderance of the data still suggests that children still respond similarly, at least at the drugs that we looked at, at the same exposure levels. But I would say that the data, again, is mostly limited to observational studies, but preponderance is highly reassuring. But I'd be curious what the panelists think.
Thank you. This is Simon. I would divide the molecular targets into categories based on our experience. The first category is verified or what we call confirmed targets, such as B lymphocyte stimulator, BLIS. So we have very solid evidence from belimumab, as you have heard today. I think that's a very easy yes if we have a next drug targeting BLIS and to use the extrapolation approach. The second category is what we call unconfirmed, and drugs what we call is new mechanism of action, not necessarily new, but like we don't have solid evidence to compare the efficacy, clinical efficacy between adult and children. Currently, we have been using placebo-controlled, randomized, controlled study for these targets, for these drugs. So what level of evidence do we need to support the extrapolation approach for these drugs? So I don't have a clear answer, and I'm all ears to the panel.
Thank you. Laura?
Yeah, thank you very much. This is a very interesting question that I think we can take from two different perspectives. If the question is to be understood like, can we find some pediatric patient that respond to treatment similarly than adult, despite some very well-documented differences between the disease profile in children and adult? My question is yes, it is possible that some of the pediatric studies still respond well. Why? Maybe because some of the dose in the adult was overshooted, so that we're still in the sort of effective sort of space, even if in principle in children we would always need a systematically higher concentration to reach those therapeutic response. We can take the question from another perspective. Can we ensure that always we will have a response in children similar to adults, despite the uncertainty that currently exists? Then I would say no. I can't. I mean, knowing the differences, knowing the uncertainty, it is from a regulatory perspective very difficult to assume that this might not be clinically relevant, and to assume that all the children will respond in a satisfactory manner. And this is where we need to be very cautious, and this is where I think this workshop is important, because my key message here would be, if we want to use a model-informed approach, if we want to use an extrapolation based on modeling and simulation, it is important to start collecting informative data in order to have high quality models, being that exposure response model, being that PK model, disease profiling model, so that we are in a better position to be able to say that for the drug XYZ, based on the mechanism of action, based on the biomarker data we have, we can conclude or not that the proposed dose is feasible, can be effective. So I think it is important that we really not jump steps to say that now we only have exposure response in adults. We have several uncertainty, imperfect data in different directions, pointing to something so we can generalize that we're in a safe place. I would be very, very cautious in any kind of generalization from where we are today. But I think that we know enough to collect now clearer and less discussable, questionable data in order to be in a position to answer that question confidently. Thank you.
I guess maybe to help us hone in on that a little bit, could maybe one of the panelists or any panelist weigh in on whether severe disease in adults is different than when severe disease in pediatric patients? Would you expect response to be different between the two? Because what we heard earlier is that adult studies are enrolling patients with moderate to severe disease. So if you could comment on that. Go ahead, Laura.
Based on all the data that's been presented here and just clinically experience, it seems to me that we can be pretty confident that the patients, that there is going to be efficacy. I think the only question is whether it's totally optimized. And so listening to Vikram, your presentation I thought was fascinating and was wondering if I'm allowed to ask a question of a panelist, was wondering if you know of any specific issues that might develop to make a modeling system less effective or less predictive. Does that make sense?
It's a great question. I've sort of worked with these kinds of models all my life, so I have an inherent bias to this that I must acknowledge. But I think with all of us who work in this space, we've grown to appreciate and become critical of these models with time. They've become very good. And again, I pass the problem in two parts. One is what is happening from a concentration perspective. There, the science is reasonably well advanced that after the age group of 2, and in this case, we're talking 5, for the classical drugs that we're interested in, small molecules, antibodies, our ability to predict concentration in children is very, very good. And I think we've seen that repeatedly. The FDA, of course, has reams of data on this. They do this on a routine basis. And we tend to fall in reasonably good spaces in terms of prediction. You made this point that I think this whole session has given us reasonable confidence that at least the first 2 criteria in the extrapolation framework seem to be met for SLE, for pediatric SLE. What we still don't know is, to your point, what's going to be the exact effect size in the child. And I think that's the unknown. And that's why I think we are doing these trials. So the models there are getting better. And I think if one could perhaps do an exercise, look at the drug that's been approved now and try and match the patients in adults and similar patients in pediatrics and try to compare the response that we are seeing for the same kind of disease severity. I'm sure there's a few cohorts of this which will overlap. We'll get some more degree of confidence that the effect size is what we think it's going to be with the same concentration. So the question, I think, is will we be able to predict with great confidence this is the effect size in the child? I don't think we're there quite yet. But the question, I think now, with the example that was shared, is that how much data do we need? And I think that, to me, is the central question that, augmented by models, statistical and pharmacological models is allowing us to get a reasonable distribution of the expected effect in children. And maybe this space is not demanding a pure single point measurement to say the efficacy is X, but we expect in a child with this disease severity to get this range of efficacy. And that may be, and I'm not a clinician, so I say this with a lot of deference, maybe the need of the hour for that patient as this evolves. Or we wait for 6, 8, 10 years, whatever the time horizon is, because every drug, if it's going to do the same thing, the next drug that's approved in child is another 6 years before our statistical colleagues would intervene post hoc and reduce the sample size. So the question is, why do that? Why don't you reduce the sample size right off the bat and do the testing that Dr. Ghosh said in a pre-specified manner and streamline the program?
Yeah, and I'll just add a couple additional comments. I think related to the original question, severe disease in adults and children, I think in general, just in my clinical experience, tend to be pretty similar. So when patients get admitted to the hospital or ICU, it tends to be infection in both groups. But from a lupus perspective, lupus nephritis, CNS disease in both adults and children, and then sometimes, things like pulmonary hemorrhage or very severe pericarditis are sort of the other things we see really across the age spectrum. And so I would say really severe hospitalization tends to be fairly similar. The way I sort of, I'll sort of throw in a plug maybe for one of our later sessions talking about trial design is, related to how much data we need, this is certainly a fantastic opportunity, right, for PK/PD studies, right? So our open label PK/PD, great time to confirm a target exposure and that dosing as well. And a well-conducted PK/PD study can really provide some great confirmatory evidence whenever there's any uncertainty, right? And so I guess where does the uncertainty come from? Well, I think it's drug specific, right? So with belimumab, we know that belimumab is typically dosed really at the height of that exposure response curve where it's very flat. So we have very, very high drug concentrations. And so we can feel pretty confident that we've sort of saturated that effect with belimumab, but that may not be the same with other drugs. And so I think adult PK/PD data essential to guide that pediatric PK/PD data as well.
Great point. Glenn?
There we go. Okay. Two questions. Maybe I'll ask Mckenna first. So, you've heard Dr. Balevic just talked about, the belimumab studies looked at I would say it's a very high dose, right? There was no difference in exposure between 1 milligram and 10 milligram, and we chose the 10 milligram, right? So that sort of assumes that it's kind of a high dose that we looked at, that we approved, that's for lupus. And I also heard that you say that, there were some unexpected side effects. Now, we can't presume that because the very high dose that if you were on belimumab, which I know you're not, but whatever drug we approve, if we don't have good exposure response or dose response information to sort of optimize the dose, we do run the risk of picking doses that are higher, that have more side effects, and maybe sometimes even preclude approval if the side effects are too bad. Just from your perspective, should we be shooting for optimal dosing on the pediatric side rather than just a dose that works?
I think that's a really challenging question because obviously you don't know the optimal dose. I think that ultimately that should be the end goal, but at a certain point also you want to make options available to pediatric patients. So if that means maybe starting with a higher dose to get that medication out to pediatric patients and then adjusting, I think that that's maybe a good option if that means that those patients could have access to that medication sooner. But I mean, the side effects, especially for me, are a big consideration and something I would consider when starting a new drug. So I don't know. I think it's a tricky question, but I tend to lean more on the side of maybe starting with a higher dose and then trying to optimize from there if it means getting patients the medication.
I just like to add, though, that even if we look at a population and come up with an optimal dose, it doesn't mean it's an optimal dose for a given person. And some of the side effects are appear to be idiosyncratic, right? So I would hate to hold up availability, of a drug for pediatrics on population dosing sometimes, right?
Yeah, and let me clarify that it wouldn't necessarily be to optimize the dose within a pediatric trial. I think Vikram's already described that we have a great way to model, but it does require, I think, on the front end, that we look maybe at optimizing dosing in the adult trials, right? So that you can see sort of where the exposure efficacy response lies and maybe target doses in children that would be, I'll use the term optimal. That may not, again, to your point, Laura, that may not prevent any of the or some of the side effects anyway. But that's what I'm sort of looking at, not to necessarily look for more doses in the pediatric study, but to maybe do a little bit more dose optimization in the adult studies.
May I just make two additional comments on that? One, at least this is a personal belief. I think we are doing just not in SLE, but generally across many indications where it's feasible, including oncology now with Project Optimus, which people are aware of. There is an increased focus on doing dose response studies. And as a consequence of that, we are getting more and more detailed concentration response analyses. And the response here, at least for me, includes both domains, the efficacy piece as well as the safety. So that is getting characterized. So ideally, if we have that in adults, we are able to triangulate on a window of concentrations that we want to target in children. And then also appreciate that when I give a dose in children, I too am getting a range of exposures. It is just not a single point because of the variability and other factors. So indirectly, one can look at exposure safety as well as other relationships in that patient cohort as well. And then that takes us down to then understanding and saying, well, I think we dose too high, we're getting too much safety. What are we going to do? The drug works. Are we going to repeat the study at a lower dose? Or can we practically think about some interpolation of that exposure response analyses to give some prescribing guidelines? This, I think, is a more difficult question to answer. I think everyone struggles with this. But in rare diseases, that's the path that will eventually have to hold in lieu of conducting large trials repeatedly or introducing arms or thinking about dose response arms to split the sample size to allow dose response to give us that. Because the challenge is if I could do 200 subjects, I would, but I can't. So how do I split the difference here? I think that's what is the question. And then how do we maximize information out of that study?
Armina?
I have another question for Dr. Sina. I was really intrigued by the modeling approach and how that could assist pediatric drug development. Now, based on what you heard this morning, on the similarities or the complete overlap of clinical features, of laboratory features, and also organ maturation at the age when children with lupus normally are diagnosed beyond the age of 5, what exactly would make you think that the extrapolation between pediatric and adult, that the response in a younger person is different than that in an older person that's considered adult?
Well, actually, nothing. Actually,
thank you.
But I'm not. I'm actually not a clinician. So I again, with deference to that element, but That has been very eloquently described. For me personally, the data is incredibly convincing, actually. And the one drug that's been approved, I mean, they can speak to this. I think their members, I think they did a really good job of honing in on this with this principal assumption that the response is similar. Because if they didn't believe the response is similar, they would be doing those response studies and splitting their sample size to worry about that, but they didn't. They went with a larger sample size, which then post-hoc got truncated. So the belief they went in to was the response is going to be similar, right? And they can speak for this. So simply nothing. I mean, I don't know how else to say it. Yeah.
Yeah, and I'll just, in 10 seconds or less, quickly add, my interpretation, our statistician can correct me if the belimumab data is correct. That tipping point analysis, I think, was super informative, right? The way I sort of interpret that weight is that if we sort of assume that adults have a 55% similar, or children have a 55% similar response to adults, then again, statistically, post hoc, we would say that the drug is effective. And so I guess one question is that sort of a correct interpretation? And if not, how can we sort of quantify the degree of similarity we need based on belimumab data?
I guess that's my turn. Yeah, so there's a number of different interpretations. So we can look at things like the effective sample size and roughly how much information is coming from adults with respect to pediatrics. And I don't recall off the top of my head what the effective sample size was associated with the 55%. It's in the paper that was cited by Dr. Roberto this morning. So roughly how much of the information is from pediatrics versus adults, and we can use that to kind of calibrate to what our level of trust is in the data. So yeah, there's different ways of doing that, and I think the exact statistical language will vary. So yeah, there's some complexity there, but that's roughly your interpretation is roughly roughly okay.
Yeah. So I guess with my rough approximation, where I was going with that is, again, it helps us to kind of understand how much similarity and dissimilarity, right, we can sort of can accept, right? And so that I just sort of put out there to maybe sort of help frame, sort of help quantify the degree of similarity and dissimilarity, at least for the belimumab study.
Nicola? It's at the bottom of the mic.
Yep.
No. Yeah.
So thanks. So first, I have a comment to follow up on Liniao's point, and I completely agree that it's critical that all the information that we can get from the adult program should be front-loaded, particularly to better understand the exposure response for safety, efficacy, PK, PD, and whatnot, to help us with these conversations and these discussions. This is kind of a, assumption for me. So assuming that this has been done and we have established exposures that are effective in adults, my question is for our EMA colleagues to delve a bit deeper into their concern. And my question is more to understand, is the concern from EMA that if you have established efficacious exposures in adults, and you have similarly situated adult and pediatric active disease, is the concern that pediatric patients would respond differently from adults, or is it just that patients with more severe pediatric disease may not respond as well? Because to me, that makes a difference of how we approach extrapolation. When we talk about extrapolation, we talk about extrapolating efficacy from adults to kids, assuming everything else is equal. And we all acknowledge that severe disease exists in adults and kids. And actually, adult trials have excluded many of the severe cases, particularly neuropsychiatric lupus. So we don't really know how that might work in severe adult patients. So I really want to better understand, is the concern that pediatric and adult patients with similarly active disease would respond, differently?
Laura, would you like to respond to Nikolai's question?
Yes, I can start. I see Roberto's hand is also up, but I can give it a start, and maybe Roberto might be complimenting. So I have already raised my hand even earlier to make this point, but just to answer the direct question that was asked here, my personal concern, and maybe not talking necessarily on behalf of EMA, but the concern that might be shared by some of my colleagues is exactly about your assumption, the very first statement that you assuming that they did everything else is equal. That's the issue, because the data we have now, and some of your statements even, I would say, support that, are not robust enough, because you said that we have no clue about the more severe patient in adult, the more severe phenotype in adult. They are excluded from trial. And we know that this phenotype is much more expanded in the pediatric population as compared to adult. So when we're performing the extrapolation, we need to have that consideration in mind. This brings me to my second point, that is related to model-informed approach. So exposure response characterization in particular. I need to say that I do appreciate, and I find this very interesting, that several speakers have insisted on the fact that data collection in adults need already to be designed in such a way that they are the most informative possible for the pediatric extrapolation setting. This is the way to go. And that I fully support. But my second point is that when comes exposure response analysis, I have heard several people talking about exposure response with different meaning. To me, for instance, when I have a box plot where exposure are sort of compared between two categories of patients, this is not my first guess of an exposure response analysis, to tell the truth. I think that now the science has evolved enough for us to have more robust exposure response analysis. And having this high quality modeling requires particular kind of data to feed that. And happily, now we have had recently some very good communication, publication work that are showing how we can upfront determine what are the kind of data we need to inform exposure response analysis in pediatric, not only in children, in adults. Assuming that you have some exposure response in adult, how can you collect data in pediatric to enrich that exposure response model so that it becomes informative for the pediatric population? And this was my first point, that there are still some uncertainty in that similarity that can be very broad in spectrum, that might be addressed with some well-performed exposure response analysis. But here I didn't have in mind some sort of descriptive matching of exposure in different responders categories, but really a more quantitative sort of exposure response analysis. And I would even myself even push also for more quantitative disease progression model. All in all, we would have robust modeling and simulation that can really put us in a good position when we want to use them for high impact, high risk application, which I think we've in for this particular disease. So these would be my 2 cents on that one, but maybe I leave it over to Roberto if he has more to say.
We actually have several people lined up to ask questions. So thank you so much for that perspective, Flora. So Len?
Yeah, this is a quick follow-on to that. So I was very impressed with the similarities. In fact, we've used it in many talks in the past about the similarities between the patient population, between the adult and pediatric belimumab, the endpoint that was used, the diagnostic classification, so similar. And then you see that the responses were so similar. So, that gave us a lot of confidence in a post hoc way to agree to a Bayesian approach. But the one thing that still bothers me a little bit is that when I look at the SLE-Dr diagnostic classifications and the comparison between adult and pediatric, like only, what was it, like 19% of the pediatric population had renal disease. And that seems different than what we think the general pediatric population is with renal disease, or am I missing that? So my question is, do we think the populations we're enrolling into lupus trials or into the belimumab trial represent the overall population of pediatric patients with lupus?
Well, the trial was not open to patients with nephritis and CNS disease and the other, I guess, subsets of lupus that we're so concerned about in pediatrics. So yes, but if you're talking about, I mean, this is about general lupus here, and I haven't, as part of the planning committee, I mean, we made the decision not to include those other groups. I actually think that the case for some of the subset comparisons and extrapolation is even stronger because pediatric patients without organ system disease at a current time, we know that a significant percentage of them are going to go on to get organ system disease, but a patient with lupus nephritis and an adult with lupus nephritis, I mean, they seem even more alike. Does that answer the question?
Yeah, it does. Thank you. And I have something that I'll wait for another session on study design idea. So.
Okay, thank you. Go ahead.
Yeah, I just want to bring in the perspective of a provider, and I've treated a lot of lupus patients over my career, pediatric and adult in New York City. We were the only lupus center for, many, many years. So I think the response, my personal experience is the response is very similar. There's not any physiologic change at age 18, that would make you, and we're mainly talking about, teenagers who are presenting with lupus. So I think we have to keep in mind all the other factors that might affect why children have more severe disease. They might be diagnosed after a longer period of time, latency period. They're often coming in with severe disease, even renal failure sometimes. Often these are patients who are in underserved communities, patients of color who might not have access to medical care. Also, there are very few pediatric rheumatologists. Most pediatricians have no training in diagnosing lupus. So we have to think about all the other factors that might make these patients have worse outcome. But my, personal experience is once you get them the medication and you get them into a good treatment program, they can do, they do equally well. They do respond to the medications. The other thing to keep in mind is we're talking about teenagers and young adults. And this particular, they're kind of individuating and developing. And there's a lot of non-compliance. I mean, I can't say that there's not non-compliance in adults, but I think in this teen, young adult population, we have to think about, those factors that make them do worse. And then think about what we're doing as their physicians. They're on impossible regimens. They're taking up to 10 medications at a time. And they're often, staying with, grandma part of the week, home part of the week. This is putting a lot of stress on their families. So they're not able to really keep up with these regimens. And that's partially our fault. we don't have anything better to do with them. So, I mean, we have to think about all the other factors. It's not just their disease physiology, it's everything around it.
And the way the drugs are delivered, right? 3 horse pills twice a day is pretty hard.
Right, you know, teenagers, if they're playing sports, you know, asking them to take medication 12 hours apart, you know, we're, you know, kind of putting it on them. But these are 14-year-old, 15-year-olds, you know, families, their family situation, even when it's great, you know, you're working with your teenager on so many issues at the same time. So I think we have to think about, you know, a little, you know, let's provide what we can. When they're 17 and a half, I don't want to have to say to them, you can't get obinutuzumab, you know, because, you know, it's only available if you're 18. There's not a reason. And the patients are doing badly. So I think we need to really think about how can we help and facilitate them doing better.
Thank you so much. We're at time, so we're going to go ahead and take a break. Thank you so much to our panelists for an excellent discussion. Thank you to our EMA colleagues who are joining us. We know it's in the evening there for you, so thank you for joining. And we will reconvene at 3 o'clock, so a 10-minute break.
June, and now they wanted to have a double-blinded parallel designs also in children with lupus. And as you already heard, there was an 8-year delay in getting the medication approved for children, meaning 8 years where the children had to expose to more steroids than otherwise if the medication had been approved. So I looked, so I do know that a delayed study start really makes recruitment more difficult. I did a Trinetics database search in anticipation of this workshop today. And before IV belimumab was approved in children, 191 patients in this Trinetics database at 33 different health organizations were already treated off-label. For subcutaneous belimumab, it was even larger. And now IV belimumab study is, IV anifrolumab study is still enrolling, and already 104 children with lupus get this medication off-label. Shown on the right side was an email I got from my clinical pharmacist last month, and she proudly told me that at the Children's Hospital in Cincinnati, you can now get subcutaneous of anifrolumab, and we can prescribe that. Mind you, that the study in children ex-US has not even started enrollment. Okay, here we go. So besides delays in enrollment, there's also that staggered enrollment that's used in pediatric studies, that you first study the older children and then the younger children. So the IV belimumab took a very long time to enroll. There was 16 months of screening for every patient who was ultimately enrolled. And although the study was done in 10 countries, only fewer than 2 patients per month were enrolled. So after the older age group was enrolled, there was a stop in the study enrollment for at least 3 to 4 months for the PK evaluation. So everything was shut down, and patients who were lined up to potentially be enrolled in the study, we had to send them away because we really didn't know how long that gap would be, or whether there would then be a study amendment that would even prolong the gap in enrollment. During that time, I need to do something with the clinical research coordinator, which are hired at my center. They need to be paid, but they are only paid by the study visit, so I need to reassign them. So that it really makes then the restart and the re-enrollment very hard. And then, you know, this stop in enrollment really delayed the enrollment of the younger patients even longer, and then actually prolonged the time to completion of the study. And all 13 patients in this younger cohort were enrolled ex-US. Now, we talked about that we could do adolescent-only studies, or including adolescents in adults. Give you 2 examples. The voclosporin we already talked about. I think the study was open for enrollment for 4 years, or almost 4 years. We only could enroll patients over a 5-year period. Imagine an adult lupus study where you could only enroll patients between the ages of 30 and 35. How many sites would you find? You found a couple of pediatric compassionate sites to try to enroll, but really they couldn't enroll patients because with the eligibility criteria in place, there was hardly any patient to find. Now, there's also some companies that started including children into adult studies, like for the ianalumab study. However, there is no pediatric site, and I don't think that a parent of a patient will go to a new provider to participate in a double-blind placebo-controlled clinical trial. So placebo exposure or continuation of standard of care really limits the interest of study participation. It is hard for us to explain why we should use placebo. We know about the disease similarities. Off-label, we always look to the adult evidence when we use medications in children. So for a provider, it's hard to say why we should there should be a placebo blinding here. And based on the number of patients to be enrolled, I don't think that we would see a safety signal that is different from what we know from adults based on the enrollment of hundreds and to thousands of patients. A favorable randomization scheme to limit the amount of placebo exposure really doesn't help with enrollment. For the parents who think about the child to be enrolled, it's their child who has a risk for placebo. And they don't care whether the 4 other kids did not get placebo. If their kid gets placebo, it really doesn't matter. And also, if you do that stratified or unequal randomization, there is the potential that you actually have to increase the overall sample size to show some measure of effectiveness. The risk of receiving placebo despite active lupus makes also providers hesitant to consider their patients for trial enrollment. If my patient is doing already poorly on the standard of care, and now they are flaring and they may be eligible for the study, now I have to enroll them in a study where they get placebo from the get-go and where I really cannot escalate therapy without making them eligible for study continuation. It really makes it difficult. Then there are these very highly restrictive eligibility criteria. They're mostly taken over from adults. Now, I'll give you an example. So I have a child with lupus who needs to have active lupus. It's 6 initially for the belimumab study. The SLE threshold was 8. They cannot have been on a high dose of steroids of more than 0.5 milligrams per kilo during the screening period. And the month before the screening period, they can also not have been ill because otherwise they would have gotten higher doses of steroids. Then 3 months before they are considering participation, they cannot be having any changes in immunosuppressive medication or even antimalarials. So finding these patients is exceedingly difficult. And then there are these long washout periods, even before the screening can start. And here I really have ethical problems because in essence, I have to eyeball a patient for potential study participation without them even having signed a consent form that they would be interested in the study. Because with a consent form, the screening period would start. So during these long washout periods, flares are real. And patients are, ending up in the hospital, and those flares then require the use of medication that are prohibited in the study, in the eligibility criteria, based on the study eligibility criteria, and then the patients drop out of the study. And many of the washout periods can only be addressed if we have new onset patients, because then they haven't gotten a biologic yet, so they don't have to wash out for 6 months or 3 months, whatever. But then there is the limitation of a minimum study period or a minimum duration of standard of care. If one of our patients is active lupus for 3 months, we will start an immunosuppressive, we will start a biologic to make them not have damage. And that again makes it very hard to recruit to study. Now we talked about placebo. Now the open label, the subcutaneous belimumab study was open label. For the 25 patients, they still needed like 2.1 years to kind of recruit. And I would like to point out that none of the patients in the IV study was allowed to roll over. Why? I couldn't find a scientific reason for that because if the focus is PK, shouldn't they be able to roll over? And that actually would give the clinician and the medical community valid or important information how to move patients from IV to sub-Q preparation, but that wasn't allowed. And so 25 patients were enrolled over 2.1 years. And if you look at the distribution of those patients in terms of their racial background, the vast majority of them were white. And as we know, at least in the US and in many countries, many of the lupus patients are Hispanic or of Asian origin or African American, given their increased risk. So why is that only so few children with lupus with, Black or Hispanic backgrounds enrolled? My suspicion is it's that there are marked socioeconomic barriers to study participation. I'll give you an example from one of the clinical trials I'm doing right now, funded by NIAMS. So we looked at patients who were eligible for the study, and 26% turned us down because the parents couldn't come up with scheduled visits, which were actually aligned with standard of care. They said they couldn't adhere to all the labs and to the procedures. One of the concerns was long traveling to the study sites and the length of the study visits. And all of that made it really hard for them to participate. So when I sum up this, there's loss of income. When a child with lupus participates in the study, the entire family participates. If you're on an hourly wage, during that time when the family is at the medical center, there is no income. They just have expenses. Many of this family have multiple jobs. The kids are missing school. There's often a language barrier. There are long travel sites, even in Ohio, where there is an abundance of pediatric rheumatologists. On average, our patients come from 2.5 hours away. So if you think about that, it really makes a big difference. And then the kids are severely impacted by the disease. For them, coming to a study visit and sit in the car for a couple of hours, then spend the medical center for 4 to 6 hours for study procedure, and then drive back on the same day is really hard. And we cannot forget that these families have other children, and they need to be taken care of too. They cannot come with the study patient to the center and sit there the entire day. And then there is the issue of the central laboratory. I realize that it makes it maybe easier to analyze safety signal, but it's really a bane in the existence when you try to recruit into a study. I'll give you an example. So a patient with lupus is very ill. They come for a screening visit. The patient is very ill, yet they get the results at least a week later. So during that time, I really don't know how poorly the patient is doing. And if I almost have to get duplicate shadow labs, because if I'm concerned about the patients, I need to know what's going on. But then very often the labs need to be redrawn because they get clotted. I've never seen as much clotting in any lab than in the central labs. And then the labs need to be redrawn. If a lab needs to be redrawn, that means the patients drive again to the study site. They need to be redrawn again. We wait another 7 to 10 days. Because the clotting and the failure to get the results is especially difficult when the weekend is closer, we only can do study visit Monday, Tuesday, and Wednesday. If we do a quantiferin test, it needs to be cooking in the center for 24 hours. And for sure, nobody want to have a central lab to arrive, at the site on a Friday because it may not be accepted. If FedEx is behind, redraw is in your future. So there's also one other issue. Very often when children are starting to participate in a study, the long-term access to medication is uncertain. It's not stated in the consent that they will have long-term access to it. And, theoretically, the site could do individual INDs, but that's really hard to maintain by a clinical site. And if I tell my patients, you can't be in a study for 2 years, but after 2 years, we don't know whether you have access to it, that is another stumbling block, another barrier for families to participate in the study. So here are my key messages. Delay in starting medications in children with lupus make the recruitment much more difficult. Duplication of the eligibility criteria from adults and adults designs like the parallel double-blinded placebo-controlled studies really are difficult in children. We have more multi-organ disease, and therefore there's a higher risk of flares in organ areas that are not in lead to the discontinuation from the study, and the washout periods are just very difficult for a pediatric population. Placebo exposure is a major problem, and central lab use adds to the study burden and complexities. And last but not least, the entire family participates in a clinical trial, and that needs to be considered when a study is planned and when reimbursement for study procedures are being negotiated with the sites. Thank you.
Thank you, Hermina. Our next speaker is Sul Yavuz. She's the Global Clinical Program Lead at AstraZeneca, and she's going to provide the industry perspective from lessons learned from conducting clinical trials.
Hi, everyone. Thank you so much. It's great to be here today, and thanks for the opportunity to speak with you all. So I would like to start with, you guys heard all these delays, but if you can see visually, it's really interesting to see not only for lupus, all pediatric drug developments lag substantially behind adult approvals. So it's unfortunately, SLE is not an exception. So we heard all that Benista is approved back in 2011, but pediatric approval came after 8 years later. And anifrolumab approval occurred in 2021, and our study is still ongoing. So let me walk you through about our pediatric SLE study Blossom. This is a phase 3 study. It's a 3-to-1 randomized placebo-controlled trial, and enrolling approximately 100 patients aged 5 to 17 with moderate to severe disease activity. It's also a weight-based multi-cohort design, and participants must meet 2019 EULAR and ACR classification criteria and the disease activity requirements. As you can see, this is for part of study. Part A is the extensive PK study for the dose determination. As Hermina mentioned, it gives us at least 4 to 6 months delay to enroll and determine the dose and get approval from all the EMA partners, EU CTR, and roll over the patients to when the dose determined. So this is one of the challenges we face. So when we look at the overall challenges, so what makes pediatric SLE study is so challenging? I am an adult rheumatologist, and I was an investigator in my former life. So I witnessed several failures in SLE development, right? So, and SLE is, as you know, a very heterogeneous disease, and even some consider that it's not that one disease, several diseases. So pediatric is not that different. However, I think from industry perspective, one of the major challenges we are facing is the differing perspective from regulatory and key agencies. This significantly delays the study start. This is one of the obstacles we are facing. The other is we all know that pediatric SLE population is extremely small. And as Hermina and the others mentioned, placebo-controlled trials, even though those are gold standards, but and we all know that placebo is not just placebo, is on top of add-on therapy and on top of standard of care. But still, even though it's a 25%, 1/4 of the child, think about, if it could be allocated to placebo arm. So those, decisions are not quite easy to make, particularly if your drug is available for adults. So those are, the challenges. And the other one is the eligibility criteria. Yes, those are scientifically, understandable. However, we have to also, simulate our adult studies, adult trials. That's why we need to also keep eligibility criteria similar to adult studies. But it also, shrinking the eligible patient populations. So I will come back to those, issues again, but I would like to start with the most important challenge, perhaps. So our regulatory journey started in 2013. As you heard, that EMA, particularly based on the guidelines, wants us to, approach them after phase 1 study. So initial proposal from the company was an open label PK safety study and extrapolation for efficacy. It was initially agreed conditionally, contingent upon the adult study results and positive exposure response relationship. However, the other agency didn't agree with that approach and strategy and immediately rejected. So I don't want to go through all those steps, but 2021, once we approved for IV anifrolumab, and we changed the design significantly, and based on those all changes and modifications, 2023, we received approval from both key agencies. So basically, what I would like to say here is that I think the key takeaway from this developing a globally aligned study plan requires, at least for us, multiple rounds of scientific discussion and several iterations of the protocol to address different perspectives and regulatory requirements. So I would like to touch on briefly to RCT versus randomized withdrawal trial designs, because also, it was discussed heavily during this regulatory agreement process. I mean, both have pros and cons, but basically RCTs, we know that those are gold standard, and we discuss, I mean, if you don't use at least partial extrapolation, it really requires large sample size, and long trial duration particularly is quite challenging. However, on the other hand, at least 50% of the investigators I spoke with until now, they are not also quite happy with the randomized withdrawal trial, even though it looks like, you are treating patients ahead of time, and so for uncontrolled disease not to expose patients with uncontrolled disease not to expose to placebo. So as you can see, balancing scientific rigor, ethical considerations, and also feasibility is quite challenging. So overall, if I can summarize and the lessons learned from our experience here, those, different opinions, lack of harmonization across regulatory agencies results in most of the time for sponsors with multiple protocol amendments, and particularly even some countries don't accept, placebo-controlled trials. It also, another hurdle for us. So the point is early scientific alignment between key regulatory agencies in conjunction with clear and harmonized guidance can really help facilitate global pediatric drug development and minimize the delays associated with divergent regulatory expectations. So when it comes to population size, it's also, quite clear from all the talks we heard today. So the population is extremely slim. So even though besides the slim, when we look at the clinical phenotype, again, we all heard that phenotypic spectrum similar with adults. So even though CNS seems, for example, slightly higher in pediatric, if you look at the recent meta-analysis and if you compare it as except seizure and chorea, I mean, all other 19, at least 2019, based on 2019 criteria, all 19 subsets are quite similar in adults and pediatrics. And also, when we talk about the disease activity or higher disease activity, so when I look at to our adult studies, at least 45 to 50% of patients also had at least 10 and above SLEDAI score, which also indicates, a higher disease activity. So we know that the difference, at least at the onset, at the diagnosis, for several reasons, pediatric patients might have a higher disease activity. However, overall, when you look at the clinical phenotype, those are quite similar for every sense. So there's one other interesting study, actually. This is a survey, and it shows that it's a global survey, included 40 countries. If you look at the results, 70% of pediatric rheumatologists preferred open-label PK/PD studies over RCTs, both because of ethical concerns or concerns from the families. And we also know that frequent flares requiring rescue treatment, either resulting in SF or early discontinuation from trial. Even though you're designing your study as a randomized controlled trial, at the end of the day, when you finish the study, you may have a very small sample size to really have concrete results. So I would like to summarize what I wanted to say today is, again, we all discussed today, pediatric and adult SLE share the same underlying pathophysiology and similar clinical phenotype. Although disease in childhood SLE presents with a higher disease activity, and we all should be aware of those patients experience more frequent flares and accumulate them much faster. But disease classification, disease activity assessments, and all treatment guidelines are the same. So I would like to conclude my talk by saying that we shouldn't consider those pediatric patients as just small adults, as we all discuss all time. Pediatric SLE drug development must be tailored to realities of the rare patient population, ethical constraints, and parent expectations. But on the other hand, we have to deliver, of course, benefit, risk, and safety for kids. Thank you so much.
Thank you so much, Sul. We're going to pivot a bit and transition to the patient family panel. So I'm going to ask our patient family representatives, as well as our moderator, Stacy Ardoin, to join us.
Well, good afternoon. This is really exciting to be moderating this session, and I'm so grateful to Lily and the planning team for how thoughtfully they've incorporated the patient and caregiver voice into both the planning of this workshop, as well as these opportunities to hear from our patients and caregivers directly. So really, this panel is where we exclusively want to hear from our patients and caregivers about what they've heard in the discussions today and based on their lived experience, so we can learn from them. I know we've heard from some of our panelists previously today, but I just would like to take a moment to level set and have each person just provide a brief description of your name and where you are and what you're doing in life before we get started. So Mckenna, would you like to start at the far end?
Sure. Hi, everyone. Mckenna Bowes. I am originally from Pittsburgh, but currently in West Virginia for medical school. Yeah, I'm very excited to be a part of this panel as well. Thank you, Dr. Ardoin.
Hi, my name's Chloe Raymond. I'm from Boston, Massachusetts. At age 11, I was hospitalized for ITP, which is when they discovered my lupus antibodies. I wasn't diagnosed for many years later. I've enrolled myself in research by donating biosamples in hopes to lead to new treatments. In addition to working with CARA, I'm also a member of the IPAC. It's the Implementation Partners Advisory Committee. We are working on the About Time project, which looks at how to implement healthy heart strategies for children with lupus.
Hi, everyone. My name is Elizabeth Santa Cruz from Kearney. I'm a caregiver to my daughter Mia. She was diagnosed with lupus when she was 9. Thank you.
Hi, everyone. My name is Mia from Kearney, New Jersey. I'm an NJIT student, and I was diagnosed with lupus at the age of 9. And thank you, FDA, Cara, Lada, for inviting me to participate to represent the lupus patient.
Hello, everyone. My name is Nia Cooper. I'm 20 years old, and I'm from Catonsville, Maryland. I'm honored that this is my first opportunity to participate in lupus research and advocacy. I'm currently going to my junior year at college at Towson University, majoring in healthcare management, and I was diagnosed with lupus in the summer of 2022.
Great. Thank you for those introductions and all for being here. So we had created some questions of our own to go over, but we're going to start with the question that's on the screen, which is, hearing about what we've talked about this morning and how the treatments work both in adults and children. Just generally, what would give you more confidence that the treatments you're receiving are correct for you? And I would invite anyone who would like to make a comment to go ahead. So this ties in a little bit. I'm going to kind of switch because we had kind of two sets of questions. This ties into some of the questions we had asked before. So really this ties into, what makes you feel comfortable taking or having your child take a medication that's approved by these trials. So I think when we talked about this before, Chloe and Mckenna, you had some thoughts about this. It's our last question.
I guess I'll start. I know that there's some uncertainty with extrapolation, but I believe it's a beneficial approach in finding new medications for people with lupus. I think that in the right circumstances, extrapolation is a good strategy as it opens new options for medications. I also wanted to point out, we were talking about steroid use earlier. I feel like I'm very fortunate to have a great rheumatologist who listens to all my needs, and having that has been really good for me. But I feel like a lot of young people are forced upon like the steroid route because they don't have many other options. So I think in this research, it will just open up many more options for those people to know that, because we just know so much that's bad about steroids. So finding medications that will not only contain flares but not have as many negative side effects is beneficial for us.
Thanks, Chloe.
Personally, as a parent and caregiver, I support using adult data when there's a strong scientific justification. If existing evidence can safely reduce unnecessary delays and duplicate studies, it should be used to bring promising therapies to our children sooner.
Anyone else? So I'll move on to one of our next questions, which is a little bit more general about participating in clinical trials. And we'd like to know what factors are the most important to you when you're deciding whether or not you want to participate in a clinical trial, when you think about your perspective as a patient or caregiver of a child with lupus.
I can start off. I think for me personally, a big factor that would influence my decision to enroll in a clinical trial would be the time commitment. I have a very busy schedule like most other lupus patients and families. And I've done a lot of reviewing grants and proposed studies and providing feedback on different designs. And a common factor that comes up is studies want patients to come in weekly or biweekly. And unfortunately, that's not always feasible. So I think that that's a really important consideration when you're recruiting patients for trials. It's a big factor for me personally.
I know for me, for my lupus journey, I started out on the holistic route. And I feel like when it comes to participating in clinical trials, full transparency is very important to my family and I. And then just considering the potential side effects, both long-term and short-term. And similar to how Mckenna said, just how participating in the clinical trials would impact my school or academic life as well as my social life.
Again, as a parent and caregiver, flexible scheduling is very important to us. Can we combine telehealth visits with in-person visits? I heard in the before that appointments are usually between Monday through Wednesday, but that's something not realistic for us, especially for the children. We don't want our children to miss school. As it is, they're going through a lot. They're going through a lot. So can the appointments be in the evenings? Can the appointments change to be on the weekends? We don't want to suffer any loss of income. What about blood work? Instead of traveling 2-3 hours for blood work and then do it again 10 days later, can someone come to our house? Can the lab work be done locally?
Great, thank you. And we've talked a little bit during this session about trial design, some of which require patients with lupus to take a placebo medicine on top of their already standard of care medication. And we've heard from the academicians and the industry partners about this approach. I'd love to hear from the patient panel your thoughts about how comfortable you are in receiving a placebo medication on top of your usual standard of care medication.
I think something that would really concern me about enrolling in a clinical trial would be the possibility of a flare. I would be disappointed if I was enrolled in a clinical trial and received the placebo. And I know it was one of the questions was, into addition to usual medications, you would be enrolled in a trial. But usually when you go into a clinical trial, it's because you're doing your worst and you're trying to find alternative routes for yourself. So I would just feel really discouraged if I gave all this time and effort and then just made my condition worse.
I understand why placebo-controlled trials are sometimes necessary to answer important scientific questions. However, I will only feel comfortable knowing there are rescue options and close monitoring, and it will make me feel more willing to participate.
Great, thank you very much. Well, you guys kind of tackled our last question very effectively at the beginning, talking about extrapolation. I just wanted to open the floor to you to comment on, any other comments you'd like to make based on what you've heard in the preceding presentations this morning. Anything else that comes to mind that you'd like to share based on your personal experience?
I guess I'll go. As I said before, I was diagnosed later in life. And when I first was diagnosed, grappling all the medications was very much a challenge. And a lot of them did not work. So I just feel like this is so important for young kids to have these options and these different avenues, and just, making sure that they feel comfortable and they feel like the end goal is attainable for them.
Thank you. Any other comments?
I just want to say that we need to make sure the clinical trials are accessible and realistic to the families.
And I'll just briefly add, I know I had mentioned this a little bit earlier and it's come up as well, but I think it's not only just about developing new treatments. As mentioned previously, some of the most frustrating and unexpected aspects for me has been like dealing with insurance and actually accessing these medications. Recently, I've restarted my IVIG, and insurance only improves home infusion. So I have to deal with picking up the medications, scheduling with insurance, scheduling with a nurse. Similarly with my rituximab, they approve different biosimilars every time I get the infusion, and I react differently to each one. So it's a little bit frustrating to deal with that. So I think that when we're talking about new treatments for pediatric patients, we have to look at the whole picture. And that includes after the medication is approved, how can these patients actually get those medications?
Wonderful.
Stacy, can I
Yes,
ask a question?
please.
I just wanted to ask a follow-up question to Chloe, because she made a really interesting point about placebo trials. And everyone keeps saying, oh, but you're on standard of care. if you're on standard of care and doing well, would you enroll in a placebo-controlled trial if you're already doing well?
No, I wouldn't.
I don't know if anybody else would be willing. If you're already doing well, why would you enroll?
Yeah, I just feel like the risk of a flare if I'm doing well is not worth it to me.
Do we have any other questions from the audience for our panelists?
I do have a question, actually. Maybe Chloe can expand on this and others can as well. In terms of your tolerance to placebo exposure, would your perception be different if the product has not been approved in adults yet, in the sense that we don't know if it's going to be effective or safe, and we're trying to figure that out? And the study has enrollment open to pediatric patients, but there's potential exposure to placebo.
I mean, I had mentioned before that I am all about increasing options for children just because we are so limited with only one that's approved. So I would participate in a trial with a risk for a placebo in that situation. But if it was, for instance, what Vincent had said, if I was already doing well, I wouldn't. I would go out of it.
Thank you.
Amina?
Maybe a little bit of a self-serving question. So if you have active lupus, and obviously standard of care didn't work because otherwise you wouldn't be flaring. If you were offered a trial where you get for sure active drug right away, let's say for a couple of months, and if you didn't respond, you would drop out. But after, let's say, 6 months or 8 months, you were asked to be randomized to either get no drug anymore or continue the drug. And if you don't get drug anymore, you have a risk of a probably minor to moderate flare. Would that be more acceptable than getting placebo right away?
I would take a chance in that situation because if you're doing that unwell, I just feel like you're willing to grasp any opportunity that you do get.
This might be a provocative question. I don't know how aware you guys are of cell therapy, but the idea of cell therapy is that you would get a one and done treatment that could potentially leave you with no disease activity and no need for medication. And we don't know what the duration of that would be, but what are your thoughts about A, how attractive that is? B, how long a duration would you need before you were willing to take the risk? And by the way, you'd have to be in the hospital for a week or maybe 2, depending on where you live, when you get the treatment. So how does that strike you as an option?
Can I ask a follow-up question? Just a clarifying question are you referring to like CAR T therapy or something similar?
Okay.
Personally, I mean, the idea of not being on a medication and essentially being cured, I guess, for an unknown amount of time is very appealing. I think that for me personally, I'm very well managed now, so I would kind of have a hard time transitioning to that. I think that my disease would have to be in a worse state to consider that because right now everything is going very well. I think the hospitalization, the week or 2 required to receive that treatment would be worth it in my mind because if that solves your problems, essentially there's no hospitalization in the foreseeable future. So I think that that's a very small price to pay in a way for a treatment like that. I do think that I would also, in addition to being in maybe a worse or more severe disease state, I would like to see some more data because I think that that's a pretty recent development over the past couple of years. So,
Yeah, it's definitely an emerging field. How do the rest of you feel about it? I mean, it's kind of what would it take for this to be attractive to you?
I mean, if my daughter wasn't doing well, spending 2 weeks in the hospital, that will mean nothing because as it is at the beginning, I spent like more than 30 days between appointments. So that wouldn't be an issue. And if there's an option that my daughter might be doing great afterwards, we will do it right away, especially if the insurance covers it, because that will be another topic.
Well, right now it's in trials. So the
Exactly,
trial will
yes.
cover it, but
Yes, but we will definitely do it.
All right.
Other thoughts? I guess the corollary question is, how sick do you think somebody should be before it would be worth doing that?
If it is affecting your quality of life, I mean, if you're not able to function, I have seen my daughter going through joint pain. My husband carrying my daughter because she's not able to walk. I have seen her, if when you ask her what's 2 + 2, not being able to answer. Being affected at school, her grades decreasing, and between protein in the urine, all mouth ulcers, a rash, and everything else that involves lupus. Like it's because it affects a child in so many different ways. I think a parent will be more open and willing to try it.
Yeah, and as we know, like the cellular therapies are really exciting, but not something we're focusing on in this workshop today. But I do really want to thank all of our panelists for your bravery and your smarts and confidence for sharing us with your experience. Thank you so much for being here.
Thank you so much, Stacy, and our wonderful panelists. Thank you so much. Our next speaker is Rebecca again, and she's going to talk about extrapolation of efficacy from adults. Thanks, Rebecca.
All right, thank you. So we have talked a lot about extrapolation and highlighted some wonderful opportunities and strengths of extrapolation. I did want to make sure that we touched on the concept of limitations in extrapolation. Where are the gaps? Where do we need to be potentially a little extra cautious and thoughtful? It's been said before, and I think it resonates with everybody in the room. Children aren't just little adults. It's not an adult that's been shrunk down to a smaller size. There's more to it. And I think that that begs the question, well, what is actually different about children? And I'm going to try to answer that at a bunch of different levels, and some of them are going to be pretty basic and simplistic, and then some that hopefully we will, as a field, continue to think more and more about. So for starters, there's differences in drug metabolism, which surprises nobody in this room. Probably not between a 17-year-old classified as a child and an 18-year-old classified as an adult, right? The biology does not seem to respect our legal definition of adulthood, but certainly the younger a child is, the more there is need to do PK studies and how much can be modeled or done in trials. That is an important thing to establish. There's also differences in how children are able to take medications. This has been touched on a little bit, but if you want to do a trial with young children, they often cannot swallow pills of any kind or size. And even children who are old enough to swallow, maybe not large pills, maybe not whatever single denomination pill is made. May, if it's a subcutaneous medication, need a smaller dose than exists in a pen. So not only in terms of clinical treatment, but also in terms of participation in trials, there's a need for the dose to be available in a way that is appropriate for the age and size of the patient. And that may mean that there needs to be a liquid formulation or a medication that can be crushed, as opposed to one that has a coating that cannot be crushed. So that is an inherent challenge. And how do you make sure that there's the appropriate sizes in subcutaneous pens, et cetera? Obviously can't extrapolate that. I think another key point, and this was touched on, is the concept of adherence. And adherence rates have been shown again and again to be different in children and adolescents than in adults. And this is particularly important when we're talking about a medication that is taken orally or home administration of a subcutaneous medication. So adherence rates are suboptimal across the board in lupus. We know it's a major challenge in lupus, but the non-adherence rate is double in children and adolescents through multiple studies compared to adults. So we talk about how the trials are often not powered to really assess efficacy. And if the result were to be, it looks less effective in the children, it's at least as likely that the reason is that a lower percentage of the children are actually taking the medication, getting therapeutic levels. So even if we find the finding that we were worried about, that was the impetus really for doing the study, I don't know that it indicates that there's actually a biological difference or a difference in response if comparable concentrations were able to be achieved in children. So that's another important, not extrapolatable instance. Children, of course, are still receiving vaccines throughout their childhood, which means their immunologic repertoire and response is different, and B, if they're put on medications that might interfere with the immune system, their ability to respond to those medications, right? The immunogenicity of vaccines. So just wanted to put up in terms of our routine vaccines immunization schedule for children. The good news is by the age of 5, patients have received approximately 85% of the vaccines that they will throughout their childhood. So it is an important thing for us to keep in mind for very young children, but 5 and up, fortunately, not as problematic. So reassuring age 5+ is how I would talk about in terms of the population for which we can safely extrapolate. Okay, digging into a few more things, monogenic lupus, we talked about how that's not the thrust of most of this workshop, and I wanted to just highlight why. So here is a list of some of the genes known to cause monogenic lupus, and you can see that they're kind of categorized into the types of problems that they cause. I talked to you earlier about SAVI, which was STING1, which is in the green box as a type 1 interferonopathy. And I wanted to also introduce in the yellowish box, C4 deficiency, one of the complement deficiencies, kind of as a concept of extrapolation across biologies. So the picture on the left is the one you already saw of a young girl with SAVI, and that is an interferonopathy. And on the right is a young girl with a familial monogenic C4 deficiency, and that is a monogenic lupus known for having normal interferon levels. So with SAVI, I mentioned already how there are some case reports of anifrolumab being used and JAK inhibitors for a longer period of time, all targeting the interferon signature very effectively and treating SAVI patients. And I'm probably never going to be able to report to you any trials of that being used in C4 deficiency because nobody would knowingly use that medication for this particular physiology. And so the idea of a medication that works at a population level becomes a little bit less applicable in monogenic lupus. But when we are talking about non-monogenic lupus is much more extrapolatable. I did want to just mention, because it was a fantastic case report that came up earlier this month in the New England Journal of Medicine. It talked about a patient who had a known DNase 1L3 deficiency, not producing any of this important enzyme, which was the trigger for his lupus. He was diagnosed at 18 months of age, and in his teens had never achieved adequate control of his lupus, despite all the other medications used for him. Through compassionate use, he was given recombinant DNase 1L3, and according to the case report, in a matter of hours, they saw improvement in his cutaneous manifestations and over days to weeks in all of his manifestations. So when there is monogenic lupus, matching one-to-one with the therapy makes sense. And then when there's multiple pathways to a more generic polygenic form, then our other medications that work in adult lupus are much more extrapolatable to children. So reassuring again for age 5 and up, whereby if you have not developed lupus by that age, it is unlikely to be monogenic. Okay, I also think it is really important that we talk in depth about the concept of ongoing growth and development, and sort of the safety concept that relies because children, again, are not little adults. And this is going to be covered in the safety talk tomorrow as well. Dr. Sun has some great information to give about this, but I wanted to just introduce the topic a little bit here. So because there's ongoing growth and development, there's opportunity for some differences in efficacy. As somebody who cares for both children and adults and specializes in lupus, I am glad to say that in non-monogenic lupus, none of us have seen differences in the response. So it was touched on a little bit earlier about the concept of severe. If I had to think of the 10 most severe cases of lupus I've ever seen, about 5 of them were adults and 5 of them were children. And you can definitely say the patients who are so severe that they die or might die Might they not respond as well to a new medicine? Perhaps, but that's true for children or for adults. Your typical moderate to severe spectrum so far seems to be responding comparable to the various medications. But we also need to think about that safety signal and what impact might the medication itself have on growth and or development. So I wanted to touch on that. And I think of a bunch of different dimensions in terms of how medications might interact there. Everything from linear growth or a child's ultimate height if they're receiving a medication before they've reached their full height, to development of their metabolic state, bone development, puberty, brain development, immune system, etc. I think a really good case for understanding this, actually, unfortunately, is corticosteroids, sort of the mainstay of our therapy right now, but it shows just how detrimental a medication can be on all of those domains. So absolutely impacts a child's ultimate height, causes dyslipidemia and weight gain. When given to a child whose bones are still developing, it has a much higher rate of avascular necrosis, so the bone collapsing on itself, leading to deformities, chronic pain throughout life, etc. Brain development. The brain is still developing, and we know that it can therefore impact white matter integrity. It can also increase rates of problems with memory, mood, attention, etc. And steroids have huge impacts on the immune system, although that's the one place I will say that fortunately, when the steroid is stopped, the immune system has a tendency to return to its prior state. As we think about newer medications and their impacts on these different domains, I'll highlight JAK inhibitors as one. So there was definitely concern early on that there might be an impact of JAK inhibitors on linear growth, and fortunately that did not pan out, although we do see some dyslipidemia and weight gain for children and adults who are put on JAK inhibitors. The other case in point, unfortunately, is cyclophosphamide. So what happens right now when a child or adolescent has very severe lupus, right? The sort that might destroy an organ or result in death. We really have two main things that we can use to hit that disease hard. It's high-dose corticosteroids on the left, and it's chemotherapy, cyclophosphamide listed here. And that unfortunately does have a lot of the detrimental side effects that we worry about. So cyclophosphamide can cause primary or secondary ovarian failure. It can cause permanent infertility in men. And it has long-standing impacts on the immune system. So long past when we stop treating the patient, long past when the medication is out of the immune system, it can be lifelong detriments that we see in some of our adults from cyclophosphamide that they received when they were 10 years old. So that's impacts on growth and development and safety, and super, super important. But one thing I did want to highlight, and Dr. Sun will go more in depth about this tomorrow, is unfortunately these very safety signals that we want to pay attention to are things that are not immediately evident. They are things that you are unlikely to discover during the course of a trial. So if you give a 9-year-old a medication, a 9-year-old boy, and you want to know if it affects their ultimate adult height, we're not going to learn that in the period of a trial, and the same is true for many of these safety signals. So they're very important, but our traditional approaches probably aren't going to reveal the answer we're looking for. So we need to know, where do we need to do additional deep investigations, particularly if there's some preclinical animal studies that indicate concern for impact on growth and development in an acute sense. Infection is something that we can study in a trial. And then we probably need to find a way as a community to really lean into post-marketing safety studies for these patients, because we won't know for 5, 10, 15 years, malignancy even further off, the impacts that these medications really have on children. Okay, just a couple of summary take-home points I wanted to highlight. So one is, caution is very much appropriate when extrapolating from adult studies to children under the age of 5 for all of the ways we just discussed. For children over 5, really what we need is PK and dosing studies, for sure. Attention to growth and development perturbation signals. So anything that mechanistically we're worried about from preclinical animal studies, for example, and could warrant further studies if it's something that we think would be revealed during a trial period. Emphasis on post-marketing safety studies, particularly leaning into growth and development. And then every opportunity possible to get children with severe lupus off of the medications that we already know to be highly toxic, like corticosteroids and cyclophosphamide. And in this sense, I think it's important to think sort of about our paradigm when we're considering safety. So, you know, what am I taught in medical school about a safety study in a trial? And it's new medication A versus placebo. But for a child with lupus, I don't actually think that that's really the comparison that we're needing to look at, or that's really the safety question that should be front of mind. What happens with a child with lupus is we're comparing new medicine A to organ damage or death from active lupus and to the use of high-dose corticosteroids and chemotherapy with known toxicities. And so how can we think about safety in that perspective? And with that, I'll say thank you.
Thank you so much, Rebecca. That was excellent. So we're going to go ahead and transition to the panel discussion. And I'm going to ask Laura Schamberg to come up to moderate this session. We'll just ask the panelists who have not already introduced themselves to introduce themselves, and then we'll go right to the questions.
So if folks can introduce themselves, maybe start at we've been starting at the other end, particularly folks who haven't presented or introduced themselves yet.
So I'm going to introduce myself because I think Sule has spoken previously, so I'll start. My name is Raj Nair. I'm the Division Director for Rheumatology and Transplant Medicine. I've been working with the FDA since 2013 within the rheumatology division through reorganizations. I am an adult rheumatologist, but I am supported by a bunch of pediatric rheumatologists who educate me, so I feel like I do have a lot of great minds helping me, and so that's why I'm up here.
Hi, I'm Jordan Roberts. I'm a pediatric rheumatologist and an associate medical director at AstraZeneca in immunology clinical development based in Boston, working on some of our pediatric lupus studies.
So evidently we have a question that's been sent in for the
sorry.
patient representative, and I
Sorry,
don't have.
can you hear me?
Oh, sorry. I'm sorry. Caroline,
Yes.
can you introduce yourself, please?
Yes, I'm Caroline Orish-Benichou. I'm a clinical assessor in the Icelandic Medicines Agency and a member of the Scientific Advice Working Party at the EMA and the chairperson of the Immunoinflammatory Working Party in charge of drafting guidelines. And we have plans to update the guideline on SLE in 2028, when data will be available from new drug developments for products that we have seen in PRIME and Scientific Advice, including recently several CAR T cells. Thanks.
Thank you.
So Chloe, this question's for you. What are the big ways lupus affects your daily life that clinicians, researchers, and clinical trials are not addressing?
Honestly, now I'm very well managed, but there are like certain instances where reaching out to my provider, I have an excellent provider, as I've mentioned before, but I would feel more confident in research showing that there's a treatment that's safe and effective within a group of people that are similar in my age or similar symptoms to lupus. I believe in like having my doctor have solid explanations as to why she believes that the right course of treatment is right for me. I like to be made aware of benefits that would come from specific treatments, such as like in like negative side effects. How do we monitor if it's working, if it's not, to me in an accessible way for me, like not going once every 2 weeks, where we can make virtual appointments to discuss, et cetera. I would also, sometimes it's hard to make adjustments in your treatment plan. And I think having that available would be beneficial.
Great, thank you.
So we've already, going through this, you'll see that the questions for this session, we've had quite a bit of discussion about them, but not heard from these particular panelists. So if we start with the first one, is there, everybody can see them, right? There's no great reason for me to just read that out. Is there anybody who would like to add to the discussion we've had? Oh, I'm sorry, Caroline.
Yes.
Thank you.
Yes. In answer to this, I would say regarding the gaps or strengths of evidence regarding the pharmacology, for example, and targets, I think that there is quite a high level of evidence that the targets of efficacy of a drug would be sufficiently similar. However, we have not at all discussed, and I think it is also important, the targets of, there is a high level, so there is a low level of uncertainty as regard targets for efficacy, but as regard targets for safety of especially new products with new mechanism of action. I was also thinking of the CAR T cells. There is a high level of uncertainty or for other mechanism of action, whether the targets for safety or for the toxicity of drugs would be sufficiently similar. And to me, this is still a knowledge gap. And regarding response to treatment, we have always similarly spoken of efficacy response, but not of safety response. And I think that it is important. I remember some panelists explaining that perhaps it would be better to have just one dose that works instead of an optimized dose. But an optimized dose means both the highest possible efficacy, but with an acceptable level of safety. And perhaps it is answer to the next question. I could speak again on what I think would be a way to address these gaps, but probably, as we have said, exposure response modeling. And I mean, perhaps just exposure matching couldn't be sufficient if there is more aggressive disease, more difficult to treat, and then exposure response modeling, as we have discussed in the MIDD session, would be really important, but it should be both exposure efficacy response and exposure safety. And otherwise, I regard also this difference in response to treatment. My understanding and what we knew and what have been discussed all along is that the only important key example that we have is belimumab. And then I think it's, I'm not the only one to raise this issue, but when we saw the comparative data from pediatric and adult belimumab studies, we saw that there is a similar response to treatment. But I was also surprised to see that we have similar baseline characteristics. And knowing that generally in the pediatric population, the disease activity is higher, that we have more severe disease, that we have neuropsychiatric features and hematologic features that we rarely see in adults. My questioning was whether the pediatric population in the belimumab development was sufficiently representative of the whole disease spectrum and the whole population that would be candidate to a new drug, and especially of the most severe one, who are the patients with the highest unmet need. So just to say that it's very reassuring that we saw this similar efficacy response, but that there is, to me, remaining uncertainty. And that's why I would again insist that perhaps exposure response modeling, starting from the beginning with exposure response collected in adults to base the building of a model would be perhaps a key thing that would allow, as was already said, minimizing the number of patients involved in a trial designed to collect high quality efficacy response data. Thank you.
Romina?
Caroline, I have a quick question. You were concerned about differential safety between children and adults with lupus. How much confidence would the enrollment of 93 patients give you that the safety in children and adults is the same?
Oh.
Or different?
No. I think that in the belimumab trials, we have not seen major safety differences. But what we know also now, belimumab is registered in children since a few years, and still children with pediatric lupus have great difficulties to achieve low disease activity and remission, and to maintain this remission once achieved. And so we need new drug. And probably this is the, when we say they have a more difficult, more aggressive disease, more difficult to treat, this might be solved either by perhaps an increased dose insofar as the safety allows it. But it's also perhaps illustrate the need for other new drugs with new mechanism of action. And these CAR T cells are really promising based on the few data that we have seen in prime designation that are that are really promising. And then we have no idea to what extent the safety would be similar for CAR T cells. But there might also arrive new products with new mechanism of action during the development, for which we don't know whether the target of toxicity would be the same in adults and children. It's not only the maturation of the immune system, but for liver toxicity, it is the maturation of the liver. Perhaps I'm wrong, I thought it was around 8 years of age. Some other targets might be fully mature later. I was thinking of pulmonary, the development of the big bronchial arteries, bronchial. So there are organs who are not fully matured before a later age than 5 years of age. That was what I was meaning. I was not speaking of the belimumab issue, for which we have relative reassurance, I would say. Thank you.
Just for clarification, this session is specifically focused on extrapolation of efficacy, and we're going to have additional discussion related to safety tomorrow.
So what I have heard, from very elegant presentation is that based on the data we have, that the exposure response in children with lupus over the age of 5 is comparable, no matter how you get to that statement, either by modeling or clinical experience, off-label use, registry use. So we cannot find, so we have no evidence that it's different up to now. Can we say for sure that it's never going to be different? No. But there is no evidence to the contrary. So if something doesn't exist, how can you how can you argue that it could exist? If there is no precedent to it, why would you suddenly expect that there's a complete shift in that observation? It would make no sense.
Rod? Yes. So I kind of want to go back to one of Lily's slides from the very beginning about extrapolation and the concept of extrapolation. Extrapolation does not mean exact disease. It doesn't mean that you have to have the exact disease in pediatrics and adults. So it's okay to have some additional questions But it's a spectrum, a continuum. And so basically, how related is a pediatric disease to an adult disease, and what are the gaps, and what is it that you need to have to fill those outstanding gaps? So I just want to make that clear.
And I think that's great because as pediatric rheumatologists, we have diseases for which there is a big difference, right? We would not say that JIA and rheumatoid arthritis are the same disease. There are some subtypes that are diametrically different in their manifestations and their response to therapies. But lupus is one of the ones where we so far have seen total overlap.
One that's well supported by the scientific community. We use the same classification criteria. We use the same, response to therapy assessments, the same activity and damage measures. So everybody realizes that really we are dealing with the same disease. And one does, when is a pediatric, I dare to say that most of the children with lupus, they grow up to be adults. So when they are adults, then suddenly they get access to medication they wouldn't have gotten. So if we are really consistent, wouldn't we say that an adult with lupus who had early onset of disease, they shouldn't be exposed to dangerous medication because they may have a different disease?
Lily, I think you have a question? But we don't know who they.
Limiting the discussion to the panelists just to get the conversation focused, but I just wanted to build up on a point that Caroline made earlier, that exposure matching may not be appropriate, and hear from clinicians. Do you see a need to evaluate exposure levels that are higher than exposures that have been evaluated in adults and have been established to be safe and effective? And if so, in what scenario?
That was my point. It was not at all that the disease would be different or that the criteria would be different. It was just we have seen that it's not because we have seen that we have only the example of belimumab, which is one mechanism of action. And we cannot be sure that for other targets, that for other mechanism of action, or just because the disease is more aggressive, perhaps we wouldn't need higher doses to achieve low disease activity of remission, now that we know is the treat to target paradigm is the goal of therapy. Of course, and that's why I just said that perhaps exposure matching could not be sufficient. It could lead perhaps to what have been said, something that works, but we would never know whether we could achieve a higher rate of patients, higher proportion of patients with low disease activity or remission, and a higher proportion of patients with sustained low disease activity and remission. And because all the damage that are made during childhood and adolescence will also make a more severe disease in adulthood, this is important to prevent damage, to prevent disease progression and achieve low disease activity and remission. And that's just why I said we are not sure that exposure matching would allow the same thing, specifically when we see that despite belimumab, still a significant proportion of children do not achieve that or do not maintain this low disease activity and remission. And so my proposal was just to emphasize what has been said on the importance of exposure response modeling, which would allow modeling that to have an idea of what kind of dose. It's not mandatorily that a higher dose would be needed, but we would have an idea of that. And it should be, and I agree this will be session 4, also it should be accompanied with an exposure safety response analysis, because ultimately marketing authorization will be based on a favorable benefit-risk ratio. So the optimal dose should be, as I said, the one providing the higher level of efficacy with an acceptable level of safety concern. That would be my answer. Thank you.
for me. So the
Should we go to
question-
the audience,
Oh,
perhaps?
Okay, how to?
You can use this if you can't. Okay.
So I want to address the exposure matching question about Caroline. So I would say in clinical study, it's always ideal to have matching exposure to adult in pediatric studies. In clinical practice, maybe you can have higher or lower dose compared to adult. But for these reasons, like for exposure matching, if we do extrapolation, obviously you have to have similar exposure or higher to extrapolate efficacy. But even for efficacy safety study, you heard about the Bayesian approach. And the basis for that also based on similar systemic exposure. So if we have a lower exposure, lower dose in pediatrics, it will be very challenging in clinical studies to prove efficacy. And on the other side, the higher exposure. So I heard all the, yes, I agree that pediatric disease is more severe and there will be situations, maybe some drugs, some scenarios, the patient may benefit from higher dose. But again, it's very hard and challenging to build in clinical studies because for the higher dose, you would need to establish the safety database based on pediatric alone. And that sample set would be very hard to establish a meaningful safety database. And also for higher dose, if you think of the label, they have a dose, high dose for 17-year-old and the low dose for 18-year-old. So it creates an awkward situation in clinical management. So I would acknowledge that patient may need higher or lower dose than adult, but I would leave that individualized therapy to clinicians and to, with the extrapolation, we can get the medications sooner, more accessible to patients.
Stacy?
Thanks to the panelists of this great discussion. You know, we all know that the age of 18 is a bit arbitrary. Just, you know, could be 17 and 365 and then the next day be eligible for a drug. But I was wondering, you know, of course, the changes in the body are a spectrum, and we also have much older patients who have declining organ function, but we don't have regulatory exception for them. And I was curious from our regulatory colleagues, you know, how do you think this applies across the lifespan? So we don't have different standards for much older patients who are often excluded for these studies. They still have access to trials. How does that factor into your thinking with the pediatric population? They may have similar changes that we're not capturing in clinical trials. And how do we capture their safety concerns? And should there be different regulatory requirements for them?
Is that for me, I guess?
Yeah, the only regulatory
So
question on the panel.
I hear your point. But I guess one thing I would say is that where we do know there are potential changes in organ, whether it be liver function or kidney, we do have kind of some stipulations there on how we adjust dosing based on liver function, kidney function. And I think it's up to the pediatric community as far as what's important, as far as organ development, growth, all those type of things. What is acceptable risk when we're talking about these medications for lupus?
Raj, I'm sorry, just to expand on what you said, in those subpopulation with organ impairment or elderly, we would adjust the dose, but we would assume that the efficacy and safety would apply otherwise, right? Assuming the dose can be
Yes, that's correct.
I'm not a panelist, but I'll, you know, throw myself into the discussion here because I keep hearing this, you know, consistent concern about the differences in severity in pediatric versus adult patients. And I think when we talk about extrapolation, we should probably, you know, have this discussion, you know, before we get to the pediatric programs. In other words, should we study severe, I'm talking about really severe patients, adult patients first, to get all the information that we need to extrapolate to pediatrics, rather than try to use the pediatric programs to address questions that, you know, we didn't really address earlier. So in essence, I would advocate for us and, you know, industry and all partners to think about us, you know, now that we have the landscape of lupus treatments really expanding, to think about opening inclusion enrollment criteria in the adult trials, the adult studies, where we can study all of these more severe patients and have the information that we need to extrapolate, and for pediatric patients with more severe disease to actually have confidence that these drugs would work in those. Again, rather than try to, do this, later in development, in pediatric development.
That gets us more into subsets, right? Because most of the most severe lupus patients, both the adults and the pediatric patients, have nephritis or they have they have organ system disease. They don't have just the general lupus.
Right, and I love the points that you're
Yes.
raising, and suppose I would think of it in terms of not a separate trial of severe disease, but the ability to stratify from our existing trials and look at the patients who have the most severe versus patients who have more moderate disease, and think about some of the questions that were already raised about if somebody has a more severe disease and has partially responded, might that be the patient for whom a higher dose might potentially be beneficial? But I think the stratified analysis makes a lot of sense, and would hate to see, however, additional trials that just kind of delay approval overall.
Hi, Nidhi Goel. I just wanted to ask a question related to the development in pediatrics, and that, Hermione very well summarized the aspects related to that these tend to mimic the adult trials. How much of that is from a regulatory perspective that they have to mimic the adult trials versus if someone came forward with different trial designs addressing the specifics of the pediatric population, because we're extrapolating, would that be permitted or not? I'm just posing that as a potential question to change potentially what we are studying in pediatrics.
Or changing the adult trials so that they more conform to what's necessary to be able to do the pediatric extrapolation. Personally, I think, I guess I'm the moderator, I don't know if I'm allowed to have a platform, but that's been that's my approach that I think is most important is just that the adult trials from the very, very beginning, even before you know whether they're going to work, because you need to be able to collect the data that's necessary to address, as you pointed out, but to address the severe patients, but also to make the extrapolation more effective. Raj, sorry.
Sure. So I do agree with what you're saying here, is that it would be, in an ideal world, we do get all our answers in the adult population, that we can kind of use that information in pediatrics. And part of the reason for this is because we do require a prospect of direct benefit before we do studies in pediatrics. And I think that's something that we do struggle with on occasion when there's a proposal to study a severe subset where there is no adult data or nothing to kind of direct us that these patients will get a benefit. So that's incredibly important. And so if we can use an adult model, I think that's helpful.
Yeah, I just wanted to raise this this point about, the severity of disease and the fact, and I guess I wanted to get your opinion on this construct that we've kind of built for ourselves, in that we study lupus nephritis as a separate indication from SLE, and especially thinking about the kids, so many of these kids have kidney involvement. So why can't we just study SLE, including kidney involvement?
Well, I do agree that we should not do organ-specific study. It goes even further. When you look at current medication that are in development, they work especially for for skin and for joint disease. So do we then demand an extra skin joint study? And then there may also be that there's a cardiovascular risk. Then in children, we ask for another vasculitis study. So it's not tenable. And up to 80% of patients with lupus will have kidney involvement, depending on the ethnic and racial contribution, and a good part of them have CNS involvement. So parsing out that little segment who only has the one entity and not the other is virtually impossible.
I would agree that it's really a significant barrier in pediatric lupus trial recruitment to not be able to include children with nephritis. Even if we saw some estimates, the proportion isn't all that different. But, being as a practicing pediatric rheumatologist, even if only 40% of my patients had lupus nephritis, I worried about 100% of their kidneys. So I think we really see that when we're trying to recruit into a study where we don't yet have adult data on lupus nephritis efficacy. It's very challenging because clinicians and I think patients and parents are often rightly hesitant to move forward when we don't have all of that data. So I think as that can be moved into more integrated earlier programs for adult drugs where appropriate, and it may not be one size fits all because sometimes from early stage development or phase 2, you may have more or less data on whether you anticipate that proteinuria or other drug metabolism differences will cause a large dosing difference. So it's not always possible for products where there is different dosing, but I do think that studying, as Dr. Brunner says, the different organs differently is really, really challenging when someone's considering the totality of manifestations in one of their patients. And I think this is something that we as a field will have to think about more and more as we move into some of these advanced and cellular therapies where we're having the discussion of how sick is sick enough, because most of the time I think that will involve nephritis for a lot of our younger patients.
So one more thing I just kind of want to put out there when we're talking about SLE versus lupus nephritis is that oftentimes initial treatment's very different for a patient that presents with lupus nephritis versus general SLE, at least in adults. But I would think that probably is true in pediatrics as well. And so I guess my question would be how to put that type of information together in a trial.
Well, one way you're going to stratify them is by disease severity, because once the disease severity gets past just probably past 6, they are going to have organ system disease. But another concern that that I have is that by excluding some of these subsets, we're excluding certain groups of our patients that have so different social determinants of health and stuff. And so it doesn't end up sort of being open to all, but we are supposed to be done, but should I let these two folks?
I just want to make one quick comment. And I think it's Stacy, your comment about, well, what about older people? And we don't ask for efficacy and safety. So you remember at the beginning, we sort of started out the idea that, children are different. And that whole notion, if you're a pediatrician out there, how many times have we said in our careers, children aren't just little adults, right? But as soon as we decide that we agree extrapolation, and certainly extrapolation to the extent that we are willing to accept PK matching and safety, guess what? We're saying children are just little adults. So the point here is that we just want to be really sure that we have the data to support that there are so many similarities, because we are, at the end of the day, going to say children, in this case, with lupus, and I think I've heard a lot of information to support that, in many situations are actually just little adults.
So if I may, so the same treatment guidelines are used for children and adults with general lupus and children and adults with lupus nephritis as per the recent guidelines. So if a patient, irrespective of age, has certain disease manifestations, they are advised to get similar therapies.
Let me clarify. So what I was trying to say is that the initial treatment in a lupus nephritis patient, regardless of age, would be different than someone who has SLE, regardless of age. So, like if you're getting cyclophosphamide, rituximab in nephritis, but then you're starting a general SLE trial where you're not starting with those treatments, then it's hard to use all of that data together. That's, I think, more of what my point is, and you have different endpoints. All these type of things make it a little bit more difficult to use all the data together and make an interpretation, is what I was trying to say. Not that I feel like it's different in adults and children. That I think that's kind of what I was trying to get at.
And it's also, true that it's because of that. I mean, we allow patients with lupus nephritis. We don't exclude them, all the time. We only exclude certain patient population, which is, actively needed something else, right? So those are the distinction. And it's not that different for pediatric versus, the only difference maybe for pediatric patients may need maybe more prompt therapy, because of the damage concerns and etc. But other than that, I do agree that we are excluding all lupus nephritis patients from the trials.
So, do you want me to cut? It's okay, keep going? Okay.
I was just going to add the caveat that it's, in my mind, the paradigm for treating lupus nephritis is not fundamentally different from other severe lupus. It's more the severity that directs things. So when we talk about cyclophosphamide or mycophenolate, which I certainly would be using up front for new lupus nephritis, I'd also be using it up front for severe vasculitis from lupus and other severe manifestations.
Yes,
Yeah,
Chloe, go.
Can I add something as someone who has SLE and lupus nephritis? When I was first diagnosed with regular SLE, I had the high protein and the kidney function, and we actually were able to use the same medication to not only treat regular SLE, but the lupus nephritis as well. I was taking CellCept, the mycophenolate.
Yeah, I think if I think generally here, if I can just sort of summarize, it seems to me that the pediatric rheumatologists in the room are most adamant about the fact that we need access to new drugs, and that the extrapolation is okay. Now, it's important for us to hear more from our patients about but because the trade-off from the physician perspective is that we're willing to go with less optimized dosing because we know that we need the drugs. And if we can work on some of that to optimize the drug after the drugs are available for us to be able to use. Now, safety we're going to talk about tomorrow, and that's different. But I think I haven't heard a pediatric rheumatologist trying to pour water on. Trying to stop the movement towards.
Can I say something? It's Roberto here. Hello.
Yeah.
Hello, because I had my hand raised for a long time, and just appreciate the willingness to contribute despite being 11 in the night in the EU side. So I would like to reiterate my concept that extrapolation is not a yes or no decision. It needs to be a structured decision, and it's also not a qualitative evaluation. It's not because Roberto De Lisa or Hermione Brunner says we can extrapolate. No, it should be pediatric patient needs to have data analyzed. An extrapolation plan is an analysis of data, is an analysis of exposure response. They need to take into account the evidence that is available in adults, meaning how many young patients were recruited in the adult trial, how many of them have a certain severity, how many of them had a certain disease onset or faster onset compared to younger patients. And not only that, they deserve to have a treatment that is as effective as safe as for adults. And we need to demonstrate to patients that this was not only assumptions, but that these assumptions are confirmed. And we can only do that by analyzing for each drug what is the safety effect, what are the evidence in juvenile animal studies, what are the many adult developments have failed. Rituximab itself that we have mentioned few times, Explorer trial, Lunar trial, they failed. There could have been reasons for that. I agree with you. We need to look into that. But also abatacept, epratuximab. We are talking about including adolescents in adult patients. We are disregarding the fact that sometimes efficacy is really limited. So we need in advance to know how much we would accept a difference in the exposure response, because it's not going to be the same, I can assure you. It's going to difference in something. We have mentioned the belimumab example, and the belimumab, we have stressed a few times that the inclusion criteria were the same, so we had more or less the same severity. We didn't mention, though, in the slides that there was a higher response in the placebo arm in that trial. Sorry for being a little bit the party pooper here, but really extrapolation needs to be backed up by data and by analysis. If even we get to an indication, there will be down the line other bodies that will decide not to reimburse the medication because the evidence is not there. Thank you.
Thank you. I think we all agree with your final statement there that extrapolation needs to be taken on a case-by-case basis and be supported by evidence. I'm sure we can all agree on that. Thank you.
And if I may, Caroline Orish, if I may add something, what I have developed a little bit on exposure response modeling. It is something that is fully an extrapolation strategy, and that capitalize on benefit risk, favorable benefit risk already demonstrated in adults. That's why, contrary to what I have heard, it is not an issue of mimicking adult studies at all. It is an issue of saving a small exposure response, a small study, perhaps placebo not needed, an exposure, a small study to collect exposure response data, allowing to see whether exposure response model built in adult needs or not to be refined. And this strategy studies designed especially to collect high quality exposure response data within a model is a study that requires much less patient than a classical RCT or something like that. So it is fully an extrapolation. It doesn't mandatorily require a placebo. Sample size needed is less. You don't have this issue of power. And the same, if you want to look separately at exposure response in lupus nephritis or in the pure cutaneous form, you don't need to have had a sufficient number of adult patients in each stratum to have statistical significance. What you need just to have collected sufficient high quality exposure response data to build a model, to update it, to refine it in children if needed. And this is, I think that my colleague Flora explained it, this just needs, starting in adult, to have the condition for good exposure response, collection of data, adapting the sample size, but it's often much less, adapting the number and timing of samplings and things like that. It is pure extrapolation, and this should help. So that was what I wanted to add. Thank you.
Thank you.
I just wanted to point out that there's a bit of an irony in the world of extrapolation, which is that even if there is the perfect randomized controlled study that enrolls 500 adult patients and gives us excellent data, we've never seen a response rate of 100%, right? So we have that trial, and then I see an adult patient with lupus, and I am still trying to extrapolate from that trial for the patient in front of me about the likelihood that they will respond in the same way that the responders in the trial did. So we can never get away from elements of extrapolation.
Thanks, Rebecca.
Closing remarks.
Thank you so much to our panelists, and I'm going to turn it over to Suzette for closing remarks for today.
All right. Oh, yeah, sure. So in closing, we've had an incredibly productive day one. I want to thank all the speakers, panelists, and moderators for their expertise and contribution to today's informative and collaborative discussions. I especially want to thank the patients and caregivers who took their time to share their lived experience. Your insight has been thoughtful, astute, and so valuable in our discussions on drug development in children with lupus. What I heard today was the need from you was the need for access to treatments and more options for children, the need for drugs that address invisible symptoms like lupus brain fog, and the need for education and transparency of the effects of therapy on things that matter to children. So things like school and athletics and your social life and just being able to be a child. So thank you, Mckenna, Mia, Elizabeth, Chloe, and Nia. Thank you for being here today. I also want to thank our regulatory counterparts with the EMA who are attending from Europe and very late in their day and joining in these discussions with a shared goal to expedite drug development in pediatric lupus. And the sessions today provided many key lessons, but I want to highlight 3 things that stood out. So one, we heard that although disease severity may differ in the younger age groups, lupus is the same fundamental disease in childhood and adulthood with similar pathogenesis and pathophysiology, as well as overlapping disease manifestations. The same classification criteria and disease activity measures are used. The data show that exposure response is also similar with most drugs, but the data are limited. From academia, industry, and patients, we heard that there are many obstacles and challenges to what we are currently doing, that is placebo-controlled trials, and even placebo as add-on to standard of care. And lastly, there remains gaps in knowledge, such as differences in drug metabolism and specific safety consideration, and then the lack of data in subsets of lupus, specifically neuropsychiatric lupus and lupus nephritis. So where do we go from here? With everything we've heard today and then a discussion on safety tomorrow, we hope to tie everything together tomorrow and to consider how to address any remaining data gaps and what types of trials are feasible in pediatric lupus. And as Mckenna said so well this morning, change is good. And with all the stakeholders here working and talking together, this is how change happens. So we hope we're able to make a change that improves accessibility of safe and effective therapies to children with lupus. So thank you all for your time and attention today, and we look forward to seeing you all bright and early tomorrow.